Good morning and welcome back to this second day of the Open Session 2022. I would like to start thanking the Boehringer Ingelheim for the dinner that has been offered yesterday. I also would like to thank the speakers, panelists, moderators of the yesterday sessions, and also to thank the participation that we had yesterday online. Yesterday we had 900 colleagues that connected through YouTube, which is very good and give the sense of the innovation and digital technologies, as I said yesterday, that brings together physically and virtually researchers and policymakers. Today, as yesterday, we will start with a presentation on fast diseases. And the keynote speakers is Andrea Apolloni, who is a mathematical modeler of infectious diseases. And Andrea, since 2015, works with CIRAD, where he develops mathematical models for transboundary animal diseases, combining livestock mobility, epidemiological dynamics, environmental and social factors using qualitative and quantitative models. Andrea will present an overview of the PPR and Rift Valley fever distribution and recent events occurred globally in the last years. Thank you. Andrea, the floor is yours.
Good morning, everyone. Oh, that's cool. Okay, what I'm going to present is the work mostly done by many other colleagues of mine at CIRAD, in particular Arnaud Bataille, Véronique Chevalier, and Alexandre Drouin, who is a PhD student of Véronique Chevalier, about the recent event of peste des petits ruminants and Rift Valley fever. So the plan of the talk is almost specular. I'm going to introduce some notion about Rift Valley fever, peste des petits ruminants, and the situation in 2020-2022, and what is the risk of introduction of these two diseases in Europe. And then there is some more specific work about the competencies of some vector in the Mediterranean area, and for the PPR case, the status in nearby country, nearby area of Eurasia. So starting from Rift Valley fever, just few points. Rift Valley fever virus belongs to the phlebovirus genus. Is transmitted through the bite of mosquito, but also through close contact with infected tissue to infected tissue, and could affect small and large ruminant, some wild ruminants and the humans. The consequences, the outcome of the infection could result in mortality and abortion in animals. And for a human, it's mostly asymptomatic, but can have some impact as hemorrhagic fever in a few cases that could also lead to death. Now, as I say, the virus could be transmitted either by infected vectors that belongs to different genus, and through close contact with infected tissue. However, one of the most important way of diffusion, geographical diffusion, is through the movement of either animals or humans, either because of trade or, in the case of human, because of travels. Now, the first case of Rift Valley fever were discovered in 1930 in Kenya, and since then the other outbreaks have been reported in sub-Saharan Africa, in particular during the El Nino in South Africa, but also in 2000, few cases were detected in the Arabian Peninsula. Now, although the disease appeared in Libya in 2019, 2020, and also there are some cases appearing in Tunisia, the disease has not yet appeared in Europe. And the risk of introduction has been considered by EFSA very, very low, something like 1 probability in 500 years. So this is the introduction by the animal movement, because there is still a ban between the North Africa and the European area. However, the movement from Africa to Peninsula, as you can see in this map, there is some movement that could explain the introduction of the disease in the Arabian Peninsula. And another important point is that in the sub-Saharan Africa, there is a lot of movements that are undetected, or they are not undetected, that are illegal or not well regulated, that could explain the spread of the disease in sub-Saharan Africa. Now, five competent mosquitoes are supposed to be possible vectors for the introduction in the Mediterranean area. And these are the Aedes caspius, the Tritus vexan, and two belonging to the Culex, the Pipiens and the Teileri. So these five competent mosquitoes can be found also in the Mediterranean area. Now, if I'm going just to have a look what happened in 2020, 2022, most of the outbreaks of Rift Valley fever were concentrated in Africa. It appears to be just an African problem, and mostly among the domestic ruminants. Very few were detected in wildlife, as you can see here in this plot. And 15 countries were interested by Rift Valley fever outbreaks. And if I try again to study what is the risk that infected animals could enter in Europe and then bring a Rift Valley fever, data from UN Comtrade there are no data in the UN Comtrade that could support the introduction by legal trade to Europe. So the other important way that the disease could be introduced in the Mediterranean area, in the European side of the Mediterranean area, is through the presence of the potential vectors. So this work is the work done mostly by Alex, and studied the competence of 5 species that I told you before, the 3 Aedes and the 2 Culex. They could explain the presence or assess the risk of introduction of Rift Valley fever. So in this work, they consider in this work, they consider 5 different outcomes related to mosquitoes. And it was the infection rate, means was the virus found in the gut of the mosquitoes, was then the dissemination rate, was the virus found in other area of the mosquitoes, like legs and wings. And finally, the other one was the transmission rate, meaning was the virus found in the salivary glandular. And to assess this outcome, they did a meta-analysis. They consider around more than 500 publications in Scopus and PubMed. And after review, they come out with just 34 different studies. And what they tried to understand was to assess what is the risk for these 5 outcomes to be found in the different species, and try to understand how these were influenced by the different studies and by the different experimental design. The results found that among the 5 possible competent species the 5 species were found as possible competent: Caspius, the Tritus, the Vexan, and the 2 Culex. And among them, the Caspius was the one with a higher rate in all the 5 outcomes. It means that it's been the most competent vector for the transmission and for the infection and the transmission of Rift Valley fever. Another that is quite interesting is the third one, Teileri, that appears to have a high infection rate, but unfortunately there are not so many information about the other factor. So it's still uncertain what is the role in terms of competency. Now, once we know what are the possible competent vectors, what Alex and Veronique mostly are trying to do is to try to assess what is the presence during time in the Mediterranean area, in particular in south of France. So what they do is that they consider a mathematical model for population, for vector population, whose parameters depends on temperature and rainfall, in the area. I tried to develop this model here to assess the quantity of adult female, either nulliparous or parous, and osteoseeking, that could explain, and it could increase the risk of transmission in the Mediterranean area. From this one, the next step would be to add some transmission probability, assess the risk for R0, assess the estimation for R0, for the Rift Valley fever in south of France. So this is just an idea of what they are doing. They are considering rainfall and temperature in different areas for each pixel of the European side of the Mediterranean. Here you can see the variation along time of the two predictors, and then inform the model that I showed you before. And this is going to give you an idea for each species of the aquatic stage, but also the adult one during the year. And from this one, it will be possible to assess the risk of transmission and endemization of the disease in the different area. Now let's keep disease. Let's pass to another disease. And now I'm going to present something about PPR. So PPR is another RNA virus belonging to the family of morbillivirus. So it's the same family of rinderpest and the human measles. As the name say, this disease affects mostly domestic small ruminants, goat and sheep, but also there are some other components like the wildlife circle with other ungulates. Bovines are supposed to experience a serological response, although they are not going to transmit the disease. While experimental infection has found that pork could be infected, infect from PPR. The transmission is a direct transmission, so there is no vector involved. Doesn't affect the human directly. May affect only from the economical point of view. This disease, in particular in sub-Saharan Africa, has a seasonal character that depends on the demographic trends, but also depends on the movement of animals. As animal moves, as in the case of Rift Valley fever, the disease could spread in a new area that are not infected. And in particular, these movements are really seasonal. Depends on the condition that drives to the transhumance one, but also depend on some human activity, like religious festivity, that demands high consumption of the disease. So initially it was discovered in Ivory Coast in 1942, and then in 2010 appeared in China, and in 2018 it started appearing also in Georgia and Bulgaria. The disease is currently under eradication, supposed by 2030, because there is a vaccine that is quite effective. So if I look in 2020-22, there are 13 countries in Africa, 11 in Asia that has experienced some outbreaks of PPR. And as you can see, the number of outbreaks and the number of cases varies between the two continents. And in this case, most of the cases were detected in domestic ruminants, although the wildlife start becoming quite important. Now, taking the data from UN Comtrade, we try to understand what could be possible introduction path, and it appears that Bulgaria is quite a not spot, since it's connected with many other European countries, and also there are some movement from Bulgaria to Turkey that also could affect Cyprus and Ireland that are quite far. At the same time, Germany, it appears to be exposed to other areas that has recently experienced outbreak of PPR, like Egypt and Nigeria. However, this is a real partial one, because there is all the movement in sub-Saharan Africa and all the movement between the countries that should be considered more in details to have an idea about the introduction and diffusion of the disease. Now, what I'm going to present briefly is some report about the status in Eurasia. This has been some result from the roadmap meetings that take place in 2017, 2019, and 2021 in this area. And in this case, there are many countries that are involved, 13 countries that goes from China to Turkey, and also consider Russia to the north and many former Soviet republics like Georgia, Tajikistan, Uzbekistan, and Turkmenistan, Kyrgyzstan, and Kazakhstan. So the case from the epidemiological point of view is quite different because we have countries like Mongolia, China, Iran, and Turkey that are experiencing currently the disease in the with their current outbreaks, while Tajikistan and Georgia, they haven't reported any outbreak since 2016 and 2013, while in many other countries, no PPR, the PPR has never been reported, although for some of them, there seems to be a circulation that has been undetected. In most of these countries, vaccination takes place at different levels, but also some surveillance and targeted surveillance in this area, in particular in the buffer zone in the border area. So just resuming, it appears that in Iran, that is doing a lot of vaccination, PPR is still circulating, but it is mostly due to the wildlife cycle, to wild goats and sheep. In Tajikistan, PPR is probably still endemic in the country, although there's no be reported. And one of the points that has been raised is that many stakeholders are not involved in the plan for the eradication. There is the need, this is something like all the representatives at the roadmap meeting say that there is a lack of awareness of the different stakeholders that could make the difference on the application of the eradication strategy. And that's it. I have still 4 minutes, or?
You have 4 minutes for answering questions.
Okay.
Thank you, Andrea. Thank you very much for the presentation, also for the work that you have been, that you are doing with CIRAD for EuFMD, and in particular on assessing the animal mobility and connected risk, particularly in North Africa, Middle East, and Southeast European neighborhood. Do you have any question? I see Vasily from Georgia.
Hello. Yes, good morning. I just have a comment. You mentioned Georgia, and just would like to give you some more detailed information. Last outbreak in Georgia was in 2016, and since 2016, Georgia vaccinates the entire population in the country. And when you mentioned the other examples that other countries vaccinate and it still circulates, I believe that it's all about the quality of vaccine, and after quality, it's all about the cold chain, how you store the vaccine and how you use the vaccines. Last year, we did a scientific project, and we researched 3,200 animals. It was a DITRA-funded project, and so far, so good. Just a little update. Thank you.
Thank you.
Thank you. Are there any other questions? On the back, Don King.
Yeah, thanks very much for the presentation. I'm just wondering about the risks that might be associated with particularly rift coming into North African countries. I mean, if we think about FMD, if you remember from the presentation yesterday, we see increasing movements of presumably animals from West Africa into countries in the Maghreb. Do you see any potential for that to also change the risk profile for rift coming into that region?
Can you repeat better the question? Because there is a lot of movement. So this is going for PPR. We have seen flow in both sides. We have movement from Tunisia to Nigeria, from Nigeria back to Tunisia. So the risk from sub-Saharan Africa to the north of Africa and also to Middle East is quite great for both the disease. The problem is just the movement are not so well regulated outside countries. Some countries like Senegal, they have quite a nice system for mapping the movements. Outside, they need just some ad hoc activity to study the movement. And there are two other points. One is the commercial movements. So we have, like, for example, for the religious festivity, we have a huge amount of animals that move in a short period of time. And although they are quite regular, every year they shift for a certain amount of time. So every year is something like 2 or 3 weeks before they shift animal. Second point is transhumance. That is the other, that is animals going, looking for better grazing area, and they cover thousands of kilometers, either from north to south, going from the area more desertic to the greener one. And what happened is that in the last years, with rainfall and temperature, rainfall decreasing and temperature increasing, this movement become more, they accelerated this movement. They start earlier and take more time. So it's going to increase, in Africa, it's going to increase the risk of transmission. Now, till now, the problem is that we don't have many information about from Africa to Europe. When we ask to present something, it was something like, what is the risk of introduction in Europe? So from Africa to Europe, it appears from the Comtrade data set that there are no fluxes indirectly. Another important point is about wildlife that has not been taken account. And this one is going to be a huge problem because also wild animals transhum. Although this is even more unregulated, this is going to change a lot, both in Africa, both at the border between Turkey and Europe also, and in the Anatolian part of Turkey.
We have other last 2 questions. First, Aldo Dekker.
I was wondering in the model on Rift Valley fever, I always understood that in the Rift Valley in Africa, that especially abundance of mosquitoes is the main driver for transmission. The idea is that there's low levels of transmission which does not cause much clinical disease, but only when mosquitoes are more present, animals are exposed to higher doses of virus by multiple bites, then so that's an important issue also for the risk for Europe. If we don't get that abundance that happens in Africa, then the consequences, which is part of the risk, are lower. Or am I mistaken?
Okay, I'm not really expert on Rift Valley fever. The only thing that I could say is that during the last outbreak in Senegal, we found a lot of outbreaks in areas far from the mosquitoes one. So this means that the main in urban area where the density of mosquito was quite low compared to the historical outbreaks area in the north of Senegal. So this means that, is it true, mosquito has a role in the transmission, but mobility is getting more and more important for the dissemination of the disease. This is the main point. And there are some other models some models, mathematical models for other vector-borne disease that also say support this hypothesis. And that the role of mobility is taking a lot of a more important weight compared to the mosquitoes one. Because
we have one last question here in the front, Giancarlo.
Thank you. Thank you, Andrea, for your very informative presentation. In relation to PPR, I think you have mentioned that the lack of engagement of the stakeholders was identified as one of the major obstacles, let's say, to the implementation of the eradication program. My comment is that I think there is also another important point is access to animal health service, especially in marginalized communities. And I think these things are going to be somehow addressed with the Global Eradication Program 2, which will be officially announced on 4th of November, I think, if I remember correctly. My question is, if you are currently working on developing any sort of model for transmission and spreading of PPR that may possibly assist the implementation of the Global Eradication Program 2, as you recall, the objective is reaching eradication by 2030. Yeah.
A quick answer, please.
Yeah, okay. Yes, and we are doing in a different, I would say, layer, because we have some numerical, mathematical model taking account both mobility and transmission. But at the same time, we are working with local community and doing what is called companion modeling. And we try to engage a local community to better understand how the vaccine work, what is the immunity, but also what are the importance of other intervention, like identifying animals, sensing, surveying how many animals there are in the area. And this is something like that is not a mathematical model. It's something built with the local actors. And one of the points is that the local actors say, oh, we would like to vaccinate, but like, for example, the vaccine arrived too late for us. And we would like to vaccinate. We have no problem, because we say, oh, the price is correct. We can handle this. The problem is that the vaccine is not available at the right moment. It's not enough, arrived too late. And also the fact that they have to revaccinate every year the same animals. Now they discovered that vaccine could confer almost lifelong immunity, so there is no need. These were information that they didn't know before. And we try as much as we can to put everything together.
Thank you very much, Andrea. Thanks for our first keynote speakers of today. We have received also some questions online, but we don't have time, so I will ask you then to reply online to them through our team who is following the online part. Thanks again. And we have to start our third session of the Open Session 2022, which is focused on virology and diagnostics. And the chair of this session is Stefan Zientara, who is a virologist and head of the virology unit at HANSES, the Food Safety Agency in France, and also the chair of the STC. And Stefan has been assisted in the preparation of the session by Kirill Krstevsky and Kees van Manen, laboratory expert and virologist, who Kees will also assist in moderating the panel discussion, supported by Silvia Baluca that you met yesterday. So, Stefan, the floor is yours.
Thank you very much, Fabrizio. And dear colleagues, dear audience, it's my great pleasure and honor to share this session on virology and diagnostics. Welcome to all the participants in the room, and also all the people who are online, and we have special thanks to people following the event very early in the morning and very late at night. I've got the great pleasure to have two colleagues moderators. So as Fabrizio says, Dr. Silvia Baluca, not Kirill, who unfortunately could not come, and Dr. Kees van Manen. I will very rapidly describe your CV. Silvia, you tell me if I made any mistake. So you are a veterinarian serving as a regional expert on veterinary paraprofessionals. Silvia, you assist EuFMD in development and delivery of a model for training veterinary paraprofessionals towards sustainable business and improve animal health services. You completed your PhD on veterinary epidemiology and economics, focusing on transboundary animal diseases, particularly foot and mouth disease, from Mackay University in 2014. And now you are serving as a senior lecturer in Mackay University, and you are also current president of the Uganda Veterinary Association. This is correct?
I handed over the presidents.
Okay.
Yeah, but I'm the immediate past president of the Uganda Veterinary Association.
Okay. Thank you, Sylvia. And Keith, Dr. Keith von Manen. Keith assists EuFMD in development and delivery of training courses, workshops, laboratory assistant, PCP support for FMD and other fast disease for many years. Everybody knows Keith. So you are a virologist, senior scientist based in the Netherlands, and you completed your PhD some years ago on the diagnosis, epidemiology, and vaccinology of equine herpes virus and equine influenza virus infections. Correct?
Thank you.
Okay. So, as you know, the COVID crisis has shown the value of innovative tools, and it is important to consider how modern technologies and digital transformation can support and improve the diagnostic capacity for fast diseases, and also ensure availability of diagnostics, personnel capacities where they are needed. So the session on virology and diagnostics will consist of 2 keynote presentations, 6 short oral presentations, and there are also 15 posters that you can view here. So the main objective of this session is our deeper insight into host-pathogen interaction and innovation in diagnostics and data analysis, and presentation can contribute to better inform the way we understand, monitor, and control fast diseases. So as I told you, we will have 2 keynote presentations, one given by Dr. Donald King, physically present, as you know, and Dr. Zheng from China, who will be online. So everybody knows Don King. So Don has a very extensive experience with foot and mouth disease, and he is the head of the World Reference Laboratory at the Fulbright Institute in UK. So now, Don, the floor is yours. You have 15 minutes, 20 maximum. Thank you, Don.
Thank you very much, Stefan. Thank you for the kind words of introduction. And good morning, everyone. Hope you don't mind seeing me for the second time. And good day to everybody who's joining the conference online. So the purpose of the presentation is really just to give a sort of a snapshot overview of some of the work that's been undertaken to look at diagnostic methods and novel diagnostic technologies. And I suppose in the presentation, what I tried to do is sort of highlight some of the other talks that will be given in this session, and as Stefan says, maybe direct you towards some of the posters that give you further information about what, some of the work that's been taken. Before I start, just perhaps start with 2 sort of small confessions. One is the perspective of the presentation is very much thinking about methods that are focusing on virus detection and virus sequencing methods. Of course, conscious that there's a huge amount of work being done elsewhere with other technologies, and thinking about the diagnostic continuum, this early recognition of animals that might have a disease such as FMD is really critical, and there's some really exciting methods there looking at, for example, animal behavior and other approaches for farmers to identify those animals that might be exposed to these viruses. I'm not going to talk about that at all in the presentation. The other small confession really before I start is actually I gave a similar presentation to this in 2008 at the open session in Richaud. And at that time, also presented these as novel new technologies. And I'd say perhaps now some time has passed. And as Stefan says, I think we've learned a lot from COVID. And I think perhaps within the livestock space, it's timely now to reflect on what we're trying to achieve with these technologies, what the demand is for these technologies, and why it is perhaps that some of the really exciting work that's being done in the lab is not necessarily being translated into the tests that are used in routine use. So before I go into the presentation, just use this slide to acknowledge a lot of the partnerships that we've established over the last 10, 15, even 20 years to look at novel diagnostic technologies for FMD. And I think the key message is here that these partnerships are really critical. And so you need to generate not only the test itself, but you need to validate the test. You need to provide confidence to the users of the test that the test is going to meet
the expected criteria. Quite often you need to work with other academic partners to access reagents and materials to develop the test formats. Also working with teams in FMD endemic countries here. The talk will mainly focus on FMD, but of course a lot of what I'd say will have some application for other diseases too. But working with those teams in the countries where the disease is endemic is also critical, because it gives you access to those materials, again, that help you validate the test. And then, of course, working with the commercial sector is also quite important. And so, again, thinking about the legacy of the work that might be done in the initial phases of a project and taking it through to a product that actually has some commercial viability and will be sustained over a long period of time is really critical. So just use this slide to acknowledge a lot of people and also past members of the World Reference Laboratory that have contributed to the work that I'm going to present today. So let's start with a sort of a consideration of what we're really trying to achieve here and what the main driving forces are for changing the way that we do diagnostics. And I use this sort of very simple cartoon, which hopefully everybody recognizes, which is the sort of the existing sort of diagnostic approach that is used for many different livestock diseases, where you have an animal that presents with suspect clinical signs. There is obviously it's really important that those animals are recognized and identified as having clinical signs, usually initially by the farmer and then also by local veterinary services. And then sometimes samples are submitted for laboratory investigation. Of course, we know the problem with centralized laboratory testing is that this sending of samples to the laboratory takes some time. It also comes at some expense. And that, if you remember from the presentation yesterday, really limits our ambitions perhaps to undertake wider scale global surveillance for diseases such as FMD. So what are we trying to do? Well, we're trying to fit this space to get the very rapid data back to the field teams to understand what's occurring. And here there are perhaps two principal drivers. One is a driver which was probably the starting point for the deployment and development of these technologies, which was the idea of these point of decision tests, where you can very quickly identify and at the very earliest opportunity identify animals that have a clinical disease. And this approach is particularly important for disease-free countries, and really was one of the key messages, I think, that came from the 2001 epidemic in the UK, is that was this need in previously FMD-free countries to have these very rapid tests to allow decisions to be made very quickly about the status of animals and whether control measures need to be in place. But I think it's fair to say, as we sort of develop these approaches over the last 20 years or so, we now also see an opportunity really to use these tests to enhance the global surveillance of disease. And I think this picks up on this idea, and if you remember from my presentation yesterday, almost a half of the samples that come into the reference laboratory network are poor quality samples, which are coming at quite considerable expense in the shipment of those samples to the lab. But once they're received to the lab, very little information can be retrieved. And so having a much clearer line of sight between the field teams that are collecting the data and the lab teams and the epi teams that are analyzing the data is really critical. And I think some of these tools have real prospect for and real potential for filling that gap. And this hopefully will ensure, will ensure that the best quality samples are collected for analyses. So a lot of these approaches, of course, have been driven by advances in human personal medicine. And I'd say, although it's important that we reflect on the impact of COVID-19 and the really dramatic explosion of some of these technologies that have been used in the past few years to do local diagnosis. And you can see here on the slide, I'm sure there will be further reviews in the next couple of months looking at the number of tests and lateral flow tests that have been used. But even just in the US, over a relatively short period, it's been estimated that more than 2 billion LFDs were used for COVID diagnosis. So this is a huge explosion, and people are now very aware of the potential use of this technology. But notwithstanding that point, I think in the livestock space, we've been working in these technologies for, as I said, for a number of years, and we now need to reflect on why perhaps there isn't a wider application of the methods. So as I go through the presentation, just going to really very quickly go through sort of 3 case studies. One is looking at immunoassays using the lateral flow test formats. The second example I'll use is the mobile molecular test formats, both using mobile PCR and isothermal formats. And then finally, I think really excitingly thinking about newer approaches that can be used to generate viral sequence data in a really easy way and a real expansive way to get a lot of more information. But as I say, I think before going into this, I think I'd like us all to sort of just reflect on this, what we consider what's called the valley of death, which is this gap between this initial enthusiasm by academic partners to generate a publication and this low uptake of some of these technologies by the commercial sector over a sustained way. And so obviously there's lots of publications that describe tests that potentially could fit some of what I'm describing today, but very few of those tests actually make it through to a commercial product. And I suppose the message is, when we're working for the test technology, I mean, of course, a lot of the time that's driven through academic curiosity and a desire to get something novel published, but really should we be engaging with the commercial sector at an early stage to understand the demand for these tests? Okay, so let's start with some of the examples. So the one example, of course, that I think many of you will be familiar with particularly for FMD diagnosis is the tremendous work that's been done over the last 10 years or so, or even longer, to develop antigen lateral flow tests for FMD. And these are very rapid tests, very similar to the COVID test, give you a result in less than 10 minutes, and can be used in the field, and actually have been used in the field. You know, for example, when we had the FMD cases in 2007 in the UK, on some of the farms, these tests were used very rapidly to identify animals that were infected with FMD. So the tests work, and not only can they be used in the field, but they can also be used in the lab to, again, triage samples that are the most interesting samples that should be taken for further forward analyses. The performance of these assays is now well understood, and in general speaking, the sensitivity and specificity of the test is very similar to the antigen detection ELISA, which is sort of a very well-recognized standard test that is used by many laboratories. And there are pan-serotypic versions of this test, as well as serotype-specific versions of the tests that have been developed, and some of them are still under development. And at this point, just direct you to some of the posters and a further presentation in this session that will describe serotype-specific LFDs for FMD. But going back to this point, I think, you know, tests in this format have now been available for FMD for some time. And I think for those of you who have previously accessed the LFD from Svanova, you'll know and be aware probably that there are now difficulties to access these materials. But I think it's an important question, which is why, more broadly, why have these tests not been used, and do the costs inhibit the use of LFDs for routine wide-scale testing? Of course, we know that LFDs are not only useful for laboratory use, but they can also be used in, not only used in the field, but also can be used to collect samples that can be subsequently analyzed in the lab. And there's a tremendous amount of work being done both at Pirbright and other laboratories such as ONSES to use these devices to recover viral RNA that you can actually, dropped it again, excuse me. This has got a terrible slope on it. From which you can recover the live virus if you want to, and of course that leads to the opportunity for sequencing, which perhaps fills the gap that I was describing earlier, this idea that you can collect samples in the field that you know are FMD, you can then ship those samples to the laboratory for further analysis by sequencing. And there's some posters in this session from colleagues at ONSES that will take forward that argument and describe recent work that's been done in that space. Also need to say something very briefly about nucleic acid methods, and So both PCR-based methods using, again, pan-serotypic assays as well as lineage-specific assays, and a whole suite of lineage-specific PCRs have been developed recently by a number of different laboratories. So those are now available, can be easily transferred onto mobile molecular formats, as well as, again, many of you will be aware of these very simple-to-use isothermal formats for FMD and other detection using technologies such as LAMP. And again, there's a whole range of these publications that are in the literature, but again, very few of these have been taken forward for routine use. And I suppose, again, here asking the question, does access and the costs associated with these tests limit or actually perhaps even prohibit their use for wide-scale use? Notwithstanding what I've said there, again, in the last few years, a lot of work has been done to try to transfer these molecular assays into a very simple format that can be used in the field. And here, using lyophilized, dry-down reagents has been a very important step forward, and work that has been undertaken, again, in partnership with scientists in East African countries, has demonstrated that these dry-down assays work. So the technology works, and the performance of these assays is broadly similar to the same type of performance for the tests if they were run in the laboratory. So it's not the technology itself that's inhibiting the process here, it's more understanding the demand and access to the materials and the reagents required to do the tests. And of course, thinking particularly about molecular tests and perhaps the sequencing-based platforms, it's this idea that you can hide a lot of the complexity of the test within the assay. And again, I think the lyophilization of reagents is a good example of this, and I'll give you another example on this slide, where perhaps the user who's using the test in the field doesn't need to necessarily understand how the test works. And an example of this is work that we've done recently at Pirbright in collaboration with the University of Glasgow, using a very simple system for lamp detection, which uses a smartphone heater where the test is performed on a chip, and the user really all they need to do is take the sample and put it on the chip, and the results are very apparent for the user then. So again, these types of formats and this type of inherent simplicity is going to be very critical for the use of the test. So towards the end of the presentation, just want to say something about the tremendous work that's been done to improve and enhance sequencing capability for diseases such as FMD. And again, many of you will be familiar with the evolution that's occurred over the last 20 years in the sequencing technologies that have been available to us. And it's fair to say now that the technology is no longer the limiting factor for sequencing. And if you take, for example, again, looking at COVID, just a huge number of genomic sequences have been generated over a relatively short period. And actually, eye-watering number of sequences that previously we wouldn't have considered possible. And actually, when we started to sequence, for example, viral genomes such as FMD, when we had small studies that maybe comprised 50 genomes, we considered that to be a big study. But now you can see that the whole paradigm and the whole idea here has changed over the last few years. And there's an expectation, I think, now that when samples are collected in the field, that those should be sequenced. And as I say, the technology is not limiting us here. Perhaps the only factor that's limiting us here is simple to use analytical pipelines here, thinking about bioinformatics to really make sure the data is put in one place and people can access the data for different purposes. And I think that's going to be a challenge for the community is to develop those tools. And again, thinking specifically about sequencing methods, I'm not going to say very much about the MinION sequencer because Andrew Shaw will have a talk later on in this session, but it's fair to say that this technology again works, and it works very nicely for FMD, gives you an approach that you can use very quickly in a reasonably portable way to sequence the virus, and perhaps addresses some of the limitations that exist particularly in FMD endemic countries where access to the equipment at the moment can be a problem, and certainly servicing equipment over longer periods of time can be a problem. So here you can generate viral sequences with with very little equipment, just a PCR machine and a sequencer. And the preliminary data, and like I say, Andrew will say more about this in his presentation, demonstrates that we can easily generate protocols for FMD sequencing. And really, this really should open now an opportunity for us to generate a lot more sequence data. And this idea that the sequence itself, I think, gives us the highest resolution of the nature of the virus that's present in the field. So just to conclude the presentation, just to sort of perhaps pitch a slightly new way that diagnostics might be done in the future. You know, you can imagine that when samples are collected in the field, you can generate and deploy tools that will allow the veterinarians or the animal health officers or the farmers to answer the question, is FMD present? They also will have, perhaps in their hands, tools to also identify the particular viral serotype that's present in those samples. And that, as I said, gives us the opportunity for this very rapid, immediate feedback. So if they see what they think is FMD in front of them, but they get negative results, they have the opportunity to resample further animals or even to start to think about other diseases that might be present on the farm. And then we have also now tools in place to ship inactivated materials perhaps to a laboratory. I think thinking about sequencing in the field is perhaps, I mean, it's not going to happen immediately, and I think we always imagine that the sequencing will be done at least in a laboratory, but it doesn't necessarily have to be in an international laboratory where samples would need to be sent through expensive ways. So it could be done in a local laboratory where you would undertake sequence analysis and characterization of the field isolates and interact with that network of sequences that are available for other samples. So this is just a sort of a concluding slide that perhaps summarizes some of the points that I've made. I think as we think about all of these different technologies that have been developed, selecting the winning technology is often quite a challenge because you can't immediately see that when at first sight. And again, working collaboratively helps share some of that risk because different labs can pick up and have expertise in different activities. Bridging this gap between the assay development and the marketing of the tests is important. Engagement of the end users, and particularly thinking about whether or not the test has real practical utility and demand at the earliest stages, I think could be and should be quite important. There are also sometimes considerations about freedom to exploit the IP, as well as some of the specific reagents that might be needed for the tests, and these partnerships between commercial and academic sectors are really important here. I'm not going to say anything at all today about the acceptance of the tests, but again, thinking about the WOA manual, I think there are opportunities to move some of these tests into the more formal accreditation process so that tests become widely accepted for diagnosis, where validation may be quite critical. And then the final point from me is that there's also opportunities to think about the data that's collected and making sure that the data is translated into information. And I think we're here tomorrow about further work in that space. So thank you.
Yeah. Thank you. Thank you very much, Don. You have exploited more or less the time. We have time for one question. We have no time, but if there is one burning question, you have -5 seconds to answer. No questions for the moment? Okay, fine. So thank you very much, Don. And so now the next presentation will be done by Dr. Zheng from the Lanzhou Veterinary Research Institute, belonging to the Chinese Academy of Agricultural Science in China. And it will be about virus-host interaction in EuFMD virus infection, and it will be online. So Dr. Zheng, the floor is yours.
Hello, everyone.
Hello.
Greetings from Lanzhou, China.
We don't hear you for the
I'm
moment.
Professor Zheng Haixue, the Director General of Lanzhou Veterinary Research Institute. First of all, I would like to give my thanks to EuFMD for your kind invitation. Today, I would like to take this opportunity to share my findings about innate immune evasion by food animal disease virus. Food animal disease is a severe, highly contagious animal disease. It infects mammals such as cattle, sheep, goats, and pigs. It has an incubation period of 1 to 10 days and spreads rapidly within a livestock population. Outbreaks of food animal disease, especially in EuFMD-free countries such as UK, Japan, and Jordan, can have serious economic consequences. In order to infect the host, the virus must go through 3 barriers: innate immune response, cellular immunity, and humoral immunity. At the early stage of infection, the innate immune response is important. At the late stage of infection, especially starting from day 6, the adaptive immune response takes over and begins to protect the host by producing the antibody. Meanwhile, cellular immunity is also involved. If the virus wants to establish persistent infection, it must overcome the first barrier, that is, innate immune response. Following infection, EuFMD will replicate rapidly and cause acute disease in animals. Our question is, how does EuFMD skip the innate immune response to establish persistent infection? Based on those observations, we proposed 2 questions. 1. How does EuFMD regulate innate immune response system? 2. How does innate immune evasion contribute to pathogenesis and adaptive immune response? Innate immune response is important for the host to clear EuFMD infection. Like other viruses, EuFMD is reported to have evolved multiple strategies to evade and attenuate the host innate immune response to facilitate viral production and replication. However, the detailed mechanism is complicated and unclear. Given the important role of the innate immune response, to find out EuFMD innate immune evasion will give us more insights into prevention and control of EuFMD. My presentation has 4 parts. 1. Regulation of pattern recognition receptors by EuFMD. 2. Regulation of key innate immune molecules by EuFMD. 3. Host protein mediated antiviral response against EuFMD. 4. Development of antiviral treatment based on the EuFMD proteins. EuFMD develops many strategies to evade the innate immune response. The RIG-R-like receptors and node-like receptors are important for assessing EuFMD infection and triggering innate immune response. I found that EuFMD2B and 3B proteins interact with RIG-I to degrade RIG-I and promote EuFMD replication. Meanwhile, EuFMD2B interacts with LGP2 to degrade LGP2. At the same time, EuFMD2B and 2C degrade NOD2 to block innate immune response. In our results, we observe the co-localization and interaction between 3B and RIG-I. Those events block the activation of RIG-I, further suppressing interferon beta production. After EuFMD infection, the interferon signal pathways are activated. Except for the pattern recognition receptors, many innate immune molecules are playing important roles. MAPS is the key adapter protein that helps to phosphorylate and ubiquitinate TPK1. The activation of TPK1 could transduct the signals and trigger the interferon response. Our team found that EuFMD proteins could directly or indirectly disrupt the activity of MAVs, for example, the VP3 and VP0. TPK1 is a novel E3 ubiquitin ligase we found that TP1 is involved in the degradation of VP3. Once the innate immune response was activated, the F3 was phosphorylated and translocated into the nucleus for transcription initiation. Our team found that EuFMD VP1 interacts with F3 and inhibits its phosphorylation and nuclear translocation. This finding was published in Journal of Virology as a cover story. Besides, EuFMD3A could suppress F3 activity through DDX56. By using overexpression and co-RPIC, we found that EuFMD-VP3 interacts with MAVAS and interferes with the formation of MAVAS complex, further blocking type 1 interferon production. Meanwhile, its activity is dependent on the C-terminal domain. Meanwhile, several virus proteins could also attenuate the antiviral response through regulation of host proteins. For example, EuFMD3A protein degrades G3BP1 to impair host antiviral response. VP1 interacts with RPSA to maintain the activation of MAPK pathway and promote EuFMD replication. On the contrary, many host proteins could further amplify the antiviral response to limit virus replication through multiple mechanisms, for example, the TPL2 and GMGD6. As I said before, F3 phosphorylation and translocation into the nucleus is an important event for interferon signal pathway. We found that the host protein GMGD6 recruits RNF5 to induce the K48-linked polyubiquitination and proteasomal degradation of activated F3. The GMGD6 is an important regulator of innate immune response. Our findings also provide a perspective for manufacturing the EuFMD vaccine. We previously found that TPK1 degrades the VP3 through catalyzing the K48 ubiquitination. On the one side, we established the TPK1 knockout cell lines. On the other side, we constructed the recombinant EuFMD virus by mutating the ubiquitination sites. Both strategies could help to improve the viral titers and antigen production. Meanwhile, we try to develop antiviral strategies based on the properties of EuFMD proteins. We determined the crystal structure of an EuFMD2C fragment to 1.83 A resolution. Meanwhile, we designed an anti-EuFMD peptide derived from PBL, which inhibits the EuFMD2C ATPase activity, bound to EuFMD2C with nanomole affinity, and disrupted EuFMD2C oligomerization. These findings provide perspective to develop EuFMD-specific treatment. This figure summarizes multiple mechanisms employed by EuFMD to evade the innate immune response. EuFMD could target the events prior to the interferon production or the downstream YAG-STAR pathway to suppress the innate immune response. Prior to the interferon production, the pattern recognition receptor RICI and the downstream transducers like MAVAS, TPK1, and F3 could be targeted and downregulated by EuFMD proteins. Following interferon production, the YAG-STAR pathway was activated. The key components of YAG-STAR pathway could also be targeted by the EuFMD protein. Apart from type 1 interferon signal pathways, several classic pathways related to host antiviral response were also disrupted by EuFMD proteins. For example, the toll-like receptor 4 pathway and the MAPK pathway. Those events result in the suppression and the invasion of the host innate immune response. I would like to give my thanks to all my team members and Wuhan, China National Food and Animal Disease Reference Laboratory. Thank you very much for your attention. Before I begin my presentation, I would like to make a promotion by giving you a brief introduction of Lanzhou Veterinary Research Institute. It's located in Lanzhou, Gansu Province, the geographic center of China. Our institute has a long history. It's founded in 1957 and crowned as a nationwide top academic research institute. It's home to 3 OIE reference laboratories and the largest platform of high-level laboratory facility for animal biosafety in China. I'm the current Director General. My team is dedicated to fundamental research and control measures of food and animal disease and African swine fever virus. I focus on epidemiological patterns and evolution, molecular basis for phenotype variation, persistent infection and possible transmission by carrier, molecular mechanism for immune evasion, adaptive immune response, and EuFMD infection peaks. If you are interested in my project, please contact me by email. We hope we could establish cooperation with EuFMD and all of you in the near future. Thank you very much for your attention.
Thank you very much for this presentation. Do you have some questions? What? You don't have questions, fine. So Philippe has no question. Questions? Yeah, Brian? Sorry? Yeah, after.
Thank you for the nice presentation, clear presentation of a complex subject and the detailed biochemistry. I have a couple of questions, actually. One is clearly FMDV induces type 1 and type 3 interferon in vivo. And we think that may be derived from the plasmacytoid dendritic cells. So these pathways that you've dissected, do they, how are they operational, that interaction operational in plasmacytoid dendritic cells as well? What's the difference between those cells and other cells?
Hello everyone. Greetings from Lanzhou, China. Because of the spoken English issues, on behalf of the professor's destination, I would like to do the translation and answer your question as much as I can. Yes, I really appreciate your question because most of our research regarding the induction of the type 1 and type 3 interferons are performed in the RBS 2 cells and PK15 cells. These cells are not DC cells and macrophage cells. And until now, we don't perform the experiment in the DC cells. So we don't have a clear answer and explanation for your question. But we don't have the in vivo data.
Okay, thank you.
Okay, after discussion with the professor, we have the animal experiment and we use the reverse genetic systems to prove that in vivo, the animals can also, we can also detect the type 1 and type 3 interferons in the pigs. I think these cytokines are mainly produced by the DC cells and macrophage cells.
Okay, thank you. Well, that's good. So there's a piece of work we all need to do is understand the differences. And it was just picking up on the final slide of you showed the profile of innate and adaptive immune responses. Just a little bit confused by the antibody profile you've got in that graph, because we know if we deplete all of the CD4 T cells using monoclonal antibodies, we still get a rapid induction of antibodies within about 3 to 4 days. So that doesn't quite fit with the data that we've seen.
We don't hear you. You are muted.
You're still on mute. Yeah.
Okay, so we still have more minutes to discuss with the professor, and we'll give you the answer as soon as we are ready.
They're waiting for the answer.
Thank you very much for your question. Actually, the last figure we just take from other articles and copy them into not copy. We just do some summarize and review of the previously published data, and we make this figure. I think for the antibody response, it will start from day 4, but for most of the animals, the production of the antibody is different. We just do a summary, and we don't give a specific introduction time for one kind of species. For example, for the pig and the sheep and the cattle, I think they are different.
Okay, thank you very much.
I thank you very much for your answers, and thank you to be direct in China. Thank you very much. And so now we can shift to the panel discussion. So I'll let the floor to Keith.
Thank you very much. Dear audience here and also online, we are going to have half an hour of panel discussion now. We prepared some questions, but we give you the full opportunity for questions also. I would like to invite Labib, Mackay Kassimi, and Santina Graccioli, and Stefan and Don King to sit here and be our panel together with Dr. Zheng online. And before I start this, I should inform the participants of the workshop of today that they are expected 11 o'clock at the reception. And then you will be 11, right? Yeah. 11:20? Yes. Okay. 11:20 at the reception for the workshop participants. Okay, our panel. Stefan does not need any introduction already. He has been introduced. Labib is from the ANSES Institute, also very well known. Don, you have heard about a lot, and Santina is well known from the Isler Institute, and we are really happy to have you here as participants in this panel discussion. And I would like to start with a question for Dr. Zheng. And so I hope he can still hear us, and I will sit down also. And that is that you have helped us with a better understanding of virus-host interactions for FMDV infections. But what is your vision on the feasibility of using antiviral agents in FMDV control and prevention in terms of pharmacokinetics, efficacy, safety, both for target animals and for human consumption? So this is a long question. And which compounds, such as peptides, you mentioned peptides, but also siRNA, double-stranded RNA, ribavirin, etc., will be registered and thus available soonest, according to your opinion? Over to you, Dr. Zheng.
Okay.
thank you very much for your question. And 20 seconds, I will open my mic.
And,
thank you very much for your time. I think you specifically mentioned the article we published in the Cell Reports in 2022 about the development of the 2C based antiviral treatment against FMDV. Actually, I think in this article we just use FMDV as a model virus to just first we crystallize the structure of the 2C, and we develop some peptides which can bind to the pocket-like loop of the 2C. Then this peptide have the in vitro antiviral activity against the FMDV. And our findings can also apply to the EV51 virus and other picornaviruses. So for the application of the antiviral agents, yes, I think we have some, yes, these antiviral agents have some advantage, especially the biosafety advantage compared to the inactivated vaccine and also some other vaccines. And also
I think we lost him for a moment. I hope we will get the connection repaired again. But for now, I would like to transfer the question to Stefan, because in a more general sense, how do you see any application of antiviral agents in let's say in free countries? For example, I'm just fantasizing, when you would have a lot of farms infected, you would have delayed rendering capacity, and you might want to stop virus excretion in pig farms or whatever.
So perhaps the first thing, but I'm not a specialist of antiviral, but so I hope that the Chinese colleague will agree with me. But I think that for the moment, I think we are quite far from having antiviral in the field. First of all, because we have also to have information about safety in different species, as you know. Then also the risk of escape variants, which are also important, that we could have viruses which adapt or which can not be sensitive to the antiviral molecules. And also to have a dossier to be used in the field. This is really a long trip. And probably last point, which is perhaps the most important, is also the price. We have worked some years ago on antiviral on PPR some years ago, and in fact the price was almost the price of all the farms. So at the end it was a bit complicated. So to my opinion, but I don't know if you agree, I'm not sure that tomorrow we will have antiviral against FMDV. But it could be effectively efficient if we have an antiviral cheap, safety, which can be also used for human consumption after when the animal have been treated, that you can use this. If we would have all these molecules, it could be effectively interesting to use them when you have an outbreak situation, waiting for hazard measures, why not? But to my opinion, I don't know if you agree, I'm not sure that in the next years we will see antiviral use in the field. You agree with me, or?
Yes, let's first return to Dr. Zheng and colleague. Please, can you continue your response, or were you finished?
Well, yes. So thank you very much. We apologize for the disconnection due to the internet issues. I think we have to go back to our findings about the development of the antiviral agents based on the 2C peptide, because we found there is a pocket in the 2C. So we just developed the pocket-like peptide, which can block the activities of the loop of the 2C, further blocking the activity of the 2C, FMDV2C. So this peptide has a very good in vitro antiviral activity against the FMDV replication. And until now, we don't have the in vitro data. But I think compared to the traditional vaccines, for example, the inactivated vaccines and also some recombinant vaccines, I think this antiviral agent has some, especially the biosafety advantage over them, because it has no risk of spreading the virus and the leakage of the active virus. And also another problem is that for the storage and the stability of the peptide, because peptides are easy to be degraded by some enzymes. I think this is a problem we have to think of, as mentioned by the previous speaker. I think how to develop the cheap, effective, and affordable antiviral agents for all the stakeholders and private sectors are very important. And nowadays, I think the key principle of the prevention and the control of the FMDV is still dependent on the stamping out and also the vaccination. I think this is the key principle we have to stick with. But also we also make some advance in developing the antiviral agents. It's our field work. Thank you. I'll give the floor back to the host.
Thank you very much. Thank you very much. Any questions from the audience for Dr. Zheng? Yeah, David.
Thank you. So it's not really a question, it's rather a comment. So at Janssen, we have been working quite a lot of years on the development of antiviral drugs for FMD. That was together with a university who is specialized in antivirals for humans, and also with a commercial company. And our biggest challenge has always been the antigenic variability of the disease virus. So for all the classes of compounds that we worked with, we observed quite frequently that when it works well for one serotype, is that even within the same serotype, if you use other strains, it's difficult to achieve the same level of activity. And then when you switch to another serotype, the activity quite often is much less. So, and that it was always the biggest challenge that we have met, is that although those antiviral drugs most of the time are working on the NSP proteins, the antigenic variability of the virus, it has a very big effect on the effectiveness of the antiviral drugs.
For your comment. Pascal?
I didn't see your talk done in 2008 in Richaud. I hope we've made some progress on diagnostics since then. But what struck me is that all the tools you presented rely on the detection of a first clinical case. It's not going to solve the problems that we've seen in past outbreaks where, like in Indonesia, there were several weeks of circulation of the virus under the radar, asymptomatic circulation. It was not better in the UK in 2001. So here we could probably learn a lot from the COVID example, where there's been some good papers on detection of virus in wastewater way before the wave occurred. So what's your view on that? I don't see much progress being made in the field of environmental sampling as early indicators of infection. What is your view on that?
Yeah, thanks for the question, Pascal. I think, I mean, you raise a good point, which is, and I think I mentioned it in the presentation, the presentation covered mainly tests once clinical signs had been presented in an animal and the correct samples have been collected. Those are the tests that we're looking at. But I think there are other approaches that could be used. I'm not going to say very much about environmental sampling. I know a colleague from Pirbright, Simon Gubbins, is here. He will say a lot more, and other colleagues from Pirbright in, I think, the present, I'm not sure which session that is in, but it's later on this week. There are some presentations about environmental sampling, which is this idea that you can look for the virus in the absence of necessarily finding the animals with the clinical signs. I think that's a really important approach. Other approaches, you mentioned wastewater. I think that's something that we still need to explore for FMD. Also, bulk milk sampling is an approach that we've used successfully and shown again that it can work as a way to monitor upsurges in cases, particularly in endemic settings where you expect to find the virus. And I know there's also interest in bulk milk sampling, for example, in countries such as the US. If they had an FMD outbreak, they might use that as an approach to give them a handle on where the virus was present, particularly across the whole of the landscape of the country. So absolutely agree with you. I think the other types of approaches that people are using is looking at behavioral changes in animals. Again, those are not specific diagnostic tests for a virus like FMD, but again, having a system in place that gives you an alarm and alert that perhaps something is not quite correct with those animals is something I think that's quite an exciting approach. But clearly that's only going to work probably in a closed system such as a farm, rather than it's not going to work probably in many endemic settings where the animals are out in a wider sort of pastoral setting. But you raise a good point. I don't know if others want to comment.
Thank you very much, Don. I would like to continue on that topic, and then mainly, and also I invite Labib and Santina to respond to that. You did not describe it in detail, but you mentioned it, that nowadays we have a whole fancy toolkit of lineage-specific PCRs. We have lyophilized reagents. We can do it in the field with tetracore or other instruments. But costs may still be prohibitive. Do you still see a place for conventional PCR, or do you still see a place for next-generation sequencing? Maybe sequencing will replace this when it becomes cheaper and cheaper.
Do you want me to answer?
Well, maybe
Labib, you can go first.
No problem. Thank you. In my opinion, the diagnostics in the field, it will depend what we will implement after the detection. So LFD or PCR, full genome sequencing, all what we want, when we do it in the field, that's what we have to do after that. To implement control measures. If we will not implement immediate control measures to block the farm or the movement of animals, so we will not do full genome sequencing. Why? We will perform full genome sequencing. So in my opinion, we need the first line tests that are rapid to detect FMD or not, to implement immediate control measures and to block the farms. This is what we need. And it should be cheap, it would be expensive, and easy to perform. So in my opinion, tests like LFDs can be useful. PCR need machines, need some more complicated, sophisticated material. So I believe that any of this can be useful. Now, regarding the conventional PCR, in my opinion, I believe this can be done outside the field, not on the field, in the laboratory. And I believe that conventional PCR, more maybe for endemic country, will be better than real-time PCR. It's expensive. And I believe that what we need is to have one PCR reagent for many diseases and to have a set of parameters, a panel of parameters that in the freezer that we can use them with this same PCR reagents. So because the kit, ready-to-use kit for specific disease sometimes is expensive and you use it only for this disease. So if you don't have any suspicion of topic, so the kit's still in the freezer and maybe expire. So in endemic country, in my opinion, it's better to have one reagent for PCR and a set of the parameters and use it. In our laboratory, we have developed this kind of PCR for classical PCR, conventional PCR for detection and typing of FMD in West Africa for type OA, SAT1 and SAT2. This kind of test for me is useful because they have the possibility to perform the test in multiplex and also in samplex or duplex PCR. And for me, it's more convenient than real-time PCR specific for one serotype or for one disease.
Okay, thank you very much. Santina, anything to add to that?
Yes, and of course, the experience that we had with the COVID-19 give us the information that the lateral flow is the tools that in the field is more affordable for all. And we and the Purebred Institute and many colleagues from endemic country, we are working, we have been working on validate the lateral flow, and we wanted to put them in the market in the very low price in order to be available this tool to many of the labs, also many vet, also in the field countries.
Okay, maybe we can continue. Oh, Don, please.
I think the point that Labib raised is a good point, which is as we develop these methods, another way to balance the risk and perhaps share the risk in the investment for the diagnostics is to make sure that the tests are as generic as possible. So not only thinking about tests for FMD, but thinking about tests for other livestock diseases and having common platforms that can address all of those requirements, I think is quite important. And in that space, I mean, I think probably members of the audience are aware of, for example, the big ambitious programs, particularly in Southeast Asia, to deploy both sequencing capability as well as diagnostic capabilities for flu. And I mean, that program was largely funded by the US government some time ago, where a lot of labs were equipped with quite sophisticated technology. And I think perhaps there was a missed opportunity there to broaden the remit and the desire to set up a test for flu, to widen it for other diseases. And I think, again, thinking about the future, I think when we roll out a technology, I think it's thinking about how widely the technology could be used. And that will ensure sustainability, familiarity, and all those things I think that are really critical for sustainability.
Yeah, thanks very much, because I think that I will come to the audience. But I think that there are, despite what we think here as FMD society, there are countries for which FMD is not the most important disease. And they have other priorities. And when you can use harmonized diagnostic systems that are also applicable to other priority diseases, fast diseases, even other diseases, that may be fruitful. Aldo?
Yeah, I think you had a very good remark that costs are inhibitory, and we have to think what we use these diagnostic tools for. Because if you really look at how Europe controlled foot-and-mouth disease, we didn't have NSP ELISAs, we didn't have sequencing, we didn't have, and still we were able to do it, and mainly based on that just people reported it. So training and things that the EuFMD is now doing, but I think that might be more important in the control of disease, the disease recognition, direct implementation of control measures in the region, that you do not spread it to other regions, try to contain it without waiting for a confirmatory test, is I think much more important than doing all the high-tech technologies we developed in mostly countries that have a lot of money to do these things. But still, they are very valuable. But the lack of data on sequencing from parts of Africa is, South America, we found sequences in Egypt which we could not match with recent sequences from Venezuela, because that would have been interesting. So still, we have challenges that are important, but we should not forget that clinical diagnosis is a very good tool, and we should not overlook that one, I think. And that's a good take-home message, I think.
Thank you very much. Any?
Just a comment.
Yeah.
I totally agree with Aldo. As you know, the first step in the control of foot and mouth disease is the early detection, early the clinical signs. And when you detect early the clinical signs, you will get fresh samples, you maybe get vesicular fluid. So you don't need a high sensitive method to detect the EuFMD. So this step is very important. Another thing is we have to think also multi-diseases. So when you do sampling in the field, that this is cost to go to the field and to the sampling. So maybe we have to think of multi-diseases and to think when we go to the field to take samples, we take samples for EuFMD, for other diseases. The same sample, for example, the blood serum would be used for other diseases. So we have to think about sampling, multi-sampling for multi-disease, and also tests that can be used for multi-diseases.
Thank you very much. Yeah, just before I forget, this is nice with the digital technology. I get messages from the back of the room from the online participants. There are no real questions, but there is a reaction, and that fits in very well. I would totally agree with Dr. King. King's observation about the valley of death for many promising research findings, and that we should really engage with industry from the beginning and to understand the needs of the end users. Well, there is some industry here also, so I think it's an important message. And we have like 300 participants at the moment online. So I'll done, and then I'll go to
Yeah, I mean, I don't necessarily want to disagree with Aldo, because it's never a popular thing to do, but maybe just to perhaps sort of challenge a little bit this idea that the newer technologies are more blue sky and perhaps too expensive for use. I think as the sequencing capability gets cheaper and cheaper, I think it has a real opportunity to make sure that any control programs are deployed in the most efficient way. So you're right, EuFMD control in Europe was implemented without any of the sophisticated diagnostic methods. But when we're thinking about controlling the disease over longer periods of time across different endemic pools, resources are going to be quite limited. And I think understanding the way that viruses are spreading between farms and the way that viruses are spreading between regional districts and things like that are going to be really critical. And I think that understanding, that deeper understanding of the underlying epidemiology of disease is going to require sequence data, and a lot of sequence data. And I think, as you're right, at the moment, sequencing is relatively expensive, but hopefully in time that will come down. And already we're seeing, we're starting to see the thin edge of that. And I'd say we shouldn't exclude that as a possibility just because at the moment it's a little bit expensive.
Okay, thank you. There was a question over there. Yeah. Hi, Michael.
Yes, thank you. I really want to back up what Aldo and Labib have said. I think in a free country, the longer it's been free, the more the memory of EuFMD and awareness, knowledge of EuFMD fades into the background. And we often have made the observation, I mean, luckily it never was FMD, but we had animals that had clinical signs that very well could have been FMD. And people were looking at this for a week or 2 weeks. They were giving antibiotics. They were treating for PRRS. They were doing all kinds of things. And at no point somebody said, hey, let's take a sample, submit it to the lab, and exclude FMD. So you really, this is where you lose the time. You don't lose the time in the 12 or 24 hours that the sample is en route to the lab. You've already lost 2 or 3 weeks while people were just watching this unfold and not doing anything helpful at all.
Thank you very much, Michael. I think that's a good comment, and we will have a session on surveillance and control, of course, tomorrow. And one of the points, of course, is the early warning. Labib mentioned it, and that has everything to do also, apart from the veterinary infrastructure, with the incentives. How do farmers experience such a disease? Do they think it is rewarding for them to report early? What's in it for them? But that's another discussion. I hope we can continue that. Last one before we go to the break, to Santina, a bit of a teaser. Do you think that the new LFDs for FMD, now we have learned so much from COVID, can be as cheap as the COVID tests?
I don't think so.
Free.
The cost of the COVID test is very low, because you keep in mind that the market is not so big, so the cost of production for a small batches is not comparable with the cost of production of the big batch. But we hope to achieve a very good price and affordable for all the customer. And
I mean, just to follow that up, I mean, I think perhaps a question or a teaser back to you and perhaps the wider audience, which is what is a sensible price for an LFD for FMD?
Good question.
And because we're with our colleagues at Eisler, we're starting to consider marketing of some of these devices, and like Santina says, you're making them at relatively small batches, so there are costs associated. And the idea, I think, that we all recognize is that LFDs that were previously marketed by Svanova were quite expensive, and that perhaps was an inhibitor. But the idea, the thought would be, is if you halve the price, would that suddenly make LFDs more widely used? Or even is half the price or quarter the price still too expensive for the routine wide-scale use? And so I'd be really interested in people's thoughts about what a price should be for an LFD for FMD.
I encourage all of you to give your feedback to Don and Santina.
Just a quick comment. I think the price of LFD can be mitigated by the fact that you can use the LFD to know that we have positive samples, and you ship the samples with a cost cheaper than to send epithelium samples. So maybe if you pay the LFD €5, maybe you can get the money for shipment. So for shipment, it will not be expensive if you apply the inactivation protocol. So maybe one in another could be...
Spend more at the beginning and save the money.
Yeah, save the money and for shipment.
We will discuss that after the break, but I think LFDs as a means to transport samples, that's an additional advantage that may be included in the discussions about the price. Any last burning questions from the audience? Then I thank you very much. We have about half an hour. Okay. So half an hour break, back at 11:15, and you don't need half an hour for tea and coffee, so please look at the posters.
What you need to know about foot and mouth disease. Foot and mouth disease is a very contagious viral disease which affects cows, sheep, goats, pigs, and other animals such as wild boar. The disease causes serious production losses, even if not all animals die from the virus. An outbreak in a country can result in a major block to international trade. Humans cannot become ill from the disease, but they can play an important role in spreading it to animals through dirty clothes, vehicles, and other equipment. The signs of FMD are variable and depend on the animals. In cattle, the disease can be severe and you may notice depression, high fever, not eating or difficulty eating, decrease in milk production, increased salivation, blisters followed by lesions on the muzzle, inside the mouth, on the feet and teats. Occasionally, it can cause sudden death in young calves. In pigs, the disease can be severe and you may notice an inability to stand, with some animals in a sitting position, lesions on the feet, sudden death in piglets. In sheep and goats, the disease is not that evident and you may notice small blisters just above the feet, sudden death in young sheep and goats. FMD can be found in all fluids from infected animals. The virus can spread very easily through any of the following ways. The most common is through direct contact between an infected animal and a healthy one. Contaminated materials such as hay, feed, water, milk, urine, and others. Dirty clothing, footwear, or equipment. Contaminated pens, buildings, or contaminated animal transport vehicles. Meat or other products which come from diseased animals, if fed to animals when raw or improperly cooked. And in certain weather conditions, it can spread through the wind from a nearby farm. Contact your veterinarian or local authority immediately if you suspect your herd is infected with FMD. Do not move any animals from the farm and limit any movement of people to and from the farm. A veterinarian will come and look at the animals and take samples to send to the laboratory. The laboratory will examine the samples and confirm the presence or absence of the virus. Once FMD has been confirmed, measures will be put in place to stop the disease spreading to other farms. Farmers whose farms or animals have been in contact with the farm where the disease is present will also be contacted by the local authority or veterinarian. Everyone has a role to play in ensuring that your community and country remain free from the virus. By staying alert and reporting suspected cases of FMD, you can make a difference. Thank you for your attention. What you need to know about foot and mouth disease. Foot and mouth disease is a very contagious viral disease which affects cows, sheep, goats, pigs, and other animals such as wild boar. The disease causes serious production losses, even if not all animals die from the virus. An outbreak in a country can result in a major block to international trade. Humans cannot become ill from the disease, but they can play an important role in spreading it to animals through dirty clothes, vehicles, and other equipment. The signs of FMD are variable and depend on the animals. In cattle, the disease can be severe and you may notice depression, high fever, not eating or difficulty eating, decrease in milk production, increased salivation, blisters followed by lesions on the muzzle, inside the mouth, on the feet and teats. Occasionally, it can cause sudden death in young calves. In pigs, the disease can be severe and you may notice an inability to stand, with some animals in a sitting position, lesions on the feet, sudden death in piglets. In sheep and goats, the disease is not that evident and you may notice small blisters just above the feet, sudden death in young sheep and goats. FMD can be found in all fluids from infected animals. The virus can spread very easily through any of the following ways. The most common is through direct contact between an infected animal and a healthy one. Contaminated materials such as hay, feed, water, milk, urine, and others. Dirty clothing, footwear, or equipment. Contaminated pens, buildings, or contaminated animal transport vehicles. meat or other products which come from diseased animals if fed to animals when raw or improperly cooked. And in certain weather conditions, it can spread through the wind from a nearby farm. Contact your veterinarian or local authority immediately if you suspect your herd is infected with FMD. Do not move any animals from the farm and limit any movement of people to and from the farm. A veterinarian will come and look at the animals and take samples to send to the laboratory. The laboratory will examine the samples and confirm the presence or absence of the virus. Once FMD has been confirmed, measures will be put in place to stop the disease spreading to other farms. Farmers whose farms or animals have been in contact with the farm where the disease is present will also be contacted by the local authority or veterinarian. Everyone has a role to play in ensuring that your community and country remain free from the virus. By staying alert and reporting suspected cases of FMD, you can make a difference. Thank you for your attention. What you need to know about foot and mouth disease. Foot and mouth disease is a very contagious viral disease which affects cows, sheep, goats, pigs, and other animals such as wild boar. The disease causes serious production losses, even if not all animals die from the virus. An outbreak in a country can result in a major block to international trade. Humans cannot become ill from the disease, but they can play an important role in spreading it to animals through dirty clothes, vehicles, and other equipment. The signs of FMD are variable and depend on the animals. In cattle, the disease can be severe, and you may notice depression, high fever, not eating or difficulty eating, decrease in milk production, increased salivation, blisters followed by lesions on the muzzle, inside the mouth, on the feet and teats. Occasionally, it can cause sudden death in young calves. In pigs, the disease can be severe, and you may notice an inability to stand, with some animals in a sitting position, lesions on the feet, sudden death in piglets. In sheep and goats, the disease is not that evident, and you may notice small blisters just above the feet. Sudden death in young sheep and goats. FMD can be found in all fluids from infected animals. The virus can spread very easily through any of the following ways. The most common is through direct contact between an infected animal and a healthy one. Contaminated materials such as hay, feed, water, milk, urine, and others. Dirty clothing, footwear, or equipment. Contaminated pens, buildings, or contaminated animal transport vehicles. Meat or other products which come from diseased animals, if fed to animals when raw or improperly cooked. And in certain weather conditions, it can spread through the wind from a nearby farm. Contact your veterinarian or local authority immediately if you suspect your herd is infected with FMD. Do not move any animals from the farm and limit any movement of people to and from the farm. A veterinarian will come and look at the animals and take samples to send to the laboratory. The laboratory will examine the samples and confirm the presence or absence of the virus. Once FMD has been confirmed, measures will be put in place to stop the disease spreading to other farms. Farmers whose farms or animals have been in contact with the farm where the disease is present will also be contacted by the local authority or veterinarian. Everyone has a role to play in ensuring that your community and country remain free from the virus. By staying alert and reporting suspected cases of FMD, you can make a difference. Thank you for your attention. What you need to know about foot and mouth disease. Foot and mouth disease is a very contagious viral disease which affects cows, sheep, goats, pigs, and other animals such as wild boar. The disease causes serious production losses, even if not all animals die from the virus. An outbreak in a country can result in a major block to international trade. Humans cannot become ill from the disease, but they can play an important role in spreading it to animals through dirty clothes, vehicles, and other equipment. The signs of FMD are variable and depend on the animals. In cattle, the disease can be severe, and you may notice depression, high fever, not eating or difficulty eating, decrease in milk production, increased salivation, blisters followed by lesions on the muzzle, inside the mouth, on the feet and teats. Occasionally, it can cause sudden death in young calves. In pigs, the disease can be severe, and you may notice an inability to stand, with some animals in a sitting position, lesions on the feet, sudden death in piglets. In sheep and goats, the disease is not that evident, and you may notice small blisters just above the feet. Sudden death in young sheep and goats. FMD can be found in all fluids from infected animals. The virus can spread very easily through any of the following ways. The most common is through direct contact between an infected animal and a healthy one. Contaminated materials such as hay, feed, water, milk, urine, and others. Dirty clothing, footwear, or equipment. Contaminated pens, buildings, or contaminated animal transport vehicles. Meat or other products which come from diseased animals, if fed to animals when raw or improperly cooked. And in certain weather conditions, it can spread through the wind from a nearby farm. Contact your veterinarian or local authority immediately if you suspect your herd is infected with FMD. Do not move any animals from the farm and limit any movement of people to and from the farm. A veterinarian will come and look at the animals and take samples to send to the laboratory. The laboratory will examine the samples and confirm the presence or absence of the virus. Once FMD has been confirmed, measures will be put in place to stop the disease spreading to other farms. Farmers whose farms or animals have been in contact with the farm where the disease is present will also be contacted by the local authority or veterinarian. Everyone has a role to play in ensuring that your community and country remain free from the virus. By staying alert and reporting suspected cases of FMD, you can make a difference. Thank you for your attention.
continue. So if you want to go to the beach after the day, we have to begin now. So we'll continue our session, and so the next presentation will be made by Sabine Delannoy from ANSES. It will be online. So Sabine will give a talk about a platform, genomic platform she is leading. So normally we should have connection with Sabine. Sabine, are you here? Yes. Hello, Sabine.
Hello.
Well, so we are all hearing your presentation. We don't see you for the moment. It will come. I have to talk something, so because we are waiting for your slides, so I said, where is my umbrella? This is another challenge I have from my lab to say something. So the challenge is okay. Yeah, thank you very much. It's not a complicated sentence. Sometimes it's a little more complicated. So I don't know what to say. That just Sabine is leading genomic platform with the biomark, multiplex PCR, sequencing. Okay, Sabine is here. Fantastic. So now, Sabine, the floor is yours.
I don't know. I can't see my presentation yet. Yes, great.
Fine, great.
Thank you. So thank you for inviting me to present today. I am Sabine Delannoy from the genomics platform IdentiPad from ANSES, and we specialize in developing molecular high-throughput tools for the detection and typing of pathogens. So more specifically, today I want to talk about a high-throughput qPCR system, the biomark from Fluidigm. It uses microfluidic chips, and it can be used for multiple purposes, from gene detection to gene expression, SNP genotyping, and digital PCR. And I'll present a couple applications later. But so you can see at the bottom left, the older Biomark HD system and the newer Biomark X. We have both of these in the lab, and they use this kind of chips where you can load your samples on one side, and on the other side your assays containing either your primers and probes or your just your primer in case you're using an intercalating agent. And these are all mixed in a matrix fashion, where every single sample is mixed with every single assay in a separate reaction chambers. Each reaction chambers has a volume of 6 to 9 nanoliters, depending on the type of chips you're using. And so you can achieve a total of 9,216 qPCR reaction in a single plate in the time it takes to run a 96-well plate on a regular qPCR machine. So with these massive multiplexing capabilities and the very small volume, you can achieve a very important cost reduction, both in times of reagent and in time. And because it's basically an open plate, you can use any qPCR system that you have developed previously. It's very conducive to user development. You can really tailor the chips to your individual needs. So we have 3 different plates available in our lab. One plate where we can screen 96 samples versus 96 assays for over 9,000 data points. A smaller plate where we can screen 48 samples versus 48 assays for a little bit over 2,000 data points, and a third type of plate where we can screen 192 samples versus 24 assays for just over 4,000 data points. So because the volume of the input is very small, it's 1 nanoliter, we may have sensitivity issues. So in order to increase the sensitivity, we can perform a pre-amplification step, which is a multiplex PCR where we mix all the primers that are used downstreams and use a special Taq polymerase that has been engineered to avoid amplification bias. We perform only a couple steps, PCR cycles, and then we can run the biomark, and using this this pre-amplification, we can significantly improve the sensitivity and increase up to 10 CTs the sensitivity we achieve, which is the equivalent of about oops. I think I was cut. I'm sorry.
No, it's fine, it's fine.
Can you
We
hear
hear
me?
you.
Yes. Okay. So using this system, we can increase by up to 10 CTs, which is the equivalent of 2 to 3 logs, which is very convenient when you want to investigate complex matrices. And it's here in this example when we investigated the presence of the chikungunya virus in serum from infected animals. The next application is digital PCR. It's very used for absolute quantification of sample or detection of very low concentrated samples. In this application, our samples is partition split into a large number of reaction chambers, and until there is only 0 or 1 genome in each reaction chambers, and then the amplification occurs, and then we count the number of chambers where the reaction occurs, and using the Poisson law, we can approximate or estimate the actual number of targets in the original samples. With the biomark, we can screen 48 samples in parallel, and each of them are split into 770 wells of 0.8 nanoliters each. It's a very nice system because the volume of the chamber is fixed, so they are very homogeneous, and it's a real-time system, so you can see the amplification curves. So there's no problem to differentiate the background signal from the actual amplification signal, which is very convenient when you're looking at samples that may have some inhibition or some degradation. So now I just want, there are many application possibles. We've done many more, but I just want to present a couple applications we've done in the fields of virology, because our team is basically working on bacteria, but we've collaborated with many teams in the field of virology. And for example, with the team of Sarah Moutaillé, and we've worked with Mathilde Gondard, who is now working with Labib. They've developed a chip to detect viruses that could be transmitted by ticks. And so you can see the chip, we can detect simultaneously 21 different viruses in ticks, but it can also be adapted to other kind of, and they've developed other kind to look at viruses transmitted by fleas or by mosquitoes. We have worked with our colleague on the CEA to develop chips aimed at the detection of biothreat agents, so either viruses or bacteria. And these kind of chips can be deployed in case of big events and bioterrorism threats. And so we've also worked with our colleague from the Food Safety Lab to detect and quantify foodborne viruses. These are very tricky because they're in very, very low concentration because they don't multiply on food, and they've successfully applied to detect foodborne viruses on water samples, lettuce samples, on fruit samples. And we've also worked with our colleague in Ploufragan to do some subtyping of influenza viruses from a nasal swab of swines. So I just want to conclude from these couple applications. I hope you can see the contribution this kind of machine can do to public health risk assessment. It's very convenient to screen whether it is pathogens or resistance genes or whatever, especially when the expected frequency is very low, because it's very suitable for a very large series of sample, and it gives you really easy access to the microbiome, the virome, the resistome, or even if you want to look at many gene involved in the immune response at the same time. So it's very flexible. You can mix and match everything you want, and it's very complementary to other approach like phenotypic or whole genome sequencing. Thank you for your attention. This is our team, and I'd be happy to answer any question.
Thank you, Sabine, for this complete presentation of this high throughput PCR tools. Do you have any questions to Sabine or online? I don't know. Sorry, done.
Yeah, hi Sabine. Thank you very much for the clear presentation. I mean, I suppose can I ask a really sort of boring question, which is thinking about this technology and other sequencing-based technologies, what does this offer offer advantages over a method such as RNA-seq that might use Illumina platform?
Okay, the main difference here will be the price. The price per sample is much slower, and when you want to screen, earlier you were talking about pre-diagnostic environmental sample screening. This can be the type of tool that are adapted for this kind of purpose, when you don't really know if you will find some positive, where sequencing will be very expensive. This is a cheaper alternative.
Okay, so thank you very much, Sabine. We are out of time. So if you want to ask questions to Sabine, you take the train and go to Maison Alfort. And so thank you again. See you next week. Thank you. And now we will shift to the next presentation, which will be also online. In fact, we will have two other presentations online. The next one is Dr. Shaw from the Purbright Institute. Andrew Shaw, who will give a presentation on complete genome sequencing of FMDV using nanopore sequencing. Andrew.
Hello, my name is Andrew Shaw, and I'm in the World Reference Lab for Foot and Mouth Disease Virus at the Purbright Institute. Today I'd like to speak about sequencing complete FMD genomes using the nanopore sequencer. The first question to ask is, well, why do we actually want to generate complete genomes in the first place? After all, it's possible to generate phylogenies indicating relationships between divergent viruses simply by using the 1D region, which encodes the VP1 protein of the virus. However, there are other tasks which require more information, for which we need complete genomes. For example, mapping outbreaks at fine scales requires complete genomes. Certain phenotypic traits are encoded outside of the 1D region, such as antigenic sites or deletions within 3A. And there's also been a precedent set by the likes of Zika virus, Ebola, and SARS in order to use nanopore technology to generate complete genomes. One of the most widely used approaches for generating products for sequencing on the MinIRON sequencer is to generate tiled amplicons covering the entire genome. This has been commonly used, for example, by the Arctic network. Usually, these strategies are designed for individual outbreak strains or lineages. However, in order to expand the utility of our approach, we've aimed for a pan-FMD approach that's capable of amplifying every FMD lineage. We have designed and tested two strategies. Firstly, we have a system comprising of 20 distinct amplicons, each of which is around 500 to 600 base pair. Secondly, we have a strategy using larger products, which allows a simpler library prep. We've used a combination of novel primers as well as modified versions of existing primers. For example, here in this gel, you can see that modifying the A1D523R primer increases its efficiency, in this case simply by increasing the GM. Such as the diversity of certain parts of the genome, notably the CAPS region, there are often pools of primers which target a footprint rather than a single primer. In the schematic on the right here, you can see the numbers refer to the number of primers in each pool. Here we have gels representing 12 isolates covering the entire diversity of type A, type SAT1 and SAT2 in East Africa. The equivalents for type O aren't shown, but the results concur with what we see here. These products are generated using the small amplicon approach, where we have 20 PCRs. In general, we see broad amplification across all of the viruses, demonstrating the lineage agnostic nature of the primer sets. Clearly, the notable exception are these SAT1 amplicons, where there are currently several missing. However, following on from this data, we can redesign these prime sets so that they have better reactivity. The gels here demonstrate the use of the 20 amplicon approach to amplifying a type A lineage from an entirely different pool. And we can clearly see that each amplicon is amplified to a greater or lesser extent. Interestingly, the lunar script RT, represented by LS, outperforms the superscript 4 RT, which has been used as a comparison. On the right is a tree of the 1D region of some East African 2 sequences from Zambia. Here, we've taken the 20 PCR products and sequenced them using the MinIRON sequencer as a pool, and then compared the sequence to the equivalent sequence generated using Zheng sequencing. And we show that the virus clusters exactly where it should. Obviously, the more prime sites there are, the more opportunity there is for failures to occur. Furthermore, one of the advantages offered by nanopore sequencing is the possibility of using longer fragments. With this in mind, we selected the most reliable sites based upon the in silico and empirical data from the 20 fragment approach, and then designed a new strategy using larger amplicons. In this case, we've tested the PCRs using viruses from epidemiological pool 1 in Southeast Asia, once more highlighting a broad sensitivity for diverse viral lineages. Here we have representative gels for a dilution series of two viruses, a type A from Vietnam and a type A from Cambodia. In each case, we see robust amplification of each product. The eagle-eyed amongst you will have noticed that the amplicons in an earlier slide were shaded different colours. This actually reflects their incorporation into multiplex reactions. Whilst I'm not showing it here, using the 20 amplicon approach, we can amplify genomes in just 2 reactions. Here though, with the larger fragments, we're using 3 different multiplex reactions. On the right, you can see that when we use multiplex reactions, we actually lose very little in terms of sensitivity. I haven't got time to really go into it here, but we've actually selected representative viruses from every single circulating lineage and tested this 3 multiplex reaction approach. In each case, we see amplification, giving us confidence that we can get amplification of viruses from across the globe. To further compare the utility of single versus multiplex reactions, we sequence the products of multiple viruses, an example of which is shown here. In each case, we see complete coverage of the entire viral genome, using both the single and the multiplex approaches. The S-fragment PCR is inherently weaker due to the variability of the primers, and our sequencing efforts now are to investigate the impact of different ratios of the different products in the sequencing reaction, with the aim of getting more even coverage across the genome. To conclude, generating complete genomes can be a good thing, and in some cases is actually non-negotiable. Here I've described our work developing PCR strategies by which we can amplify any EuFMD genome in as little as 2 or 3 multiplex reactions. Following the amplification, we can then use nanopore sequencing to determine the complete genomes at a consensus level. Nanopore sequencing is well known for its ability to be used in resource-poor areas and is actually very rapid. And we're now looking at pipelines which can be used to rapidly analyze the nanopore data. Lastly, down below, I'd like to highlight that anyone wishing to get involved in this and willing to collaborate, can they please get in contact, as I'm looking to try out these primers with as many different viruses as possible. And finally, the acknowledgements. As with any projects, there's often multiple people involved, and this project is no different. All of these people here at the Institute have been fundamental in getting this all to work. Nevertheless, I should highlight Ugo and Keva in particular, as they've really been dedicated to this, put a lot of work in, and done endless jobs to really figure out the best ways that we can do this. And lastly, to DEFRA in the UK for funding this work. Thank you.
So, thank you very much. Unfortunately, Andrew Shaw is not connected, so there will be no questions to him because he couldn't answer. If you have questions, you can ask Don at the end of we are out of time, so we will continue. Okay, but Don is here. So the next presentation will be given by Morgan Sarry. So Morgan, who is somewhere here, is here, is a PhD student in my unit in Labib's team. And Morgan will give a talk on the interplay between EuFMD 3D polymerase and the type 1 interferon response. Morgan.
Thank you, Stefan, for this introduction, and I would like to thank the organizing committee for giving me the opportunity to present a part of my PhD work today. This work is about the interplay between foot and mouth disease 3D polymerase and the type 1 interferon response. I will start by introducing one of the major issues related to EuFMD, namely its persistence in host organisms. As the limit at which we define EuFMD persistence is really controversial, I will rely here on the arbitrary threshold that's used until now at 28 days post-infection. Viral persistence of EuFMD can affect more than 50% of ruminants. And the persistence duration varies between species and reaches up to 3.5 years in domestic cattle. Viral EuFMD persistence has been shown to affect cattle and small ruminants, but have never been described in pig. And moreover, the EuFMD persistence phenomena are totally independent of the vaccination status. And as these healthy carriers constitute a risk of transmission, they represent a real obstacle to disease control and especially to trade, since no one would want to buy a potentially infected animal. And it has been shown recently that in case of superinfection, healthy carriers can be a source of new recombinants, which could escape the immune system. And I think that it's really important to notice that mechanisms underlying EuFMD persistence are still largely unexplained. Even if these mechanisms are largely unexplained, a lot of work has been done regarding EuFMD persistence. First, a virus-host coevolution has been demonstrating during persistent infection. Then, EuFMD have been associated with immunity modulation in vitro, and the Transcript-Provac consortium coordinated by our lab had made it possible to characterize the transcriptional responses during in vitro infection. Thus, it has been shown that during an acute infection, a significant antiviral activity is detected, whereas in the case of a persistent infection, at 28 days post-infection, there is a lasting immune response that is ineffective in eliminating the virus. It's partly as a result of this work that the Persifa thesis I'm not going to talk about was born. The hypothesis we propose here is that this diminished innate response is involved in the creation of a balance between viral presence and host response, and this in EuFMD persistence. My objective here is to identify protein-protein interaction between EuFMD proteins and interferon pathway protein, and to determine the possible host specificity in order to propose new hypotheses concerning EuFMD persistence. First, protein-protein interactions were screened using 16 major proteins from interferon pathway. They were screened by nanoluciferase approach. So this method consists in is based on the development of a small bioluminescent protein that is divided into 2 subunits attached to a protein. As you can see on the scheme, if there's a physical interaction between the 2 proteins being tested, the 2 nanolux subunits can reassemble and form a complete bioluminescence protein. Most of the FMDV protein have been tested against in these 16 interferon proteins derived from 4 susceptible species, namely cattle, sheep, goat, and pig. And the most interesting results are here, and they are concerning the 3D polymerase, whose role in immune escape is really poorly described. So this heat map displays the bioluminescence values associated with each tested interaction. So by comparing the results of these screenings, we can see that many of the potential interactors are shared between species. However, not all the interactions seem to have the same strength from one species to another. We can take the example of the 3D IKK alpha interaction that seems to be relatively stronger in cattle and small ruminants than in pigs. And conversely, you have 3D IF3 interaction, 3D MAV interaction, who appear to be more important, to be relatively stronger in pigs than in ruminants. Interaction are demonstrated using nanolucifers approach, where they are confirmed by using GST pull-down. So, in the similar way that it was done for the nanolucifers screenings, we use the bovine, ovine, caprine, and porcine form of each interactors. And as you can see here, the interaction between 3D and IKK alpha, AF7, IKK epsilon, AF3, MDA5, and NEMO could have been confirmed in all 4 species, whereas the 3D MAV interaction was only demonstrating using the porcine form. As the 3D polymerase seems to be involved in many interactions with interferon pathway protein, I was really interested in the overall impact of this protein on this pathway. So for this purpose, I performed luciferase reporter assay focusing on the induction phase of the interferon response. This method consists in inducing the interferon pathway induction phase in presence of overexpressed 3D. And the reporter gene level of bioluminescence associated was measured and indicate whether 3D polymerase has an effect on the interferon pathway and whether its effect is more inhibitory or activating of this pathway. And here you have some results from this experiment. You have the mean relative induction compared to control. And if you, as you can see here, the assay related to FMDV 3D polymerase induce 40% inhibition of interferon phase induction compared to control. So these luciferase reporter assays are made it possible to demonstrate for the first time an inhibitory effect of the 3D polymerase on the interferon response. I would like now to turn to the conclusion we can draw from these results. As we show in this presentation, we have demonstrated novel interaction between 3D polymerase and the interferon pathway proteins. We also evidence the inhibitory effect of the 3D polymerase on a part of this pathway. And these results are all the more coherent, as all the interactors identified are involved in the induction phase of the interferon pathway. Now, I think that it will be really important to ensure that this protein interaction do indeed exist in real life, indeed in relevant model during infection. And if so, it would be important to consider how the detailed mechanisms of this interaction, of these interactions occurred and how they are implicated in the establishment of virus-host equilibrium and then to viral persistence. I would now like to thank my supervisors, Sandra and Labib, and also thank all the biopic team members. By the way, I invite you to go to see their posters. And thank you all for your attention. I will be really glad to answer your question.
Thank you very much, Morgan, for your very clear representation on interference between 3D and interferon response, and for your clear English that I understand very well. Do you have any questions for Morgan? I've got one, Morgan. As a 3D of the P coronavirus, I was thinking, for example, poliovirus. has been shown to have such interaction, or is it specific for foot and mouth? Or?
Yeah, the 3D of other piconavirus are a little bit more studied, I think. There are some interactions in the literature, but I think that 3D role, with 3D polymerase role in evading the immune system is unconsidered, as it was the case for structural protein a few years ago. But there are some interaction described with 3D polio and some enterovirus. But there's very few interactions described.
Okay, thank you very much. No more questions? Okay. So Morgan, thank you very much again. Thank you.
Thank you.
And so now we shift to the next presentation, which will be online, so no question because Dr. Lee is not connected. So it will be a presentation from Dr. Lee from Lanzhou Veterinary Research Institute in China about epitope mapping of FMDV 146S antibody by crossing-linking max spectrometry.
Hi everyone, good afternoon. My name is Li. I'm a veterinarian doing a sandwich PhD at Wageningen University and Lanzhou Veterinary Research Institute. I'm working on a thesis of in vitro muscles for FMD vaccine quality control using single-domain antibodies. It is a pleasure to be here to present our recent work. Here are the members of our collaboration team. As you can see, the intact antigen of FMD vaccine dissociates into 12S. The immunogenicity of 12S dramatically decreases. The virus proteins are colored according to here. Therefore, the upstream and downstream of FMD vaccine production, the quality control of antigen content is required. Traditional antigen quality control methods include sucrose density gradient. We have developed an alternative way to quantify the intact particle based on single-domain antibodies. It's a double antibody sandwich ELISA requiring intact particle-specific VHHs. Compared with traditional SDG, this method has the advantages stated here. We recently isolated two groups of intact particle-specific VHHs against type A FMD vaccine. As shown on the top, they can specifically bind intact 75S and 146S particles after SDG for different type of vaccine strain. The VHHs are very broadly strain reactive, especially for M702F and M691F. Because these VHHs have high intact particle specificity and broad strain reactivity, they are worth studying. We now investigate why these VHHs are intact particle specific. Using computation ELISAs, we have shown that these VHHs bound to independent antigenic sites. Here we present the process of XLMS. First, the FMD VHH complex are cross-linking by bifunctional cross-linker. After cross-linking, the complex are digested with various proteases, and these cross-linked peptides are enriched by HPLC and analysis by mass spectrometry and forms of these different peaks. By comparing all of these peaks against the database of FMDV sequences, you can identify which are the precise amino acids that are cross-linked between FMDV and VHHs. XLMS results indicated that the epitope of M702F is located close to the two-fold and three-fold symmetry axis of the icosahedrovirus structure, mainly on VP2 and VP3. M702F was not cross-linked to adjacent pentamer, but only to the same pentamer. Therefore, VHH's intact particle specificity is not caused by cross-pentamer binding. At the same time, we also found that the VHH cross-linked with some residues hidden inside the capsid. That is an interesting finding, which indicates that the metastable capsid of FMDV has fluidity. Similarity, the epitopes of M691F is also located on VP2 and VP3 on the same pentamer. Map9 is a 146S specific map, earlier isolated, that was mapped by isolation of escape mutants. Mutations was found at VP317 and K139 indicated in magenta here. This is the same position as M691F binding site. M691F also binds to amino acids within the capsid, as shown here. The above results show that XLMS has the advantages over traditional structure analysis methods in obtaining topological structure conformations of the same complicated protein complex. The epitopes of M691F and M702F overlapped with the epitopes identified earlier for typo 146S specific VHH M170, as shown in magenta here. The cryo-EM structure analysis of M170 reveals that its 146S specificity are caused by altered 3D structure between 12S and 146S particles. This is the same case for M691F and M702F. The conclusions are summarized here. In this work, we have found the intact specific VHH bind to at Zheng sites. The main intact particle specific sites are located within a single pentamer, and the specificity is caused by altered 3D structure between 12S and 146S particles. Also, the pentamer interface is flexible. Thanks a lot for listening.
So, as I told you, no question to Dr. Lee as she is not connected, but Aldo Dekker is somewhere in the room or in the hotel, so if you have questions, you can ask to Aldo some answers. Okay, so now we will shift to the next presentation, which will be given by Dr. Foglia from the Istituto Profilattico Sperimentale della Lombardia e della Emilia-Romagna from Italy.
Good morning.
About LFD.
Yes, thank you. Good morning and good afternoon to everyone. After a couple of years of pandemics, I think that we are all aware about lateral flow devices that are existing and they're functioning. In veterinary diagnostics, thanks to some peculiarities of these techniques, they could be very useful and powerful tools for the on-field diagnostic. In foot and mouth disease diagnostics, right now only few devices have already been fully validated and commercially available. And indeed, the lower sensitivity of the test if compared to other molecular assays is the greatest cons of the technique itself. Our original project was to transfer the techniques behind the antigen detection ELISA kit on a unique lateral flow device, able to detect the presence of the virus and contemporarily serotype it. Together with the University of Turin and In3 Diagnostic, we had to split all the reactions of the antigen ELISA into two devices. The first able to identify serotype A, Asia 1, and O, and so useful in Asia, Africa, and South America. And the second one able to identify SAT1 and SAT2, and so useful for Africa. I want to point out that both devices are able to correctly detect the presence of the virus thanks to a pan-FMDV monoclonal antibody included in the device, in both the devices. in order to avoid every kind of misunderstanding, we changed the name of the two devices, and from now we call them exclusively LFD1 for OA and Asia1, and LFD2 for the SATs. Today, I want to focus on LFD1. The first step to validate the device was to test the ability of the device itself to detect the widest range of lineages belonging to serotypes O, A, and Asia1. Together with the Pervert Institute, we tested various tissue cultures in order to cover almost all the strains circulating in Asia and Africa and belonging to pool 1, 2, 3, 4, and 5. The lateral flow device 1 confirmed to be able to correctly detect all the sample tested, and so covering, as I already said, almost all the strains circulating in Asia and Africa. The second step was to test the analytical performances of lateral flow device 1 very quickly, because we have already spread and published these results. The lateral flow device 1 confirmed to have good analytical performances, both specificity and sensitivity, almost comparable to those observed for the antigen detection ELISA. Today, I will focus on the preliminary data about diagnostic sensitivity assessment. To assess diagnostic sensitivity, we tested many epithelium homogenates, FMDV positive, considered as truly positive samples, and we tested them together in parallel with the antigen detection ELISA kit and with the lateral flow device 1. And then we scored the results of the lateral flow device from 0 to 4, depending on the intensity of the detection line, with 0 as negative and 4 as strongly visible lines. This part of the work was conducted together with the Shop Institute in Ankara and with the Ministry of Agriculture of Kenya. For the detection of FMDV, we found good diagnostic sensitivity. The 82% of the samples were correctly detected. Diagnostic sensitivity completely comparable as that observed for the antigen ELISA. For the serotyping assays, the 80% of samples A positive were correctly detected and identified. The 88% of samples O positive were correctly identified. And a little lower effectiveness was detected in the identification of serotype Asia1. Only 67% of samples Asia1 were correctly identified. But as I said, these are only preliminary data. Here I want to show you the correlation between the two tests, specifically on the serotyping ability of the tests. I mean, the lateral flow device and the antigen ELISA detection. We found a good correlation between the two tests, and even if few samples positive and the antigen ELISA skipped the lateral flow device, more samples negative to the antigen ELISA were correctly identified with the lateral flow device 1, even if with barely visible lines. And, well, in my opinion, these are very promising data. And concluding, the lateral flow device 1 confirmed the ability to detect and correctly type a wide range of lineages belonging to pool from 1 to 5, so covering almost all strains circulating in Asia and Africa. Demonstrated good analytical performances, almost comparable to those observed for the antigen ELISA detection. The preliminary data on diagnostic sensitivity revealed the ability to correctly detect foot-and-mouth disease, and the characterization, the identification of serotypes A and O returned very good results. and more than the 80% of samples were correctly identified, with a little lower performances for the identification of serotype Asia1. Just a quick off-topic information. Tiziana Trogo and I are guest editors for a special issue of Vaccines about vaccination against foot and mouth. So if you have any questions, we are here. And let me finally thank all our collaborators, University of Turin, Department of Chemistry and Department of Veterinary, and In3 Diagnostics for the development of the assays, of the devices, sorry, the Perbat Institute, the Shop Institute, and the Ministry of Agriculture, Livestock, Fisheries and Cooperatives of Kenya for the validation, all our team, and all you for the attention. Thank you.
Thank you very much. Do you have any question? Yes, Nabil. Attends, il y a le micro qui vient.
Thank you. Thank you for this presentation. I have a question, two questions, in fact. I imagine that the antibodies used in the LFD are the same that are used in antigen ELISA. Okay.
Yes, exactly the same.
So the comparison is correct. And my question is, do you plan to test more strands, a panel of larger strand with the LFD? Because the panel you test is not very large. So do you plan to test more strand on these LFDs?
Of course, because these are preliminary data. The device is not completely fully validated. We are still working on. For sure, we want to try other strains. We are very interested in the newest strains and even the South American strains to cover the panel, to have a bigger panel to confirm the validation of the device.
Okay. Okay, so no more question? No? So thank you very much. Thank you. And so the final presentation of this session will be given by Dr. Michael Eschbaumer from the Friedrich Schöffler Institute in Germany. And the presentation of Michael will be about full-length genomic RNA of FMDV is infectious for cattle.
Yes, thank you very much. It actually ties in a bit with the some of the previous presentations on LFD, so I hope you'll find it relevant. We have heard a lot about the possibility of using these lateral flow devices for the safe shipment of diagnostic specimens from the field to the lab, and I think there's unequivocal data confirming this inactivating effect of the citric acid treatment. So you treat the LFD and you're good in terms of residual infectivity from the sample that you applied to the lateral flow device. And this also then means that when you ship it, you're exempt from applying these stringent IATA dangerous goods regulations with regard to packaging and documentation for these shipments, which is all nice and well, but the problem is once you then put an inactivated LFD in a regular piece of mail, you kind of lose control over what's going to happen to your package. There's plenty of evidence, I think all of you will have experienced that in your day-to-day lives as well, that pieces of mail just disappear mysteriously, and sometimes they can then end up in places where animals have access to them in a way that we would really like to avoid. And then if you think about this, yes, you inactivated it, you destroyed the virus particles that are on there, but there is still a lot of viral genome on there, which is kind of the appeal of that treatment, because you then, once you have these samples in the lab, you can apply sequencing, real-time PCR, even recover infectious virus by transfection. The problem is then you have this positive sense mRNA that can act as messenger RNA, can restart FMDV virus production and infection on its own, and the odds are really bad. I mean, we have huge numbers of intact genomes on these LFDs, and really all you need to happen is that one of these molecules, intact molecule, gets into a cell, and restarts infection. And those are odds, like on the racetrack, I don't think you would want to bet against that event ultimately happening. So we were looking at what was in the literature. Is this theoretical possibility of mRNA getting in an animal, starting infection, is it really just theoretical, or is there a practical relevance to that as well? Unfortunately, previous studies where people looked at this infectivity were always done with some sort of transfection reagent, lipofectamine, lipofectin, or anything else, which doesn't really mirror the scenario that we had in mind. So we couldn't really use these data on FMD. There's one on rabbit hemorrhagic disease with kind of the same problem. And then there was this one PERS study where actually they did inject PERS RNA directly into tissues of live pigs without transfection reagent. And actually there they did see PERS infection in this paper. So we then decided, well, we're just going to do that ourselves. We're just going to look at this ourselves. So we looked at different viruses, addressed their, looked at their transfectability, if that is a word. We settled on AERØHEAD22 in the end, extracted high-quality RNA from it, diluted it a little bit, and then just confirmed, yes, there was a lot of FMDV RNA in there. We inoculated susceptible tissue culture without transfection reagent, no CPE, so we said it was clean, there was no virus. Then tested, can it be transfected? Yes, it can, and quite well too. So then we just went on. We injected this RNA into the tongues of 6 cattle, again highlighting no transfection reagent. And then what we saw, I mean, to our surprise, I guess, that these cattle got really sick, and they got sick super fast. So it was really just within 24 to 48 hours, all 6 animals, high temperature, clinical disease. We found virus generalization very early after infection as well. They all had obviously huge lesions on the tongue where we injected. They all had lesions on the feet. So there really was no difference in this comparing injecting them with virus versus injecting them with virus-free, full-length FMDV RNA, like fulminant disease in all these animals. So that was kind of not really, I mean, kind of what we expected, not what we wanted to see. But it's kind of artificial. You put it in a syringe, you inject it. So we looked at something more realistic, where you take the same RNA and you just drip it onto the tongue of these animals, so another set of 6 animals, and spray it into the nose with a nebulizer. I also quickly examined whether this nebulization had any detrimental effect on the transfectability of the RNA in vitro, because these high-pressure syringes can shear the RNA. We didn't see anything. So this is still full-length, intact FMDV RNA if you spray it, and of course also if you drip it on the tongue. And then we looked at these animals, we looked at them closely for 10 days, and we didn't see anything. There was no disease, no lesions, no fever. They did not shed any virus or viral RNA, and at necrops we didn't find anything out of the order at all. So first group of animals blew up in our face. Second group of animals was perfectly fine. So I think I have to kind of adjust the title of my talk. If full-length genomic RNA is infectious by injection. And so from this, what we conclude, and what I would like to tell all of you who want to use these LFDs, inactivated LFDs for shipment, is that yes, it is, the RNA can be highly infectious if it is injected. The RNA is probably innocuous if animals are exposed to it non-invasively, just like their mucosa gets into contact with the RNA. In our hands, that did not cause infection. So the risk of contagion that is associated with these properly inactivated LFDs, I think, is very low. But it's still generally a good idea to kind of maintain positive control of samples that are being shipped. So that doesn't mean that you have to package them as if they were infectious, but at least kind of slap a tracking label on there. So if it gets lost, if it ends up in a ditch somewhere, at least you have a rough idea of what happened to it. And so to get back to this joint opinion or recommendation of EuFMD from the beginning, I completely agree with the fact that it's inactivated, that it's safe, and also EuFMD has said, hey, please keep track of your samples. You don't want to lose control of them, and I think that is also a very important point to keep in mind when working with the LFDs. Just as a very last bit, I'm out of time anyway. When we were looking at the literature, as I told you initially, we didn't find anything pertinent to our question. And then after we had actually published this, I kept looking at the literature and relevant papers kept popping up. And then I found a paper from 1959 where a person from Switzerland actually pretty much did the same thing that we did without real-time PCR, without fancy lipofectamine or anything. But they did the same thing, and they actually also found out, came to the same conclusion. So it's a nice confirmation. Every 70 years, you have to go back and look at the literature. That's it. Thank you very much.
Thank you. Michael, just thank you very much. Do you have any questions to Michael? Yes, Vina.
Australian Centre for Disease Preparedness. Michael, I mean, this may sound ridiculous, but you sort of think, you know, what if it happens to land in a cow's mouth and they're chewing on it and there's actually, you know, some damage to the tongue? Do you think it's worth trying to sort of, you know, scarify the tongue or do some damage and then drip the virus on and see if it's then infectious?
I would actually like to do that. We were discussing this when we were planning the second experiment. The reason that we ended up not doing something like that was that we just couldn't find consensus of what we should actually do. Like some people said, well, give him an actual LFD to chew on, or give him, I don't know, some glass shards or something nasty like that. Or, as you suggest, like controlled scarification like you would do with a guinea pig foot pad or something. I think if you come up with a good protocol that is standardized, that is kind of reproducible, comparable, it's absolutely worthwhile to look at this kind of intermediate thing between stuff we know that works, stuff we know that doesn't work, and something that may be more realistic and might work or might not. But probably the larger number of animals then, because if you only do it in 2 or 3 or even 6 cows, I don't know how reliable the result is going to be.
Yes, Labib? I'm sorry. Short questions, short answers, please.
I will do my best for the question, but for answer, I let Mackay. Thank you, Mackay, for this question. My question is, I think you performed this test with RNA extraction from cell culture, affected cell culture. Did you try to do this from RNA extractor from inactivated LFD? And my second question, did you try to just to put the LFD inactivated LFD with the pigs or Charleston to see what happened if the pigs would eat the LFD, they would become infected or not? Because this is what maybe can happen in the field. So is the LFD in somewhere, and pigs maybe will eat the LFD, and then, okay. So did you try this?
No, so we did not actually, like I said to Vilna, we did not have any sort of, I don't know, artificial scenario where we give LFDs or parts of LFDs to animals. And I think that should probably be done. To answer your first question, we did not do the lesion material or extracted from the LFD for this because we know, and ANSYS has shown that quite nicely, that if you extract it from the LFD, you have full-length RNA, you can rescue virus by transfection. So I think full-length RNA is fungible in that way. As long as it's intact, full-length RNA, it doesn't really matter whether it comes from culture or not. It's really just a numbers game. I mean, we wanted to have as much high-quality RNA as we could get. That's why we went with the culture. You could do the same thing coming from the LFD, and then if you get like the same amount of infectious, of intact genome off the LFD, I would expect the exact same results. But we went with the culture just because it's easier, quicker, and we thought it would give us more material to work with.
Very, very, very short question from Don. Very, very short answer from Mikael.
Yes.
So, yeah, thanks, Michael. I mean, I think you paint a very clear message, which is, you know,
which is what we believe, the RNA is infectious. I mean, I think it's just worth noting that different countries have different rules. Always the UK is different, but I think the US is also different. Yes. And so in the UK, and I think the US, we would still not be able to receive LFDs in the post from infectious country, from that potentially are infectious, because we consider the RNA still as a regulated material, and so still have to come on IR to category B. And I think that's part of the narrative, and just make sure that when you put the recommendations together that go into the Biorisk Committee, that they consider the different legislation that's in place in different countries.
Very short answer, I completely agree.
Okay. Okay, so thank you very much. Thank you. And now I leave the floor to Sylvia for the panel discussion.
Okay, thank you so much. I think we'll start off with questions again from, but haven't we need to invite all the panelists who are present and even those online. We've now been informed that Andrew Shaw is now online and will be able to join us in this panel discussion. As our panelists come on stage, I would like to share some experiences. One experience was while I was doing my PhD several years back. One of the respondents that I had identified, whom I was supposed to interact with, the time when I arrived at his place one morning, I found that there was nobody in the home. They had all moved in the night. They had disappeared to nowhere. And the reason was that in the previous days, another clinical case of foot and mouth disease had been identified in his herd. And that was not the first time they were identifying a clinical case in his herd. So all the communities around him, all the neighbors came upon him and said, "It is you again. You always bring for us trouble, and you make us lose our animals, you make the markets to be closed." So he faced a lot of pressure. To make it worse, he was part of those that are considered minority or who migrated to the area more recently. And he had a small piece of land. He didn't have much land. So he was always moving around to look for grazing areas for his animals. And of course, we know what that means in terms of spread of foot and mouth disease. So twice he had been caught being the first to have the index case of clinical foot and mouth disease. He'd been harassed, and his children were being harassed. So they made a decision, the whole family and all their animals, to disappear in the night. And when the officials woke up in the morning, they could not, they tried to look for them, they tried to follow them, and they could not find them. And then another case was one of the field vets whose district had reported foot and mouth disease outbreak. Subsequently, each time it was this district reporting. So this time when it happened, some security people went and arrested him and put him in a police cell for about 10 days. Say, why is it your district? It means you're reckless. You're always giving movement permits without inspecting the animals. Of course, what they were doing was supposed to stop the spread. Unfortunately, those two incidents tell us that there are some complex issues sometimes which we cannot see. So the other farmer faced what we would call social stigma from the community, which made him to escape. And when he escaped, we don't know through how many districts he moved until when he found safety. And then this other official, after being arrested, I'm sure he will try not to report that there is a case in his area of operation. So those two instances tell us that I think there is more we need to do to encourage self-reporting by farmers, and also to encourage the field vets to be able to report. Short of that, it complicates timely detection and timely reporting. The other thing that I wish we could fully understand is the role of the airborne droplets in the transmission of the foot and mouth disease virus. And vehicle control. Sometimes we stop animal movement, we don't stop the people movement, we don't stop the vehicles from moving. I hope those issues can be interrogated one time and we understand fully what role they play. Okay, I would like to thank our panelists who are ready to take all your questions. I believe even the panelists online are ready. So I would like to begin with the physical audience. In case you have any question, you can you can mention the panelists you would wish to answer your question. Or if it's a general question, you can still raise it and we will request one of our panelists to respond to you. So can I see if there are any questions from the physical audience before I hand over to my colleague to bring us any questions from the online audience? Of course, we do appreciate the online audience for keeping with us since morning, and we are coming back to you. But let's start from here. And in case we have exhausted the questions from the physical audience, then I'll hand over to him so that we continue with the questions from the online audience. Thank you very much. Okay, I think there is only one hand. Let's have that question. And I will go by the chair's guidance, short question and short response.
Thank you, thank you. It's a comment and sometimes it's a question. As you know, there is many projects that are developed, research projects with endemic cancer for FMD. And these projects are regarding other diseases, not of foot and mouth disease. And many samples travel from these endemic countries to European laboratories. So my question is, what is more dangerous, inactivated LFD or such samples? This question may be for Michael and Don. So I believe that, okay, I understand that the RNA is infectious. This is what we know. People who are working on positive sense RNA viruses know that if you transfect the RNA into the cell, you can get infection. We do this with MCV. But the question is the RNA inside the LFD, inactivated LFD, can come back from the LFD and then go infect animals if the LFD is transferred to another laboratory authorized to handle foot and mouth disease like viruses. So this is the question.
I think the answer is already in the terminology. I mean, the lab is not authorized to handle. Oh my God. Because I can. So the lab may not be authorized to handle infectious FMDV, but there is no infectious FMDV on the LFD. So if we go by that strictly, then yes, you can give that LFD to labs that are not FMDV labs. I would always, as much as you can, try and make it very clear to them that they should only use that for molecular work. So if they want to then do sequencing, PCR, whatever, for some of these other diseases that may be on there, that's fine. But it's not a good idea to put it in tissue culture for any reason. Even if you don't have transfection reagent, we've seen with some cells that small amounts of RNA seem to be spontaneously taken up by the cells, and you can get infection from clean RNA in BHK cells without ever using lipofectamine, for example. So as long as they do molecular work, it's fine. No exposure to tissue culture, no exposure to animals in these lab settings, and then I think that's okay.
Yeah, just to complete what Labib said and what Don said, it's true that in some countries it's forbidden to import infectious genome. But some years ago, 4 or 5 years ago, it was decided one day to check everything entering in France in Charles de Gaulle airport, which, as you know, is a big airport. And they find 500 kilograms of illegal bushmeat coming from everywhere. And in this bushmeat, they find Ebola virus. So one day. So I think this is something we have to take into account into risk assessment, that when you have LFD inactivated, and even I would say if it is not inactivated, because I'm not sure that the probability is that cow itself extract RNA, infect itself, and probably the probability is very low, I imagine. The message is that if you follow the inactivation procedure, the risk is zero.
But there are the legal implications that Don has brought up and that you have confirmed.
Yeah.
We all go for this technology. Michael, just to end the discussion for now, I hope we still have some time for the other topics also. But you mentioned that the risk is small. That's a different terminology than negligible. And that's what we like in the GMO world, for example. And of course, that relates also to the comments of Wilna, that you did not look into damaged epithelium or erosions or ulcerations or whatever. Would you consider it necessary to do additional experiments? And in addition to that, would you like to do similar experiments for FDA cards? Because in my lab, we receive a lot of FDA cards for all sorts of diseases, not for FMD. And there are some publications about FDA cards. Of course, they do have the disadvantage that they are not for diagnosis, but they are a very easy way of transport.
And they're much cheaper than an LFD. I mean, right now, if Labib starts giving them away for free, then that will change. But yes, that's a really interesting question. I don't know, just from my gut feeling, saying something is low, very low, that's a scientific result, but negligible is opinion. I don't know.
Come on.
I think that's something that people have to assess individually, more relevant to what you're looking at, because negligible to me always says negligible compared to other risks involved with the same process, which may be more important and may be a better, like if you have limited resources to address all the risks, do the other risks first, and then the negligible ones come later. So I think in the scenario that we describe, where you take the LFD and activate a chip it, yes, the risk is negligible, but again, there are countries that say no, full-length FMDV RNA is still dangerous, and it's not something that they would be happy to see neglected. I don't know if that makes sense, but that's kind of my take on these two different terms. And to go back to a question about the FTA cards, we actually have done some work on that as well, only in vitro so far. We haven't done any animal experiments, but we've looked at FMDV, LSDV, PPRV, and blue tongue virus on, I think, 6 different, well, they're not, FDA card is one thing, so just call them, I don't know, biosample collection cards to have like a more generic, non-trademarked term for these. And we found that LSDV was kind of hard to inactivate and to reliably inactivate on the FDA cards. But with FMD, if you spot it on the card, you leave it for a day. All the cards that we tested did not allow us to recover infectious virus by direct elution and in culture inoculation. And then we looked at like nucleic acid recovery. You extract the RNA, you do real-time PCR, compare it to the liquid sample that you began with, do sequencing for FMDV to do transfection, and you pretty much get similar results that you get for the LFD. So I would, going back to what I said earlier, that doesn't matter where it comes from, full-length RNA is always the same. I think the scenario, the risks and everything with the FDA cards is very similar to what you see with the LFDs, and you can inactivate the FDA cards on top of what the card already does. If you're worried, you dip the LFD in citric acid, you can also dip the FDA card in citric acid if you want, or heat it.
Yeah, thanks a lot.
Maybe he has a question.
Okay, I'll use this one. Sylvia, you want?
I think there is one more question from here, and then after that you'll move to the online audience, and the questions we'll have a slide uploaded here for us.
usually we say that FMDV is not zoonotic viruses, but we say also that there are some anecdotal cases that people show some vesicle that may be done. So my question regarding your question is it is neglected risk or there exists not exist for FMD as a zoonotic virus? And the question also, if we inject the virus, the FMD to human, we can get FMD. I am sure that one day a virologist told me this, if you take human cells and you infect these cells with the high doses of animal viruses, maybe you will get ECP on these cells. But this doesn't mean that this virus is zoonotic. So maybe the risk, we have to mitigate the risk regarding the context of using this material.
Thank you very much. I think I fully support the conclusions of the Biorisk Management Committee, so let's close the discussion for now. Don, you had...
Well, I was just going to open it up again, but I mean, I don't disagree necessarily with what's being said. I just think that we have to be cautious here. I mean, what Michael's presented is a scenario that proposes this risk. Whether or not it's a plausible scenario, we can debate for a long time. But the point is, I think that most of us working with FMD, particularly live FMD in Europe, are working in high containment facilities. We're working in those facilities for a pretty good reason, because of the risks of potential spread of the virus. And I suppose I'm not that concerned necessarily with LFD and RNA coming back and being infectious. What I'm more concerned is this idea that perhaps as the mission creeps, that you'll have potentially university laboratories and other scientists working with these LFDs that are not experienced with working FMD. They don't understand the risks, and it's more a fomite risk that I'm worried about, not the associated risk, that comes with the RNA. And I think that's just what we've got to be a bit careful in this. I mean, I completely support the ambition about shipping LFDs and making that cheaper and safer and everything. But this idea that it becomes universally accepted that LFDs are sent for testing, I think just needs a little bit of careful reflection, I would say.
Yeah, thanks Don. And there is a nice poster about a transport tool, which also comes into this, a transport tool for inactivated LFDs. So please have a look, because that will at least prove that the sample has been inactivated. Okay. Sylvia, you would like to do the online?
Okay. I hope, is this working now? Yeah, thank you very much. These are the questions from our online audience. The first question, I don't remember who will answer this, but let's read it out and one of our panelists will respond, including the online panelists. The participant is asking whether there are withdrawal periods for vaccines, for instance, FMD vaccines, or this is applicable for only drugs. That's one question. And the second question is asking, do you think full-length genomic FMD virus RNA can be used in cattle-potence testing of FMD vaccine? And the last question, do you think the baseline level of IFN signaling pathways, and I think that is ISGS, is higher in swine compared to other susceptible species, which results in the persistence, I hope that is persistence, okay, in persistence of FMD in pigs. Those are the three questions, and I would like to request the panelists to respond. They're a bit general, I don't know, but let me ask our panelists take time to respond, and I hope our online panelists are able to also view these questions so that they can also make their responses. Thank you. We can start. Maybe we can start from here.
Yeah, so would whoever start with the third question, which is directly addressed to me. I really don't know if there is difference between baseline level of ESG proteins in swine compared to other ruminants. To my mind, I don't think that I've seen a paper, an article focusing on this expression, basal expression levels. But if so, it would be really interesting to do such experiments. And if it was done, if baseline levels of interferon proteins in pigs are indeed higher than in other ruminants, it could be one of the explanation, a part of the explanation for why there's no viral persistence which has been described in pigs.
Yeah, thank you very much. Let me request I think this is Morgan. Oh, yeah. No, I'm mixing up.
Yeah.
Okay, you can go. I think the next question is yours. Right? Yes, you can proceed.
I mean, I think you could. Of course, you're injecting a different material than what's commonly injected and what's been injected in hundreds of potency tests before. So I don't know if it's worth making that switch, losing direct comparability with the old tests. At the same time, I mean, with a tongue injection, you're bypassing, at least at the inoculation site, you're bypassing a lot of the immune response anyway, so it probably wouldn't make much of a difference. But I question why you would want to do that, because sure, you're eliminating kind of like the risk of infection, contamination, whatever that you have with your challenge virus itself. But in the potency test, these, at least the controls are going to become infected, are going to shed virus, and also the animals that turn up to be protected from generalization will produce virus locally and also shed virus. So you have to conduct this in a containment space anyway, so I don't really know what you would gain by using mRNA instead of virus for the inoculation.
I can imagine.
Thank you very much. Continue.
Maybe to the audience, as far as I know, I don't know about vaccines that have withdrawal periods. Anybody else has a different experiences?
Yes, thank you. In Europe, we have withdrawal periods for vaccine as for drugs, but usually it is linked to the consumer safety and the presence of residues, of chemical residues. Therefore, we have only withdrawal periods for some specific cases like zoonotic vaccines, where there is a presence of the microorganism at the injection site. And it is the only example where we have a withdrawal period.
Okay, thank you very much. It's interesting information. I think we have to wrap up. We are a bit late, so despite thank you very much for being on the panel and also online. But I just want to summarize what we have heard. It was very interesting. I think we have learned more about the virus-host interactions, but we also have concluded that it's still far from the field application of antivirals, whatever antivirals. We don't think at the moment that this is feasible. We have talked a lot about diagnostics, and my main conclusion would be that, of course, everything is quite interesting and promising, but that maybe a big proportion of this will end up in the valley of death, and that's what we don't want. So one of the lessons that Don learned me is that we really need to think, and probably you do that already, but then it's a lesson for me, that we have to think clearly what are the needs of the people in the field, both the vets and the farmers. What do they need? What does really help them? And what makes the barrier lower for diagnosing fast diseases? And also to interact as early as possible with the commercial industry or with the industry to see, to optimize the chances to get these things into the market. Anything to add, Stefan?
Oh, fantastic. Thank you. Fantastic summary. Thank you very much. Thank
you. Thank you very much. Then you have a limited time now before lunch starts at 1. but lunch is 1.5 hours, so if you have time left after lunch, you can go back to the posters. Okay, have a nice lunch.
Thank you.
history that it might be an FMD.
And I think we've done a brilliant example of that this week.
What you need to know about foot and mouth disease. Foot and mouth disease is a very contagious viral disease which affects cows, sheep, goats, pigs, and other animals such as wild boar. The disease causes serious production losses, even if not all animals die from the virus. An outbreak in a country can result in a major block to international trade. Humans cannot become ill from the disease, but they can play an important role in spreading it to animals through dirty clothes, vehicles, and other equipment. The signs of FMD are variable and depend on the animals. In cattle, the disease can be severe, and you may notice depression, high fever, not eating or difficulty eating, decrease in milk production, increased salivation, blisters followed by lesions on the muzzle, inside the mouth, on the feet and teats. Occasionally, it can cause sudden death in young calves. In pigs, the disease can be severe, and you may notice an inability to stand, with some animals in a sitting position, lesions on the feet, sudden death in piglets. In sheep and goats, the disease is not that evident, and you may notice small blisters just above the feet. Sudden death in young sheep and goats. FMD can be found in all fluids from infected animals. The virus can spread very easily through any of the following ways. The most common is through direct contact between an infected animal and a healthy one. Contaminated materials such as hay, feed, water, milk, urine, and others. Dirty clothing, footwear, or equipment. Contaminated pens, buildings, or contaminated animal transport vehicles. Meat or other products which come from diseased animals, if fed to animals when raw or improperly cooked. And in certain weather conditions, it can spread through the wind from a nearby farm. Contact your veterinarian or local authority immediately if you suspect your herd is infected with FMD. Do not move any animals from the farm and limit any movement of people to and from the farm. A veterinarian will come and look at the animals and take samples to send to the laboratory. The laboratory will examine the samples and confirm the presence or absence of the virus. Once FMD has been confirmed, measures will be put in place to stop the disease spreading to other farms. Farmers whose farms or animals have been in contact with the farm where the disease is present will also be contacted by the local authority or veterinarian. Everyone has a role to play in ensuring that your community and country remain free from the virus. By staying alert and reporting suspected cases of FMD, you can make a difference. Thank you for your attention.
suggested that it might be an FMD.
And I think we've done a brilliant example of that this week.
Good afternoon all. Before we start our third session, I would like just to thank the participants to the workshop on fast risk monitoring for the fruitful discussion that we had and the inputs provided. So we are starting our session, the fourth session of the Open Session 2022, which is focused on vaccinology, and will be chaired by Professor James Wood, and member of the Standing Technical Committee. James is the head of Department of Veterinary Medicines and Professor of Equine and Farm Animal Science at the University of Cambridge, UK. And James has been assisted in the preparation of this session by David Mackay and Martin Ilott, both experts in vaccinology, vaccines, and also leading the project that we have on the pre-qualification for FMD and similar transboundary animal diseases vaccines. The session will be moderated by Martin Ilott, assisted by Buddha Hamadi. Thank you, and James, the floor is yours.
Fabrizio, thank you very much for your kind introduction. It's a great privilege to be here, and it's a great privilege to welcome you all to this session this afternoon, which, as you've just heard, is on vaccinology. The focus in this huge field of vaccinology is going to be on innovation in vaccine technology, not just for foot and mouth disease, but also considering our other fast diseases as well. We need to think about innovation, but then within that context, we need to think about safety, about maximizing efficacy, and cost effectiveness. It's not enough to produce a fantastic vaccine that no one can afford. One of the great challenges with innovation and moving from the conventional vaccinations that Martin has just pointed out that he started his career with, and I think he's got a bit to go, but he thinks he might finish his career with, is that the regulatory processes around them will need to be modified as well. So there will be challenges and barriers in terms of introducing innovative vaccine strains, innovative technology. And we need to think about those. We need to think about the new frameworks that might be needed to take these on board, and also to consider among the broader context of that, how we ensure there's rapid sharing of relevant strains or relevant strain information where we don't need the specific strains because of the technology that might be used. You all know from the program we have a great panel here, and then we will have a great panel discussion after this. I'm very pleased to introduce the first speaker for the session, Professor Brian Charleston, who is the director of the Pirbright Institute, and really I think needs no introduction to any of you. So Brian, over to you. Thank you.
Great, thanks James. And already said, this is a huge topic, so the idea of trying to cover this in a relatively short talk is it would be challenging. So I'm just going to try and pick out some key messages around some of the challenges and some of the opportunities around developing innovative vaccines. So the first, I mean, I'm sure everyone in the audience will know this, and you could debate whether I've got the order quite right in terms of development costs for the various platforms, but inactivated vaccine, easiest thing to do, grow it, kill it, add an adjuvant and use it, and that can be quite appropriate for many diseases, as we know, many successful vaccines. And I think the next one is alive attenuated by growing in an unnatural, the pathogen in an unnatural host or host cells and it becoming attenuated through a natural process, almost a little bit stochastic in the way that the vaccines develop, but again, extremely useful. The Plowright vaccine that resulted in the eradication of rinderpest was an example of that, so can be very successful. But then when talking to many colleagues working in the vaccine space and defending the reason for doing our work is that in some ways the easy ones have been done. Now we've got some really significant challenges to try and develop vaccines for where there isn't a good solution or there's no solution. And then you start moving on to some of the other technologies, such as recombinant subunit vaccines, so recombinant molecular biology, and then expression, and then trying to identify the subunit that will induce protection. And then rationally attenuated, the reverse genetics being available for particularly the RNA viruses, and probably that's the way we'll get an African swine fever virus vaccine, is through rational attenuation and by understanding the biology of that virus to have targeted mutation, deletion that cannot get reversion, so potentially very much safer. And then RNA vaccines, I don't think they will be intrinsically, development costs are high, but in some ways there's a barrier to the community getting involved in developing particularly RNA vaccines in light of the success of COVID, because of the trying to get around the IP that is around those vaccines, and a lot of initiatives to try and drive down the cost of goods of those vaccines in light of their success. So yes, traditional technology should be looked at in the first instance, but then I think there are examples which I'll give of where we've got to think slightly differently and use the more innovative techniques that are becoming available. So one example there is a program I actually worked with many years ago with Gregor Myers from the Friedrich Loeffler Institute and Boehringer, was bovine viral diarrhea virus. So this is an example of rational attenuation to produce a recombinant live attenuated vaccine. So this is the 5' end of the virus shown here, and you have this N-pro protein, and also you have this viral glycoprotein, which is also an RNase. And through the removal of the N-pro and removal of and making a targeted deletion in the eRNS to stop it having RNase activity, similar to the talk we heard this morning from our friends in China, this virus now cannot control the interferon system, so it can't. The virus usually does not induce interferon, but this recombinant virus does induce interferon, and so attenuates itself and is so very safe, and yet can stimulate a protective immune response. So I think that's a very nice example of where this targeted attenuation can work very well. I haven't kept up to date, but I assume it's still a very successful vaccine. Another example here is relating to foot and mouth disease virus, and some work we've been doing initially amongst ourselves, and then brought in Merck Animal Health to commercially develop foot and mouth disease vaccine. So instead of using the grow it, kill it approach, is to express the capsids using a baculovirus system. I think the first thing to say that you need the intact capsid to get a successful foot and mouth disease vaccine, and you need that capsid to remain intact. And by not being dependent on the virus life cycle and not being dependent on uncoating, you can really, through knowledge of the structural biology, really make this a very robust structure. And we've got these capsids now that are stable at 56 degrees C for many hours, which drives better immunogenicity and stability. So I think I don't know if Aldo's in the audience. He always says to me, why are you doing this? The conventional vaccine works well. I think I would say, yes, it worked well in Europe, and Europe was able to control the disease using that vaccine. But certainly what we're seeing is that some of those very unstable, particularly the SAT viruses, the titers, neutralizing titers we're getting with stabilized VLPs, I don't think you'd achieve with conventional vaccines. So another example of how the latest technologies can, I believe, will move a step change in the performance of those vaccines, but it also allows me to introduce what I think is a very early but very important science, and that's structural vaccinology. So by detailed understanding of the structure of these pathogens, we learn how to maintain or alter the antigenic structure of those antigens to drive highly protective immune responses, particularly B-cell responses. And the example I wanted to give here is a bit abstract in many ways, and this is a protein. This is a glycoprotein of respiratory syncytial virus, a major problem in both animal health and human health, different viruses, but similar biology. And this is the fusion protein, if you like. This is the protein that the virus uses when it wants to engage with the host membranes. It goes into this post-fusion state and forms a bridge, and that's how it enters the cytoplasm. And when in isolation, the isolated recombinant proteins, the low energy state is this extended state, the so-called post-fusion state, and you will induce neutralizing antibodies against it. But some really clever work done in the States by Peter Kwong and his group showed that if you could lock, by making mutations and knowledge of the structure in the pre-fusion state, this alpha-4 epitope drives highly potent neutralizing antibodies. And we've made the bovine version of this and immunized cattle with it. And it's clinically, pathologically, the animals are fully protected, and microbiologically too. It performs extremely well, better than the current vaccines. It's a bit of a bold statement, but it does. It's really interesting. Back to, again, James saying there are many hurdles. We can't get interest from commercial companies for this. And one of the reasons said to us is that these are multivalent vaccines, and changing one component of that multivalent vaccine is quite a regulatory chore. So, maybe I'm biased, but I think we've got a fantastic new vaccine there that we can't get through into the marketplace. And this similar strategy could be applied to things like Newcastle disease virus, Pestipediruminant. Most viruses have a similar type of approach to enter the cell. So again, you've had two examples already, I think, of how the structure can really be used to improve the performance of the vaccine antigen significantly. I'll come back to that in a minute, actually. And then another example, thinking about new platforms. We all know about the adenovirus platform and the AstraZeneca ChAdOx technology. We've been working on this vaccine platform. Actually, George Warimwe from Kenya is driving this program, is to try and get a better Rift Valley fever vaccine. So there are Rift Valley fever vaccines. There appears to be a safety issue around the use of those live attenuated vaccines in pregnant animals. So again, sidestepping that and putting the glycoproteins into the adenovirus backbone, and then demonstrating that that's safe, efficacious, compatible with DIVA, and indeed safe and protective in pregnancy. So back to the first example I gave, is rather than use those traditional techniques, sometimes using those new innovative technologies can get you there faster and give you a safer and more effective product. what I wanted to come back to, and I was talking about structural vaccinology, so this is the third example actually, and not the easiest slide to interpret, but the reference is there back in the height of the COVID outbreak, or the start of it. And so the point I'm trying to get across here is that, yes, you can take a sequence like we did with Rift and put it into an adenovirus backbone, but there's real value, there's tremendous scope to even get greater improvement here. And let me try and decode this particularly difficult slide to understand. So down the bottom there, you have neutralizing antibodies induced to a single shot of the Johnson & Johnson Ad26, the human adenovirus platform, just a slightly different one from the ChAdOx. So these are the neutralizing antibodies after a single inoculation with that viral vector containing the SARS-2 spike protein. And you look at the variation of neutralizing antibody responses against a log scale. I mean, huge, huge variations. And then if we go up to the code at the top, so you've got S1 and S2 and the junction between them on the top red bar of the spike protein. And so one option would be to just put an even more potent promoter in front of the spike protein. Well, that actually gives you a very poor neutralizing antibody response. But as soon as you start to put that, PP is the double proline, and that's the two bars over to the right, two red bars. So by introducing two prolines into S2, you can stabilize that structure, just as I described with FMD and just as I described with the F protein. And it's that stabilization is now really giving you that potent neutralizing antibody response. And there's also a modification of the junction between S1 and S2, so you don't get natural cleavage there. But you can go down and look at the things that actually make a difference. Just removing the C-terminus, the DCT, that also gives you a lower response. If you remove the whole of the transmembrane domain, so this is now going to be a soluble protein, so that's the DSDTM-PP. Again, you don't get such a potent response against the insert that went into the final product. Was that with the full length, using the original adenovirus promoter, removing the fusion between S1 and S2 site, and then adding the stabilizing mutation. So maybe we got a bit lucky with the Rift one, and we chose the right insert at the right time. Maybe it didn't need any more stabilization, but the point I'm trying to get across is there's real opportunity now, especially with knowing the structure and how we can modify it, and also being able to interrogate the antibody response, not just neutralizing antibody response, looking at the real profile of the antibody response using single-cell sequencing and antibody expression, and then relating that back to the structure. There's a tremendous opportunity to solve some of these challenges. Anyway. So we had an opportunity actually during COVID in that we developed the pig model for studying vaccines, particularly for flu, and developed a vast array of immunological reagents to study this. So we had the opportunity to look at 3 COVID vaccines in the same genetic background of pig using the same assays. So here's the ChAdOx vaccine that we did first. And what we did very early on was showed that if you boosted at 28 days, the original plan was for this to be a single shot. We showed at 28 days you get a significant uplift in ELISA antibodies, VNTs, etc. And then through discussions with Andy Pollard, they introduced another cohort into that study, and that's how that vaccine was now deployed. But you can see the level of antibody that was induced there. We also tested another vaccine, and this is using a bacteriophage VLP, not like the whole pathogen VLP, but you're decorating a virus-like particle acting as a carrier to get multimerization of antigens using the receptor binding domain of SARS-CoV-2. And either 5 micrograms of that RBD VLP or 50 micrograms or 100 micrograms of recombinant spike on its own. And look at the difference. I mean, you're getting very similar responses, but going right down to 5 micrograms of that antigen on a virus-like particle where you get better presentation and stimulation of B-cell responses, you're getting the same response. And that wasn't taken forward by UK government, but that's been taken forward by some international companies to see whether that's going to be viable for a vaccine platform. Then we also tested a self-amplifying SARS-CoV-2 vaccine. to see what sort of response that would get. And it's a busy slide, and we're getting close to the end of the time. But you look at the top right-hand corner, and you'll have to remember now that with the other vaccines, we're getting over 10 to the 3, nearer 10 to the 4 VNT after prime and boost. And we weren't getting anywhere near that with that vaccine platform. That's a different platform from the Pfizer-Moderna vaccines. This is a self-amplifying, which the hope would be, because it does self-amplify, it would give a better response. But there are problems there. We're not getting that response, and one of the reasons may be that you're actually stimulating the interferon system back to getting around the interferon response again. So potentially a good idea, but again, more work needed to get that technology up and running. Okay, final slide then. I've got 16 seconds to go. Great. So this is Pirbright, drone shot of Pirbright. I think many of you will know the campus, but just to show you the big dark-coloured building in the front there that will be open next year. So this is our new high containment building that will be able to handle SAPO4 agents, African swine fever, FMDV, but for the first time we'll be able to handle ACDP3 human pathogens in livestock, such as Rift, etc. So quite an additional facility that will be available at Pirbright. Thanks. Look at that.
So Brian will have spoken to many of you before, and you will always know he's a strong proponent of vaccination, but I've never heard him try to use prayer halfway through his talk. So I'm keen to try and keep all of the discussion at the panel later. So if we can release Brian and move to our next speaker, who is Owen van der Born from MSD Animal Health in the Netherlands. Owen, I think, also needs little introduction to you. Very well-known expert in this field, and Owen, it's great that you're here to talk to both the audience here in the room, but also, as Brian did before, the online audience as well, which is such an important part of this meeting here. Thank you very much.
Okay, thank you, James. Good afternoon, everyone. I hope you're not suffering too much from the after-lunch dip. I've been in the pharmaceutical industry since 2010, and I've been responsible as a project leader in R&D for developing vaccines, mainly against pig diseases. And during those years, I have come across several vaccine strategies coming in and out, and some have been implemented. But in that capacity, I would like to share my view on new vaccine technologies. Going back to the title of the talk, New Vaccine Technologies and Fears and Hopes, I think COVID was the greatest example of that. On one hand, we had hope that we could get out of the COVID crisis, and especially we as scientists, we were believing in the vaccines. But on the other hand, there was fear in society that there are safety concerns connected to those vaccines. And well, you have all seen the protest in different countries. What are those new vaccine technologies? And Brian already mentioned a few. But before I do, I first want to, again, go back to the hopes and the fears. So the hopes that we have for new vaccine technology is that it brings better, longer protection, and it's more safe, and the vaccines are cheaper to produce, more stable, all of that. But on the other hand, we have the fears, and the fears are not coming only from the public, but they also come from the regulators. So from the regulators is mainly the GMO aspect of certain vaccines, or the fact that certain vaccines stay persistent in nature. But on the other hand, you also have fear, just the general fear of the vaccines. And one of the things, for instance, is, well, one of the last points here on the slide is the long-term negative effect on individuals. So the vaccine technologies, they are mentioned on the left. Nucleic acids, all known to you by now, messenger RNA or DNA, subunit vaccines, live attenuated, inactivated, peptide, and many, many factors. I have mentioned here a few on the right-hand side. There are vaccinia, herpes virus factors, alpha virus factors, VSV, and there are many more. And it really depends on what you want to reach in the animal or in the human, what type of immune response you want to trigger, more B cell, more T cell, or a balanced response. And well, you have a whole set of vectors to choose from. Why actually do we need new vaccine technologies? As Brian said, we had those classical ones inactivated. They were working. But we started to work on different ones. Second generation vaccines, the subunit ones, for instance, and the third generation vaccines, the viral vector platforms and the nucleic acid platforms. We started to work on that because the first generation vaccines had their limitations. They were not efficacious enough, they were not safe enough. And what is also very important is the slow response towards outbreaks. For instance, if you have to develop a live attenuated vaccine, you can do that in the classical way by passaging in cell culture, or genetically modify it, and you have to test, and that takes a lot of time. And often you're not successful because there's no good balance between safety and efficacy. So we had to look at different technologies. But the challenge with those second and third generation vaccines is that we need to understand what provides protection, especially if we take a small part of a pathogen. And I've shown here on the left-hand side several pig viruses. And you can see that the lower two ones, the circovirus and the parvovirus capsids, they are very simple. It's just one protein. You can make virus-like particles, and these particles protect like anything. It's working very well. But you go up in complexity, and now we are at the problem in the swine field is African swine fever. It's such a complex virus. There's so many genes. Just tell me which of the 170 genes is providing protection. What of those 170 genes, which one do you have to put in your messenger RNA platform or your DNA platform or your vector? It's a struggle. The same for also very complex organisms, the bacteria. The same principle applies. But there are also successes. So there are not only limitations, there are certainly successes. And here on the right-hand side is probably the reason why we can all be here in the room. It's the COVID vaccine. The spike protein is providing excellent protection. That's not true for all coronaviruses though, but for this one it's working well, luckily. On the left-hand one, the one that Brian already mentioned, one I have been involved in quite heavily in the last years, is the virus-like particle platform for the mouth. And as you can see in the table, similar amounts of classic antigen and VLP antigen provide the same level of VNTs and the same level of protection. But it's easy to produce, and it has many more benefits, as Brian said, in terms of stabilization and all. There's another platform I want to introduce. It's one that is in MSD Animal Health. It's the Sequivity platform. It's on the US market. And that platform is a kind of on-demand platform. So, for instance, a farmer, a big producer, has a problem with influenza, and they can send a sample to us, and we sequence it, and then we put the protective gene inside this alphavirus vector platform, and we send back a vaccine. And then this producer can then vaccinate specifically against the pathogen that's circulating in those herds. And yeah, it works very well, as you can see in the graphs here below. We get good protection and good induction of immune responses. And we can do this because it's a platform. We can do this with basically any pathogen of which we know what is driving protection. And it can be done in basically a couple of days, from sequence to vaccine. Oh, it's animated. There are some limitations with new vaccine technologies. Some are related, for instance, to anti-vector immunity. Some adenovirus vectors are very sensitive to anti-vector immunity. So boosting animals is going to be very tricky. or if the mother animal has been vaccinated, then the antibodies in the offspring are preventing a good uptake of the vaccine. But new vaccine technologies can also have new possibilities, like combining technologies. There have been some reports that is very powerful, that, for instance, combining a DNA vaccination as a prime and coming back with a vector for the booster. What makes a vaccine a successful vaccine? And I want to share a little bit of light on this for the veterinary field. These are rough sales figures for vaccines in the different areas. A dog owner is willing to pay a little bit more than, for instance, a pig owner. And a poultry owner, again, much less, because it's about many, many more animals. If you now take into account, for instance, with a new vaccine platform like the messenger RNA vaccine platform, if I'm right, the cost price mightn't be now around 2-3 euros. You can already tell that at this moment, a messenger RNA platform for pigs or cattle is going to be very difficult. If we talk about new vaccine technologies, we should not only talk about the antigens, but also about adjuvants. And in that field, there has been some progress as well. And this is one of the examples for the messenger RNA platform. The lipid nanoparticles, they encapsulate the messenger RNA, it protects them, it brings the messenger RNA to the cells. But at the same time, these lipids, they also stimulate the immune response. So it's about a kind of dual mechanism that these LNPs provide. Another way of looking at new vaccine or adjuvant technologies is actually using virus-like particles. And virus-like particles at the moment are used in many areas in research and applied applications. But the one in vaccinology is about using the virus-like particle as a carrier of epitopes, protective epitopes, bringing those epitopes to target cells or presenting those epitopes in a structured manner to the immune system. Another aspect of new vaccine technologies is the application. And I think, especially in the veterinary health industry, the application is at a different level at the moment compared to human health, for instance. Looking at the left picture here, this is in a poultry farm where chicks are being vaccinated. It's just by spray vaccination. You can vaccinate many thousands of animals in a short period of time. And in swine, we can use devices, needle-free devices, to deliver the vaccine into the skin, where most of the immune cells are, compared to muscles that are just a locomotor organ. And in fishery, they are moving from manual vaccination towards basically factories of fish vaccination, where the salmon, for instance, come in and they go through the system and they get vaccinated at the right spot for higher throughput. I want to conclude with this last slide about new vaccine technologies. There are many more aspects to talk about, but I think this is also a very important aspect. It's the regulatory landscape. Classic vaccines are developed through different stages. You have to do clinical trials, preclinical trials, safety studies, all that things, and this takes a lot of time. But now with the new platforms, the plug-and-play platforms, we are able to use one production process with the same upstream, the same downstream, the same quality control tests. And it doesn't matter which sequence you have in your platform. And that means that certain tests might be shortened, certain of those phases might be omitted, and we can move much faster from a disease to a vaccine. Or as the Sequivity platform shows, we can basically provide a customer a vaccine in a couple of days. Also important for the regulator is the safety, and one thing that I want to highlight is, for instance, the DNA vaccines. It's now advised that you check whether the DNA vaccine is integrated in the genome of the vaccinated individual. So I want to conclude with saying that new vaccine technologies have brought us many possibilities, that they have improved the safety, the efficacy, and the time to market. But we also need to be aware of the limitations of certain of these vaccine technologies. And I think we, as members of the veterinary health community, we are in the lucky position that in animal health things go a bit faster than in human health, because they are very, of course, they're very cautious. But we can move faster with new technologies, new devices, etc. So thank you.
So unless there are any questions for immediate clarification, I'd like to move on from Owen's really interesting talk. And I don't see any hands up in the audience. So Owen, thank you very much indeed. Thank you. So the third in our set of keynote speakers in the first part of this afternoon's session is Pascal Hudelet. Pascal, as you all know, works for Boehringer Ingelheim, and I hesitate to mention this given what a large online audience we have, but we should thank Boehringer very much for the very nice evening that we had last night here in Marseille.
We did.
Yeah. Pascal's going to be talking about frameworks for access and benefit sharing of innovative frameworks. A very important discussion point where we're thinking about new vaccine technologies coming in. Pascal, over to you. Thank you.
Thank you. What a joy it is to present in person. Sorry for those of you who are online. I like you too, but there's nothing like being able to look you in the eyes. Even though for some of you they are closed. I don't know if it's the effect of lunch or the pastis we had yesterday, or maybe the subject of my talk, because I'm going to talk to you about the fascinating subject of the Nagoya Protocol. But I hope that by the end of this talk, you realize that this is important and it concerns each and every one of you. I will start with a tribute to my friend Claude, who's not with us today because he passed away tragically in an accident. He was a brilliant mind, a good colleague, simply great guy. All of us who had the pleasure of working with him for the past 20 years can confirm this. I wanted him to be with me on stage for one last time. He was my friend and I miss him a lot. So long, Claude. Now to jump into the subject. You remember probably that a little more than 2.5 years ago, there was the first report of a Chinese respiratory viral disease circulating in Wuhan. It took the Chinese scientists slightly over a week to publish the sequences on open databases, which allowed Moderna to start the development of their vaccine in less than a week, 3 days later. Now, the speed and efficiency of this process is just unbelievable. It's unprecedented. But I have to say, this was a serious breach of the Nagoya Protocol. If the Chinese had decided to follow the steps that are mandated by the Nagoya Protocol, we would not have had vaccines as quickly as we did. Quick introduction. What is the Nagoya Protocol? The Nagoya Protocol is a part of the Convention on Biological Diversity. It's an international agreement whose objective is the fair and equitable sharing of the benefits arising from the utilization of genetic resources. It goes back to, it was signed in 2014, and it basically says that a genetic resource is owned by the country where it's been found. And a genetic resource can be plant, animals, microorganisms, and for some countries it means sequences as well.
In practice, you have to, before accessing a genetic resource, you need to ask the authorization from the country of origin. And then you need to negotiate with the country the terms and conditions of its use. I hope you're all doing that. It's not just for manufacturers. If you do research and somehow your research gives you benefits, like a grant or something, you are subject to the Nagoya Protocol. I have to say that that Boehringer Ingelheim is really at the forefront on the compliance to Nagoya. We have high awareness in all our scientists, and we have a complete set of procedures on how to access genetic material. But this is the picture. Majority of the countries of the world have signed and are now party to the Nagoya Protocol. Only a minority of the countries have actually translated those principles into their national law, but and only very few of them make exceptions for pathogens. The intention of the Nagoya Protocol was really good, really good. It was preserving biodiversity. And by that, I mean protecting rare plants for the rainforests, preventing wealthy countries, big pharma, to plunder the riches of developing countries. But unfortunately, the impact on vaccine development, the resulting impact for us was we saw an exponential increase in the bureaucratic processes to access genetic resources, and we had to stop the development of many vaccine developments. We are stuck like a container ship in the Suez Canal. That's for my Egyptian friends here. Overall, it's been quite bad for innovation, which is the theme of this year's open session, isn't it? I'll illustrate this with a couple of case studies from that happened to us in our EuFMD vaccine development efforts. The first case is we tried to access a strain from an African country to include it in our vaccine. We contacted the person in charge, the Nagoya focal point, to ask permission to access the strain that they had sent to Pirbright. Unfortunately, the national law was being discussed, voted. There were great uncertainties on the scope, the potential retroactivity of the law, and they came back to us and asked for benefits that were not compliant with our processes. They wanted plane tickets for conferences and stuff like that. So we were unable to start R&D on this strain for 4 years. So now we've given up, no agreement. We need to start from scratch with another strain from another country. Another example, we tried to contact a country in Southeast Asia, sent our request to the Nagoya focal point, but the Nagoya focal point belongs to the Ministry of Environment. And we've kindly asked to access the strain, and they did not respond. No response. We've tried. We've banged on their door. Nothing. No response in 2 years. We have not been able to start R&D. Now we know, we understand that there's a very complex process in place, but it involves 2 ministries, agriculture, environment, and you need to involve a local lab and a local industrial partner. Well, you need to set up a local project team with legal advisors. It's going to take you more than a year. So we've given up. We have to start with another strain from another country. So part of the problem is that EuFMD is a priority for animal health. It's a priority for all of us, right? But it's not the case for our colleagues from the Ministry of Environment. So in the end, countries either don't respond to our requests or they provide us with unrealistic demands. And it's a sensitive subject here, I know, because you're dealing with developing countries versus wealthy countries, big pharma, and people tend to really overestimate the actual value of one strain that's going to be part of a big combo and not sold all over the world, just a few countries. And it's overall, it's a dynamic virus evolution, and in front of that, you have more than 3 years of negotiations with the country, with uncertainty, and 2 years of development. So, bottom line, 7 years after the start of the Nagoya Protocol, no success with any ABS agreement for new vaccine development, and we are at a complete standstill of our development of new FMD vaccines. And it's not just for FMD or animal health. There are similar examples for human pathogens. There's been published delays for Ebola, Zika, and influenza. And those have major knock-on effects on public health. Seasonal influenza is on a strict schedule, strict timelines, 6 months to from the sequence to making the vaccine available. Any delay of 1 week means a delay in the availability of the vaccine. And for COVID-19, if the Chinese had decided to negotiate with each country that wanted to access the strain one by one, then we'd finished, eventually we'd have vaccine because the virus spread to other countries. But there was an interesting paper that said that a delay in 1 month of sharing the virus samples could have led to an additional loss of 400,000 lives. So, impacts for fast diseases, the subject of today, creates a complex and uncertain environment. You can't keep up with 137 national laws. There are loopholes, and they're being exploited. The returning traveler bringing a germ with him and making it a national resource, all you need to do is import some incubating animals from Venezuela, and suddenly it becomes an Egyptian national resource. It's a threat for innovation. It means longer timeline for vaccine development, less data available in public data banks. It creates disincentives to invest in this type of diseases. Companies hate uncertainty. They see it as risk. So they'd much rather invest in other programs that are much safer, like a new flea product for dogs. No risk in that. Eventually, it undermines the objectives, the benefit-sharing objectives on the Convention on Biodiversity. It's much easier to work with a country that has not signed the Nagoya Protocol. So, solutions, they exist. The pandemic influenza preparedness framework is interesting. 2 roles, 2 objectives, share influenza viruses with human pandemic potential, increase the access of developing countries to vaccines. Does not apply to seasonal influenza, does apply to COVID now. It's coordinated by the WHO, GI, SRS, and they have a template MTA. So if you want to access a genetic resource from a lab from the network, you just sign this MTA and you deal with the WHO. You're supposed to provide benefits to them, and the benefits are used to prevent or control pandemic influenza. And there's many ways that these benefits can occur. So for fast diseases, few recommendations. First, as users of genetic resources, we must agree on a few key principles. First, we need to agree to share the benefits in a fair and equitable way. We will comply with the Nagoya Protocol. We want to comply. It's just then the uncertainty in the timeline that's killing it. So we'll share benefits in many ways. There could be joint research programs, could be training, technology transfers, licenses, supply of vaccine doses, antivirals. Yet it needs to be fair to both sides. The principle of return on investment also applies for fast diseases. In exchange, what do we need from the international scientific community? What do we need from you guys? We need all to agree on some key principles. Timely sharing of pathogens, sequences, and data is essential for surveillance and for development of vaccines and diagnostics. Fast diseases, transboundary diseases, they know no borders. So it doesn't make sense that they fall into the same category as the other genetic resources. They cannot be considered indigenous to a country. Therefore, they should be seen as a common good, and ultimately they should be excluded from the scope of the Nagoya Protocol. Second, we need you to act as advocates. with your countries, with your authorities, to facilitate access to strains and sequences. You need to raise awareness on Nagoya. You need to raise awareness on the importance of vaccines, of sharing sequences, and ultimately you need to help us push to exclude Nagoya from the Nagoya protocol. Sorry, fast diseases. But we're not naive. We know it will take a lot of time. And anyway, there will always be national regulations that we have to comply with because they override the Nagoya Protocol. So we still have to comply with those. What we need is an international agreement, an international instrument to cover the sharing of fast pathogens. Could be a template MTA that countries can use and sign with the user. And please provide some models for benefits sharing so that we can educate countries on what they can expect from such sharing, what the benefits for them are. And I'll conclude with these statements. The Nagoya Protocol was designed to support sharing of benefits. However, it's created a set of huge constraints that really hinder innovation on fast diseases. We need the support of WOHA, we need the support of EuFMD, of the FAO. All the international organizations need to push to exclude fast pathogens from the scope of Nagoya. And in the very short term, what you can do is come up with an innovative framework for fast diseases to encourage countries to continue sharing their materials and their data. Thank you very much.
Pascal, thank you very much. That's a really important talk, which I think probably we're going to hear more about in the future. And I think you might say unfortunately, but it's really important for us to think very carefully around this. We are 2 minutes ahead of time, but only 2. Good. So why don't we move really straight to our panel discussion? Right. And that will be simply affected by the movement of a few chairs. Yeah. Okay, thank you. So if the panel would like to come up onto the stage and join me and the 2 moderators, then we can start this off. Yeah, exactly. Yeah.
Good afternoon, everybody. I'm Martin Eilert, moderating the session, the panel session of session 1 with Buddha. We're both members of the EuFMD team. I think we've had an introduction to all the panelists, apart from David Mackay, and I'm sure most of you will know David. He's been involved with FMD and exotic diseases for most of his career. For those who don't know David, he's a vet from the UK. He did his PhD at London and then moved to the Purbright Institute and spent many years there researching FMD and developing diagnostic tests, and ended up heading the institute before moving then into a regulatory field where he led the licensing department at the Veterinary Medicines Directorate in the UK, and then moving to the European Medicines Agency, where he led the veterinary division for many years. David is now retired, but what I see that he's working pretty much full-time, supporting governments and non-profit organisations, advising them on regulatory and strategies and FMD and other exotic diseases. So I'd like to welcome the panel. Just to remind everybody that you can feed your questions through the app. And those who are joining us virtually, again, we can collect those. Any points you wish to make, we will collect those and feed them to the panelists during discussion. So it's been a really excellent set of presentations. I always feel a little bit uncomfortable when you have spent most of your career in the regulatory environment, and then you join a research meeting. You feel that there's some frustrations, I think, in getting vaccines to the market. But I always defend the regulatory world. I think it does a really excellent job in ensuring safe and efficacious vaccines of good quality, and that's basically what we all want. So, and the legislation surrounding the regulation of veterinary vaccines, it goes out for consultation. Stakeholders all have an opportunity to comment, and I think those fundamental principles of a consistent quality vaccine, safe and efficacious and effective, are supported by everyone. So I will then now move on. I think I'll open up the discussions. I think it covers a topic of the innovation that's driving this meeting, innovative technology for and vaccine platforms, but also some of the challenges we face in developing new vaccines with new strains. And Pascal, you gave a really good presentation on the background of the Nagoya Protocol and the difficulties that you as a company are facing. So, so I think on the issues surrounding the COVID, and I think again it is such a success story. I had a sort of personal view of it. I mean, I think we all have our personal views of it, but I'd moved myself and my family to Australia in 2019, and then watched with some concern as this new virus sort of news sort of spread about this new virus, firstly in China, and then it spread across Asia and into Europe, and causing sort of a lot of tragic deaths right across the world, and the drive to create a vaccine in a very short space of time. And my first thought was I was very lucky to be in Australia because of the biosecurity measures there, protected myself and my family and the people who work in Australia and live in Australia. But secondly, the thought of them developing a vaccine within 12 months, I thought, was just hope rather than any ability to really realise that. And I think to see a number of human manufacturers develop innovative vaccines to scale up that to industrial scale, producing hundreds of millions of vaccines, developing the processes, developing it to the standards of GMP, and then getting regulatory approval within 12 months is an astonishing story. So on that point, I will sort of open the floor, I think, to all the panel to say, how do we sort of capture that experience, and translate the success and the knowledge gained from developing and registering an innovative vaccine in the human field to the veterinary sector. And I will probably start with Erwin. And it's also interesting that the CEOs of Pfizer, one is Alex Borla, he's a vet from Greece, Thessaloniki, who sort of led the company in developing that vaccine. And AstraZeneca is a French vet. So again, perhaps there's a veterinary sort of connection there which we can sort of capture and sort of feed that into the veterinary world as well when we're developing veterinary vaccines. So Erwin, perhaps you could sort of comment on the sort of how we sort of translate some of the things we've learned about COVID and what do you think the major challenges are, and then sort of translating that to veterinary vaccines. Thank you.
Yeah, thank you. I think the COVID was an exceptional situation that made us all focus on the disease, and you saw that the pharmaceutical industry and the scientific world was basically dropping work, and the regulatory bodies like EMA, they were focusing on getting this product on the market as quickly as possible. If we want to do something similar in veterinary health, I think there has to be a reason for it that we all move very fast. So I think that was one of the key points why it went so fast. It was the absolute focus on getting it out on the market to the public. You see that for these technologies that have been used, the vectors, but especially also the DNA, the mRNA vaccines, they could be applied to animal health at the moment, but I think we still have to work on the cost of goods to make it available for food-producing animals. But I see on the front of regulatory aspects of the regulatory framework that, for instance, EMA is now focusing on platform technologies, or want to make that more easy. So I think there, COVID really made a difference, and platform technologies will be more applicable and easier in the regulatory process.
No, thank you, Erwin. And Brian or Pascal, do you want to add to the comments of Erwin?
So my view of this is we were very fortunate as a coronavirus. So huge amount of veterinary research, primarily. And then latterly, because of that knowledge of coronaviruses from the veterinary field, because they're major pathogens, and we had a dummy run, if you like, with SARS and MERS. So we knew which antigen to use. I mean, so yes, the regulatory process worked quite quickly. Yes, the innovation was quite quick, but we knew what the protective antigen was. And we'd already worked out the stabilizing mutations that I described for MERS. So we're in a very fortunate position. And when people say that to me, I say, well, let's be grateful that was not the human equivalent of African swine fever virus, because, you know, that wouldn't have happened so quickly. So we mustn't forget it's that underpinning knowledge developed over many years allows you to move quite quickly. And, you know, I've been involved with the UK pandemic preparedness, you know, how you're going to achieve that 100-day goal that's put forward. And the idea there coming around is that for each of the virus families, we must understand the generic pieces you need to have to develop a protective vaccine for that family. And then if virus X comes along, which is a member of that family, at least you've got a head start on how to get a protective vaccine for that family. And it's just, you cannot stop that underpinning basic research. I mentioned African swine fever. You know, again, I'm bound to anti-promote what Pirbright does, but not just does, us and Plum Island, you know, maintained research programs on those diseases when they were not fashionable, when they were just in Africa and no one's interested. But if we hadn't done that work, you know, we would, well, it's tough enough as it is, but we wouldn't have been anywhere. So, I think the most important message out of COVID is, let's not stop basic research. It's not, you know, the problem isn't gone. There's going to be another one. And we don't know what it's going to be.
And perhaps before I go to the floor, I turn to David, who's spent many years in the regulatory environment looking at making vaccines available, especially for fast diseases. We sort of mention about the technology platform as sort of facilitating sort of rapid approval, but again, in my experience, when we had the canary pox vectors, then you know, each application was treated as a separate marketing authorization and went through the same timelines and rigorous assessment process. It didn't really, I don't think, facilitated the sort of registration once the first one had gone through. It gives you a little bit of confidence, but you tend to follow. I wonder where we may have gone with the new legislation, actually, David, in terms of platform technology.
Yeah, no, I think that there has been progress in the new legislation, and it'll be this is within Europe, and in fact similar. I think in the US they're already there. And I think Brian pointed that point about changing an RSV strain within a vaccine. You essentially start all over again. And therefore there's a commercial hesitancy to do it. And I think that that's one thing that should be possible and will, I think, be possible if they follow the model. That effectively you can gather your package of quality and safety data for the vector, and then insert whatever your new target is, and then only have to demonstrate efficacy and that it hasn't impacted on the others. So I think there is a lot of potential. I think in terms of the COVID and the overall difference, you know, to me, it all comes down to that benefit risk. And in fact, the regulators on the human side were willing to accept originally very low efficacy because they needed something. And they were therefore actually prepared to tolerate a higher level of risk because the consequences of the epidemic were so risk. I think it's very, very difficult. I find it very difficult to imagine a similar situation in a veterinary context that would allow legislators or create the environment where legislators would be prepared to take that level of risk on the animal side. And you know, you could look at, say, influenza vaccines where the humans for years had shortened ways of changing strains every year. It never happened on the veterinary side because there wasn't. That, you know, people would say we need it, but in fact, there wasn't enough body to change the legislation. So, thank you.
Yeah, I think James, you have a comment.
So, yeah. Thank you. David, I wanted to talk to that animal influenza point because, in fact, a framework was developed through the OIE and WHO for faster licensing of equine influenza vaccines around 15 years ago. And there was an expert surveillance panel that still meets to set up to make recommendations for, you might say this is a minority pathogen, minority disease, but it's interesting, I think, as a model in this respect. And vaccines have been updated since then, but every one, I believe, has gone through a new regulatory process chosen by the company rather than using the platform type approach. I'm happy to be corrected, but certainly for many years that was the case, that the platform approach was not used. So there's both a regulatory angle and also, why are the companies not using this? And I think, the question that for me is really interesting. For me, we've been talking about platform technologies in animal health, to my knowledge, because of that example, for over 20 years now. And David, you've done recent work on that. When is this going to be possible? Because it's too long already.
Yeah, I think certainly within Europe, the platform technology framework is being developed within the states. To some extent, it's already in place. I think that you come to that terrible problem that even with the simplified approach, it still costs a lot of money to generate the necessary studies, and therefore the commercial decision to actually invest in doing those changes. So I think it is still a question of being pragmatic about what the requirements are for a change, to try and minimize those requirements to what is an acceptable level, and there may still be some way to go on that.
Yeah, I would just come back actually on the pre-fusion protein, which stabilizes, improves immunogenicity. If it's the same protein that you've actually just fixed, it's not actually changed to the antigenic content per se. The master seed's the same. So that would just be a variation to your in-processed step.
It's not, Pascal, I know much.
Yeah, so I suppose, but his understanding, the regulatory mechanisms, and I know Erwin mentioned about when the first DNA vaccines came through, and then the FDA have set this standard for doing integration studies. But again, when we think about the issues, then we saw the first DNA vaccine in Europe for Atlantic salmon. It did go through a very rigorous process, but I think we would all expect it. We have a novel vaccine going into a food-producing animal, and the regulators wanted assurance that this vaccine wouldn't integrate, because it has the potential, into the genome of Atlantic salmon. It wouldn't trigger potentially some mutation or trigger some oncogene, and it would be safe then for people consuming that food. So I think that's sort of we can get frustrated about the studies that we have to do to demonstrate the absence of integration. That's difficult, but I think we'd all want those studies to be done. So, but anyway, I think I've probably covered enough on the regulatory side. I will open the floor. Is there any questions from the audience to any of the panel members, please? We've got a microphone at the front.
Yes, thank you. Interesting discussion. It's always has been very annoying for me also as a vet and as a virologist that we don't get these vaccines earlier to the market. And I fully agree with James, the story of the equine influenza vaccines, and that panel still exists. And but I would also like to ask Pascal, because you have developed this very nice canary pox vector platform with an hemagglutinin cassette, which would be a perfect example of a very easy updatable vaccine, but still it causes too many problems apparently, or it's too costly, or why is it not every year updated? Just as an example.
Thanks for the question. I can't answer to that. I'm not privy to that particular example and development. But allow me to make a comment. There is no perfect platform. We have a canarypox platform that we've applied to a number of targets, and every time you plug something new in it, it can be a surprise. So what we're working towards is a portfolio of platforms, and if there's a new emerging agent, hopefully we are able to find out the protective epitope relatively soon, and you plug it in your different vectors, systems, and you hope that one of them will work. For the canarypox platform, I invite you to speak to my colleagues from R&D who are there and can take that.
Yeah, that's fine. It's not about the details. It's more what David also said, that in the human field we know this fast track, and every year we have quite easily, I would say, seasonal updates. And in the veterinary field it simply doesn't happen. But okay.
Yeah, we have two questions.
I mean, there is just one other thing to say, that actually if you look objectively at the requirements for efficacy for changing flu strains, they're much more demanding on the veterinary side than they are on the human side. Where on the human side, they're quite happy just to have serology and indicator groups, and I think we've always said that because you can do efficacy studies in the target animal, you should do efficacy studies, and therefore, you know, the barrier. But just to come back to the canarypox, I think there has been a change in the legislation. So up to now, every new vaccine, it was a new GMO, and effectively, in principle, you start the assessment from scratch. Well, the amendments to the legislation say that won't happen anymore. You will recognize the concept of your construct and the inserts, and that will apply to many of the others that Erwin is working on and so on. The detail of how that will work. So I think it will still make it easier, but it won't mean that it can just be done because one does have to be aware, you know, the effect of the insert into the vector and just confirm it.
But David, to follow up on the example, actually for equine influenza, my understanding is that the platform licensing allows for serological evaluation, not necessarily efficacy studies, but all of the companies who have updated their vaccines have chosen to go down the new vaccine route. I mean, perhaps being driven by marketing issues because of it's nice to show efficacy, but it's the point is it's not being used, and I think this, you know, so it's a regulatory versus commercial aspects that I think it is really important to consider this. But we're all talking and stopping Don asking his question.
Yeah, Don, please.
Thanks, James, and thanks to the panel. Great discussion and great talks. I mean, I've just perhaps a question for Erwin, it may be for others as well, which is particularly thinking about FMD vaccines at the moment globally. The market is very fragmented, and we have lots of producers that are producing products for different settings and different potencies and different strains and everything. Incredibly complicated market. What's the vision for, you know, the future of these new technologies coming in for FMD vaccines, and what's the opportunities for other companies to perhaps access some of that, the intellectual property and the benefits of those approaches? Or I mean, how does it look in the future in terms of if new technologies come available? Will they only be provided by a very small number of companies, or will all companies have access to those technologies?
Yeah. So thank you, Don. So the new there are probably only a few technologies I think that show the greatest promise at the moment, and I feel that we're working on one of them, and I'm happy with it. But so I think that what these new technologies can bring in general, you said the market is big and there are many players and it's complex. We hope with the virus-like particle technology that we can move faster or increase the supply, because it's a technology that doesn't require high containment facilities, for instance, and that would potentially allow us to ramp up in case there's a need for it. I think that those are the benefits. But in terms of sharing benefit or sharing IP, that's a sensitive point indeed, and we as a company do not own all of that IP. I mean, it was done in collaboration with, for instance, Charleston. So they have been the inventors for many of the aspects. So, but yeah, that's an area I'd rather not touch. But at the moment, we are the only ones having access to the technology, and how that will be in the future, I cannot tell. I'm sorry.
Do you want me to add? I mean, you know, well, you do know, Don, that the Gates Foundation have, put a lot behind this project, and they have a global access clause in their grants. So they're not saying who's going to deliver it, but as part of the funding is that we must make this product available to the people that need it, and I think that's. And that's the thing that sells it to them. Of all the benefits, it's the potential to increase availability is the big one. If people can't even get a vaccine, then that's you're not even at the first base. So, yeah, I mean, who knows what that model looked like in the end? But the driver now, certainly for initially, East and West Africa would be to make the product available if it proves to be successful.
I think, Don, you touched upon it in your talk this morning about how we bridge that gap, isn't it, between the development and commercialization. There's an excellent slide looking at that, and it's the same with the vaccines. And I think the thing coming out of that is how do we identify those best optimum candidates at an earliest stage possible? Because we all know that it takes many years normally to actually develop a vaccine. I think the last figures I saw is about 5 to 8 years for a veterinary vaccine, costing 18 million euros in the last report I saw from the Animal Health Europe. So you're talking about a significant cost to bridge that gap. So what we need to do is get better at sort of identifying the best candidates at the very earliest stage possible. And there's one question down here, and I think we were then.
Thank you so much to all our panelists. I had a question. I think it goes to Hudelet or Godlet, you pardon me.
Pascal.
And probably Van der Bon. I know you did highlight some of the blockages to innovation during the research and development process. And I'm asking about, there seem to be some blockages also at the level of market authorization that are also quite frustrating. And I had been thinking maybe the problem was with us in Africa. I served on the National Drug Authority, the regulatory drug authority for Uganda for 5 years before I joined EuFMD, and I was chairing the Veterinary Medicines Committee. And market authorization was really taking long. The average was the lowest is about 2 years, and people would get frustrated. So sometimes people would make contact and say, what is it? And we also would put our staff and say, why can't you deliver? They said, give us retreats. At least every month we need to be in a hotel for about a week, only doing, reviewing the application so that we can clear the backlog. Even after doing that over the 5 years, we never got rid of the backlog. The delays remained. So we requested to do some benchmarking and visited some of the regulator agencies we thought were among the best we could think about. And I led a team to visit the VMD in UK. And one of the first questions I asked is, how long does it take you to complete the market authorization process? And they told us average is about 8 months and more. So I was surprised. I realized that we're almost at the same level. So what could be the issues in your experience? What are the blockages? You can imagine somebody has taken so long through the R&D, the scenarios you gave us. You follow up one potential partner country for 4 years. You get frustrated, you abandon, you start another one for 2 years, and there's no progress. Now you imagine after you've gone through all that and you have a product ready to put on the market, you start another process of waiting, and it takes 2 years. What can be that make it a bit faster? Or what are the issues, really? I don't understand. Maybe the pharmacists or the people in the pharmaceutical industry a better place to tell us why it must take that long to complete the cycle for market authorization. Thank you so much.
First.
I'm afraid this is more for you, man.
Yeah, no, I mean, it's an interesting question, but actually, let's just look back at the COVID. Everyone was putting the pressure on the regulators to say, you know, why are you waiting? But the answer is it does take time. I think one can look at the reasons for the length of time, because it's something that agencies do all the time, and it depends on a number of different things, like the quality of the dossier, and for COVID, you know, they did this parallel review as the data was generated, it was reviewed, whereas generally that doesn't happen. So I think in terms of how can it be accelerated, actually a lot of it is down to communication, and if there's good communication and almost training by the regulators to what it is they expect to see, you can get through some of those dossiers very, very quickly. And again, you know, it's regulatory strategy. Some companies will generate large amounts of data so that when they put it in, it goes through very quickly. Others will generate very minimal data packages. And then if there is a delay, whilst more data is generated, that leads to a long, long debate. So it's a good question, but there's no simple answer. But I think the answer is good communication between the two so that the applicant understands.
What we've seen as manufacturers is the level of priority that is put on the dossier makes a big difference. 19 years ago, we developed a vaccine for blue tongue for Europe because it was an emerging disease. I have to say that the turnout of the regulatory agencies was really fast. It was a priority dossier. I guess they put the right level of resources, and they engaged in negotiation with the manufacturer on what the expectations were from the start. And we saw a big difference. And I guess for your specific cases, I can also think of a couple of things. One is alignment of expectations of the national regulatory agency on international standards. If you start asking for studies or data that only your country asks for, then it's going to be a problem, because we build standard data packages that we can use in several geographies. And the other one is mutualization of efforts with other countries of the region. That really adds efficiency as well. Just take the model of Europe, one review for 27 countries. This is important.
Not every part of Europe anymore.
Sorry, 26.
26.
I'd like to comment on the priority, and there's been, again, a lot of initiatives to try and make more vaccines available. In Europe, we have the MUM scheme for minor use and minor diseases in major species. But, you know, and you can sort of, you know, try and address some of the issues surrounding the timelines and the data requirements. But ultimately, it comes at business cost. I mean, David, again, might want to comment, but you can put all those things in place, but it's not a commercial proposition for a pharmaceutical company. They won't manufacture the batches. And I think that's one thing. You know, the veterinary sector is, you know, driven by the private industry, and it has to be a commercial operation for them. I mean, Pascal, I don't know if you want to just address that before we turn to our virtual questions.
Sorry, Martin, I missed that.
Just about we can put in sort of you know, reduced data requirements for minor, you know, use of minor disease in major species and things, but ultimately it's a commercial proposition, isn't it, to develop a new vaccine, you know, for a animal species.
Yeah, that's how it starts. It starts with a business case, and have to demonstrate return on investment. Yeah. That's the reality.
Yes. Yeah, thanks very much, Martin. We've got two questions from our online audience. I'll read out. This is for Erwin. Regarding new technologies using animal vaccines with less restriction in comparison to human vaccines, how we can be sure that the technologies are safe so the negative impact to animals will not get into the human food chain? If there are adverse impacts from animal to human, how long will you be able to notice them?
Oh, that's it's quite a difficult question, I think, to answer. looking at safety for the animals, of course, we are in the privileged position that we can test safety in the target animal right away on all different aspects. I think in human health, it requires first animal trials, and then you go to the clinical trials. So for the animals, I think, especially in animal health, we can be faster and better in determining the safety of a vaccine or a new technology in animals. Looking at new technologies being used in food animals and then being eaten by those animals being eaten by humans. I actually don't know if we have any regulatory framework for that. Of course, we have to make sure that certain substances in the adjuvant are not present that could harm humans, but whether a certain mRNA or something could have a negative effect in the long term by eating it, I actually don't know.
Yeah, no, and I think Caroline Guitret, who's in the audience, mentioned today, those factors on the human safety of vaccinated animals are always considered. Rarely, but occasionally, they relate to the active substance, and particularly with zoonotic potential. But in terms of these novel technologies, and Martin mentioned it with the DNA vaccine, I think that's often the question. And because how do you then, what is an acceptable level of risk? And I think one has to distinguish very clearly between the human benefit risk, where someone is taking a product and a conscious choice to take that product, and therefore they're prepared to tolerate a degree of risk because they will get a benefit, as opposed to consuming vaccinated animals, where they have no choice as to whether they are exposing themselves to that risk. And that has to be taken into account in the overall benefit risk. And I think that's why regulators on the veterinary side are extremely conservative, and that's whether that applies to pharmaceutical products and setting acceptable residue limits, and also for things like RNA, DNA transmission, because your consumer's not going to know, they're not going to have any choice in that decision. So you only then have the risk side, you don't have any potential benefit.
I'll just quickly add on that, actually, an example that I've come across and practically is that the BCG vaccine for cattle. There's an ambition to get a vaccine against tuberculosis for cattle, and the tentative vaccine would be the human BCG, which has obviously been proved successful for over 100 years, and obviously proved its safety with its billions of doses that have been administered to children and adults all over the world. When it comes to cattle, once you start putting into cattle, the questions are, what does it do? Does it replicate? It's going from bovine back into bovine. What titer does it reach? And if those titers are high and it gets into meat, what about the risks to humans, especially those who are immunocompromised? And there's now, we're aware about now, up to 20% of people are immunocompromised. So it can really sort of put a lot of questions. And as David said, those questions are really hard to answer, because it's, you're on the leading edge. You're putting, what is this conventional vaccine into a new species, and then wondering what impact it might have on people consuming meat from those vaccinated animals.
But Martin, can I ask a follow-up to David around the DNA question? Because I think that sometimes we think that we live in a unique world. You don't, all right, but I think that we can sometimes kid ourselves. And the fact is that globally we've been eating genetically modified tomatoes and soya, as have animals, particularly in the latter, for many years. And actually, it's so widespread, we pretty much have no choice. Why does GM beef or pork or chicken carry any greater risk than tomatoes and soya, which have gone through that regulatory pathway very easily? And I think that sometimes we look to fit a new technology into an existing framework instead of looking more of a blank sheet of paper. And I think that if we're going to actually see the benefits coming from new technology, we need to think from first principles more rather than just thinking about the regulatory frameworks that we've got. But maybe I've got that wrong. I don't know. I'm sure that that GM tomatoes did not come through VMD.
No, but interestingly enough, you could look at GM potatoes and the fact that those GM potatoes then contained antibiotic resistance genes, and then you had to look at very carefully at the risk that could actually affect
but that was
the
mad.
level of antibiotic resistance.
That was mad to do that,
So,
arguably.
Yes,
marker,
okay. So I agree that one can't kind of preempt every risk, but I think when it's a new technology, it's behoven to have a look at, see what is the risk. And I think often, say, in a situation like that with integration, you're then looking at germline, particularly, say, germline transmission, and are you then affecting the wild population and so on. So I think if you look at the legislation, the principles are very, very sound in terms of spread and shared transmission to target and non-target species. And then it is really, James, I think it's a question, how is that interpreted? And some people over-interpret it and will look for zero risk, which is never possible. Yeah. So it's really an
and then we get zero
risk.
Gains, and actually we don't benefit, the global population does not benefit from the new technology that it should be. And I think, the vaccines, I think, could be another example of that if we're not careful.
Yep.
Sorry, we're at time with one more question. But
thank you so much.
Yeah.
I think my colleague is telling me that we are running out of time, so we need to break for coffee. I would like to thank you all the panelists, and maybe you join us to thank them all. And there was one remaining question for Pascal. Maybe you can answer it online. Please,
yes, if we could be back here, I think at about 16:25.
Pascal's.
Yeah, just a reminder, there's the posters at the back of the room. You can access them through the app, and you can vote on the posters as well through the app. So I encourage you all to do that while you're having coffee, and we'll see you back at 16:25. Thank you.
Some history that it might have been FMD.
And I think we've done a brilliant example of that this week.
What you need to know about foot and mouth disease. Foot and mouth disease is a very contagious viral disease which affects cows, sheep, goats, pigs, and other animals such as wild boar. The disease causes serious production losses, even if not all animals die from the virus. An outbreak in a country can result in a major block to international trade. Humans cannot become ill from the disease, but they can play an important role in spreading it to animals through dirty clothes, vehicles, and other equipment. The signs of FMD are variable and depend on the animals. In cattle, the disease can be severe, and you may notice depression, high fever, not eating or difficulty eating, decrease in milk production, increased salivation, blisters followed by lesions on the muzzle, inside the mouth, on the feet and teats. Occasionally, it can cause sudden death in young calves. In pigs, the disease can be severe, and you may notice an inability to stand, with some animals in a sitting position, lesions on the feet, sudden death in piglets. In sheep and goats, the disease is not that evident, and you may notice small blisters just above the feet. Sudden death in young sheep and goats. FMD can be found in all fluids from infected animals. The virus can spread very easily through any of the following ways. The most common is through direct contact between an infected animal and a healthy one. Contaminated materials such as hay, feed, water, milk, urine, and others. Dirty clothing, footwear, or equipment. Contaminated pens, buildings, or contaminated animal transport vehicles. Meat or other products which come from diseased animals, if fed to animals when raw or improperly cooked. And in certain weather conditions, it can spread through the wind from a nearby farm. Contact your veterinarian or local authority immediately if you suspect your herd is infected with FMD. Do not move any animals from the farm and limit any movement of people to and from the farm. A veterinarian will come and look at the animals and take samples to send to the laboratory. The laboratory will examine the samples and confirm the presence or absence of the virus. Once FMD has been confirmed, measures will be put in place to stop the disease spreading to other farms. Farmers whose farms or animals have been in contact with the farm where the disease is present will also be contacted by the local authority or veterinarian. Everyone has a role to play in ensuring that your community and country remain free from the virus. By staying alert and reporting suspected cases of FMD, you can make a difference. Thank you for your attention.
some history that it might have been FMD.
We've done a brilliant example of that this week.
What you need to know about foot and mouth disease. Foot and mouth disease is a very contagious viral disease which affects cows, sheep, goats, pigs, and other animals such as wild boar. The disease causes serious production losses, even if not all animals die from the virus. An outbreak in a country can result in a major block to international trade. Humans cannot become ill from the disease, but they can play an important role in spreading it to animals through dirty clothes, vehicles, and other equipment. The signs of FMD are variable and depend on the animals. In cattle, the disease can be severe, and you may notice depression. high fever, not eating or difficulty eating, decrease in milk production, increased salivation, blisters followed by lesions on the muzzle, inside the mouth, on the feet and teats. Occasionally, it can cause sudden death in young calves. In pigs, the disease can be severe, and you may notice an inability to stand, with some animals in a sitting position, lesions on the feet, sudden death in piglets. In sheep and goats, the disease is not that evident, and you may notice small blisters just above the feet. Sudden death in young sheep and goats. FMD can be found in all fluids from infected animals. The virus can spread very easily through any of the following ways. The most common is through direct contact between an infected animal and a healthy one. Contaminated materials such as hay, feed, water, milk, urine, and others. Dirty clothing, footwear, or equipment. Contaminated pens, buildings, or contaminated animal transport vehicles. Meat or other products which come from diseased animals, if fed to animals when raw or improperly cooked. And in certain weather conditions, it can spread through the wind from a nearby farm. Contact your veterinarian or local authority immediately if you suspect your herd is infected with FMD. Do not move any animals from the farm and limit any movement of people to and from the farm. A veterinarian will come and look at the animals and take samples to send to the laboratory. The laboratory will examine the samples and confirm the presence or absence of the virus. Once FMD has been confirmed, measures will be put in place to stop the disease spreading to other farms. Farmers whose farms or animals have been in contact with the farm where the disease is present will also be contacted by the local authority or veterinarian. Everyone has a role to play in ensuring that your community and country remain free from the virus. By staying alert and reporting suspected cases of FMD, you can make a difference. Thank you for your attention.
history that it might have been FMD.
And I think we've done a brilliant example of that this week.
The first of 5 talks. So welcome to the second part of the Innovation in Vaccinology session at EuFMD open session this afternoon. I really enjoyed the first set of talks and the panel discussion, and I'm very optimistic that the second part will be even better and more interesting. But our first speaker in this session is going to be Alf Dekker from Wageningen, and he's going to be talking about replacement of FMD vaccine potency tests. Over to you.
Yeah, thank you. I was asked to give a presentation on replacements of vaccine potency tests. And in fact, vaccine potency tests were used in the past for registration of the vaccine, but also for batch release. But if I look at a lot of producers worldwide, I think most of the producers have replaced the batch release by using vaccination in cattle and looking at the VNT titers, or just release the batches on 146S content. So first I would like to give you some background on potency tests. Then I will give you some information on what is the best way to, or the best, or what are both acceptable ways of replacing or doing the batch release. And the main message is that small variation in determination of 146S will have very small differences in estimated protection level. So 146S determination is probably the best method. And then the nice thing is that you can also use that on formulated vaccines. So even consumers can use these kind of tests. Now, one of the things that was studied several years ago is the inherent vaccination in potency tests. So in Belgium, they tested 10 times the same vaccine, and what they found, I calculated it a little bit different than in the publication, but that the outcome was between 4.5 or 4.6 to 24 PD50 per dose, with a mean of approximately 15 PD50 per dose. And if you would take the whole experiment as one experiment, then you would find a confidence interval from 8 to 25. So also the observations were more or less within that confidence interval. Then you can ask, what kind of variation is this? Is this because you dilute it differently, inject it differently? If you do just a simple simulation, on the right side you see 10 simulations I did during the presentation of this potency test just in Excel. Then you can easily, based on the outcome of the experiment, do a Monte Carlo simulation. You already see in 10 simulations you have a potency between 6 and 18 PD50. And if you do 200 simulations, it's 2 to 24. So most of the variation is just stochastic variation. It's not really, and that's not a lot you can do about that. Now then, you can do a different test. There are people say that a PGP test is better, but in a PGP test you have to have at least 16 out of 16 animals protected to have 95% confidence that 75% are protected. So yeah, normally we say 12 out of 16 is sufficient, but if you do it only once, then you could say, no, perhaps we should put the level a little bit higher, but the same holds true for a potency test. The advantage of a potency test is that you can also measure that your vaccine is much better than what the official requirement is. Now, there is not a lot of, there is a lot of studies that looked at antibody response and protection, but there is one study in which they not only looked at relation between antibody response and protection, but also did a potency test according to the British Pharmacopoeia, where they also where they used variable amounts of antigen. So the same amount of adjuvant, variable amount of antigen. Now, if you then look at the amount of antigen and the relation with VNT titers, then if you increase the amount of antigen 10 times, you have only 2.5 times higher antibody titers. So that already shows that variation in antigen amount does not have a huge influence on the immunological response. That's an advantage. So if you have enough antigen, if your measurement was not correct, then it will not have a huge influence. Of course, if you have a poor antibody response, it's difficult to get a good antibody response. You really have to increase your antigen a lot. Now, if you do logistic regression, antibody predicts protection best. For type O, they needed higher antibody titers. But if you look at the steep part of the slope, and you look between 25% and 75% protection, which is a huge difference, then only 0.5 log higher antibody titers were found at 75% protection, and and 25% percent protection. And it means that you only have a small window in that range to see if it's a good or a poor vaccine. And that you easily could have a result which is not that predicts a poor or poor protection, but in fact it was a good vaccine. If you do the same for 146S, then of course still the relation with antibody titers I mentioned another time, but then 146S is also well correlated with protection, not as good as antibody responses. For type O, again, you need a higher antigen amount. But then, if you look from 25% protection to 75% protection, that's 180 times more antigen. So that means that if you produce a good vaccine and you have a little bit less antigen in your vaccine, then that will not have a lot of influence on your vaccine. So that's the main take-home message. In fact, quantifying antigen amounts is probably a better way to do batch release than looking at antibody responses. And especially when we know that if you look at between laboratory testing for VNT titers, we easily find 1 log difference, and so a half a log difference would be a huge difference and could have huge consequences in the prediction of protection. So, and the nice thing is, as I said before, 12S and 146S can also be determined in formulated vaccines. So even if a vaccine has been on the shelf, transported to somewhere else, then this can be measured. If you look at what has been done on in vitro quantification of formulated vaccine, you have to extract the vaccine from the antigen from the vaccine. You have to test the antigen. That could be done in a sucrose gradient analysis, but using ELISAs is much easier. There are specific 12S specific ELISAs, the 146S specific ELISA, or you could also use size exclusion chromatography. So the potency test is still necessary for registration. You have to show that you're for registration that your vaccine in a certain formulation is potent. But the GMP production system ensures that you have reproducible vaccines, and the formulation can then be done based on antigen dose. And the later QC can also be done on formulated vaccines. So it clearly shows that doing potency tests for batch releases, as we did in the past, every batch in the Netherlands that was used in our vaccination campaign was always tested in a potency test. That's something we should not do in the future. And that was the last question, slide.
Alf, thank you very much indeed. Fascinating and really important talk in terms of ensuring really good product available on the street. We don't have time for any questions now, so we'll carry them. Any questions that you have, keep them, please. And we're going to move straight to the second speaker, Karel de Luca, who's come here talking from the other part of Boehringer Ingelheim that we've not heard so much from today yet. Who's going to be talking about new insight into the humoral immune response following FMD vaccination in pigs. Thank you.
Thank you. And thank you for selecting this presentation for a talk today. I'm really pleased to present this work. I entered the world of FMDV, I would say, 3 years ago. And one of the colleagues told me, well, you should never enter it, you will never quit it. And I'm really happy to be here today, my first FMDV meeting. And we will I will resume in a few slides one of the part of our work, so working on the insights of the humoral response after vaccination with FMDV vaccines. I'm leading the laboratory of immunology, and as such, we are working on different vaccine candidates for pigs, for dogs, and others. So here is part of the work that was done these last years with the team. So I will present to you what we call internally the immune fingerprint. It's a concept that we already developed for rabies. Clearly, it's when we want to develop new ways of understanding the immune response, where we lack correlative protection, can we look at different immune parameters and make I would say, multi-parametric analysis to see if we can correlate the protection with not one single parameter that we don't have with multiple immune parameters. Because you know that sometimes immunologists are called like storytelling because we can describe a lot, but we don't correlate always to the protection. The idea is really to come with parameters that can correlate to protection. So for rabies, it was a kind of a model because we knew that we could correlate to what we had as a VNT protection. We knew that the seroneutralization is highly correlated to protection for rabies. I would say that FMDV is a kind of in-between. We know that the VNT, as it was said, is highly correlated to protection. However, in swine, there is what is called the gray zone, where the protection is, the relation between protection and VNT titers is lower. And we need to have tools to identify protection without going systematically to challenge studies for ethical reasons, for cost, for time. And we need that because we are developing new vaccines at BI, and we need to screen the antigens, the technologies, the adjuvant. And also we want markers for, to work on the duration of immunity of our vaccines too. So the way that we did it, we vaccinated pigs, and we looked at different immune parameters. So here our model is in the 01 Manisa antigen strain. We looked at different titers of IgM and IgG, the VNT, of course, the seroblocking ELISA, which is a liquid phase blocking ELISA that was developed internally, the glycosylation of antibodies, and this one will be part of this talk today. We looked also at the avidity and some of the cellular response, mainly the TFH. So to look at these immune parameters, we immunized, as I said, pigs from 10 to 12 weeks old. And we modulate the immune response by playing with the adjuvants. So we use an internal adjuvant, which is the TS6 oil-in-water formulation, oil. And we added different immunostimulants that are typical agonists of PRR, of pathogen recognition receptors, that are well described in the literature, and some of them you know them. We've got, for example, the poly-IC, which is binding to the toll-like receptor 3. We've got the CpG, CL098, which is binding to the TLR8, and the beta-glucan from Hudelet gracilis, which bind to dectin-1. So, first guest, our VIP guest, which is the neutralizing antibodies. Everyone relies highly on the VNT titers for FMDV. And though we know that it's highly correlated in cattle, as I said, there is this question in pigs for some of this gray zone. So now I will present to you the VNT titers with the different adjuvants to see if we were able to modulate the immune response with the different adjuvants that we selected. So the first, I'm sorry because I don't have any pointer. So the first in gray, you've got our all-in-water adjuvant, our own proprietary adjuvant. And you can see by day 28, we had VNT titers of above around 2 log 10. When we add the CpG in green, when we add also the CL097, we don't see a huge difference, so that was quite homologous. The beta-glucan was really worse than with the TS6 alone. And when we used the poly-IC, it increased dramatically, significantly, the VN titers by up to 3 logs. The 01 Manisa was also formulated with ISA206, which is also a benbark from CEPIC, another adjuvant, and we could see that it was quite comparable to the TS6 alone. So from that, we could say, okay, we've got tools that can modulate the immune response to assess the different immune parameters and to see if they've got some interest to look at the protection. So from this, we went to other analyses, and we kept the TS6 alone. We kept the beta-glucan as a kind of a negative or worse formulation, the poly-IC, and the ISA206. But as I said, we've got the special guest. Our special guests are the non-neutralizing antibodies. So most of the time, we look at the VNT titers, but we forget about the antibody-dependent cell cytotoxicity that are mediated by effector functions of the antibodies. So once a cell that is infected expresses the antigen at the surface, it is recognized by specific antibodies. Then these specific antibodies are recognized themselves by Fc gamma receptor that recognize the Fc part, the constant region of the antibodies. And then NK cells, for example, can kill the infected cells. We've got also the phagocytosis of infected cells. But what we know is that first, these mechanisms are also important at the early time after vaccination because they support the protection where the antibody titers are still low. And what also has been described these last years in humans, and mainly on monoclonal antibodies, therapeutic antibodies, is that the glycosylation patterns on the antibodies, on the constant regions of the antibodies, is also highly important to mediate the cellular functions of these antibodies, because it will increase the binding capacities to the Fc gamma receptor on the cells, and it will also increase the affinity, and then the mediation of activities of NK cells, for example. So now we will look a little deeper on this antibody glycosylation. So what is glycosylation? It's just different glycans, a composition of glycans. Several have been described so far. So we've got, at the beginning of the chain, you've got here galactose, fucose, the mannose in green, and then you will have the sialic acid at the end of the glycan chain. Whereas the sialic acid presence has a negative impact on what we just described before, the ADCC, we know that the high mannose glycan presence or that the absence of fucose are good for ADCC, so for cellular mediated functions, and so for the clearance also of the virus. So now we'll show you on these three parameters what we observed with our different adjuvants. So I've got a little time left, yes. So in blue, in dark blue, it's just a non-specific antibody. So you just take your serum, you look at the profile of glycosylation, and we compare that to human, because as I said, it's comparative immunology. There are no data in pigs for now. And what we could see is that we are almost 10% of the antibodies that are highly cellulated, and it's comparable to what is observed in humans with 10% of antibodies that are cellulated at steady state. Then what we did is that we took the serum, we purified the specific antibodies against FMD, and then they were analyzed by mass spec. And the profiles of glycosylation were analyzed. And what we can see is that they are reduced, the sialic acid, and as I said, sialic acid reduction is more in favor of ADCC functions. And we can see that these functions are different. This presence is really reduced with the TS6 and poly-IC, more than when the TS6 was alone or compared to beta-glucan or ISA206. So going with more a cellular function of these antibodies. We then looked at the mannose content, that you can see that we've got more mannose on the glycosylation chains of these antibodies. Again, when we compared at day 0 the profile of antibodies, we can see that they are highly mannosilated, which is really different to what is observed in humans. In humans, we've got less mannosilation. And when then you look after vaccination, 28 days, you can see that the poly-IC increases really dramatically the presence of mannose more than the other groups. So here again, you've got a signal that it could be a favor of ADCC. And the fucose, so we look at the non-fucosylation to correlate it to ADCC. And again, we can see that with the most effects that was seen with the poly-IC compared to the other adjuvants. So that could be also in favor of more potent ADCC. So our data are the first investigations on glycosylation in pigs after vaccination and adjuvantations. We can show that we've got signals that in humans would be in favor of some kind of inflammatory response and more prone to antibody functions with ADCC. And so our next step would be for sure to analyze that in the context of infection and then protection to see if we can have a correlation. I thank you for your attention.
Thank you very much indeed, Karel. So we are a little bit behind on time, so I think we'll carry forward for questions. But I've got one or two that I'm fascinated to ask you about that really interesting study. But I'm sure there'll be others from the floor. So the third in our set of 5 speakers in the second session is Claire Richaud. Claire works for MSD Animal Health and is going to be talking in a slightly different topic for this session, talking about ag technologies supporting governmental disease eradication programs.
Yeah, thank you very much. So indeed, I come from a very different side. I come from the data and the digital side of our company. So everything you guys discussed before, I didn't get anything. So I'm very happy to see that the two worlds are trying to collapse and try to create something together, because I think that's really why my colleagues asked me to be there. So today, indeed, I'm going to discuss how technologies can support the control of diseases, because apparently you cannot fully eradicate them. Yep. Okay, so little story. Let's say that we are in the nice city of Marseille and there is a ship arriving full of sheep, and one of the animals is infected with a disease. You're going to have a whole range of questions arriving. Mainly, where is the disease? Which animal is infected? Which other animals were with this animal? Where did the animal come from? When was it vaccinated, if it was, etc., etc. So the two main components that we are going to address today are related to two questions: the when and the where. So basically, we do believe that technologies can help supporting answers to those questions, when and where. We believe that the new technologies, digital, can help with identification, but also with providing more data related to the health and the welfare status of the animal, and also to tracking the shipping and the movement of the animals as well. So I'm going to discuss today two sets of technologies. I'm mainly going to discuss e-identification, so electronic identification, to answer the question of the where is the animal. So very quickly, what is e-identification? Is if in most of the countries we do have visual identification or boluses, implants, etc., we focus on electronic identification, which are microchips embedded into a little tag. The tag is going to receive a signal when it's being read, and you can get information on the official identification number of the animal, but a whole other set of data depending on what you decide to put on the chip. So that's very interesting because there is way less risk of having corroded data. There are less mistakes, mainly human mistakes, because, well, you know, human makes mistakes instead of machines. Asimov does not agree with that, but that's another discussion. And you can also have something very interesting, which is the real-time animal localization. So you answer the where is the animal. Also improved traceability via data flows. I will come to that a bit later. Second set of technologies are the 24/7 monitoring systems. So you do have softwares. I saw quite a lot of people with Apple Watches or little Fitbit, so congrats for you. You share a very interesting characteristic with our cows. They also have the same technology. So it's the same thing. They will have sensors which are going to encapsulate data from rumination, breathing, the steps they take, their movements, their behavior, and these data are going to be collected by the sensors, and the sensors are then sending this data to algorithm, sending back a message to a software or a smartphone, for instance, telling to the farmer or another third-party provider, you need to act now in real time because your animal has an issue. It's not replacing the farmer. I really want to say that. You still really need to have the human interaction with the animal to make sure that you take the right answer to the situation. So very useful for early diseases recognition. You can see symptoms up to 48 hours. You can see that there is an issue 48 hours even before having visible symptoms occurring. So you do answer as well the when, because it allows you to take the good action at the good moment so you can close the time gap. So why is it useful for authorities? It's useful for the farmers and for the entire food chain, of course, but for authorities as well, because we start with identification. That's really a starting point for a good traceability system. At the same time, it will help you to have the right data faster going into your database, because even if you have a great database, some people can discuss that, but we do have traces and BOVEX in the EU, for instance, but if you don't have the right data entering the database, feeding it, well, it's quite useless, if I may say. At the same time, it's also very useful for time optimization. You can prevent and you can act at a local authority, a regional one, really, really fast. Also good for your vaccination and antimicrobial reduction. Yeah, I'm French, sorry. So AMR. So it's very useful for your programs, and on top of that, very, very useful for the farming system in general. For profitability, sustainability, I didn't hear a lot of that, but that's quite useful for this as well. And to conclude, is this technology once, okay? So to conclude and to sum up, what could be very useful for having these technologies being extremely beneficial for the authorities, the first thing is to recognize them. So those technologies, they do exist. Those technologies are available. You don't need to reinvent the wheel. You just need to make sure that they are being incorporated into the preventative toolbox, that they are being integrated as well in international policies. Second step, once they are recognized, it's very important as well to make sure that they are well adopted. So for that, the money is at the center of everything, of course. So helping farmers, helping third-party providers to get those technologies, but more importantly, since the benefits are falling on the entire food chain, making sure that the burden of the cost is not only falling on the farmer. Very important point. And the third point, which is, I think, one of the most important, once you have those technologies, it's very important that they can speak with each other. So we need to create a connected system, a connected framework, which is encouraging the live tracking of the animals, which means starting point, e-identification for everyone is quite cheap. It's like €1 per animal. Second point, standardization. So you want the technologies to speak with each other, to be interoperable as well. So for that, you need standards, standardized data. We have great certification bodies able to help on that. At the same time, we also need to make sure that people know how to use those technologies. We need to make sure as well that people know how to invest as well on those technologies. So for that, we need to train them. For that, we need to attract talents as well to steer innovation. So we need to attract engineers, IT developers as well. In a mature market like the agriculture one, it's not an easy topic at all. So we would be very much in favor of making sure that we can attract talents and steer innovation in this sector. And to conclude, and I'm right on time, 20 seconds, with a connected global system where you can live track the animals, you can, in that case, secure localization, so answer the where, and at the same time, with early detection, thanks to closing the time gap, you can answer the when, and I hope in the future it will help to advance towards the control of diseases. Thank you very much.
Speaking as someone who's spent the last 15 years trying to put tags on bats in Ghana, and other parts of Africa. I was very pleased that Claire finished her talk talking about some of the opportunities for doing it in other target species as well. The fourth in our set of speakers is David Mackay. And I'm not really sure I'm going to introduce David because he's spent most of the... he's been here a lot longer than most of us and was on the previous panel. But David's going to be talking about pre-qualification of vaccines.
Thank you very much, James. Yep. So I'm going to be speaking on behalf of the pre-qualification team at the EuFMD, and that's myself, Martin, who's on the panel here, Jacqueline Horsington, who's in the audience, and Habib Oyedele, who works remotely. Next slide, please. So the first question is, what is pre-qualification and why is it something that EuFMD is interested in? And really it's all about promoting the use of high-quality vaccines. There's quite a lot of text on some of my slides, so please, I wouldn't try and read it all. I'm just going to try and pick up the main issues that I wanted to get across about pre-qualifications today. And so the essential challenge that we've come, and it hasn't really come across today, but it is a fact, is that most vaccines rarely have full marketing authorizations in the country in which they're going to be used. And that is, we've talked about the importance earlier on of regulation, and the small size and unpredictable nature of the market often makes it very unattractive for companies to authorize, fully market authorize their products in countries. So when competent authorities want to get hold of those vaccines, they're very rarely looking at vaccines that are authorized in their own country, so they have to rely on information from other countries, and it can be very difficult to validate the quality of that information and to know whether or not the product that's being used meets international standards. And so as one of the prime drivers behind EuFMD is to promote vaccine security, the use of high-quality vaccines is an important element of that. Next slide, please. So what do we want to do? And this is really, you know, we've set out the vision. It's to improve vaccine security for fast diseases by promoting the use of vaccines that comply with minimum international standards. And how are we going to go about doing that? We want to establish this system to independently review and make public information on vaccines against fast diseases and the extent to which there is evidence, and evidence is the key word, to support compliance with international standards. And we aim to start with foot and mouth disease, get a system working, and then one can consider applying the same approach to other vaccines. Next slide, please. So really this is just the kind of beneficial cycle that at the moment it's very difficult to get information about the level of compliance of vaccines. Once you start making that information available, more people start using quality vaccines, the market for them is improved, and therefore you should be in a virtuous cycle, and that's where we aim to promote in terms of pre-qualification. Next slide, please. So the benefits, just quickly, you know, the aim is to make this independent, verifiable information available publicly to potential users. That can inform decisions on procurement. It means that it should, one of the challenges is that because these vaccines aren't widely used, it can be difficult to really evaluate their regulatory compliance. And through PQ, we will provide access to highly experienced people who are experienced in assessing this type of vaccine. And that would be useful both to authorities in developed countries and also in less developed countries who may not have fully developed regulatory systems. And something that we'll be talking about tomorrow, this ASOPs, which if people aren't familiar means assured emergency supply options, and that's really looking at alternative ways than conventional ad hoc procurements or vaccine banks of assuring the supply of emergency vaccines when you want them. Well, if you want to come into any long-term agreement, you want to be sure about the quality of the vaccine that you're going to be procuring, because it could be several years before you actually do. And pre-qualification is seen as one way of achieving that. The other thing that we've talked a lot about in the sessions today is the need for independent quality control, and we see pre-qualification as acting as an incentive to the exchange of materials to allow independent pre-qualification or independent quality control, because if you talk to any of the reference laboratories, they will tell you one of the main problems is getting hold of the reagents to actually be able to do that control. Next slide, please. So pre-qualification is a general term. You may well know that WHO operates a pre-qualification scheme. We take the same principles, but have to very much veteranize that approach. And it's all about compliance with OIE standards, or WOA standards, as they're now called. And it works on this principle that we call qualification assurance. So we're not a regulatory agency at EuFMD. We don't attempt to do the work that a regulatory agency does. We take into account the fact that a product already has a national marketing authorization, and then seek information from the manufacturer as to how compliance has been demonstrated as part of issuing that authorization. Next slide, please. So I haven't got time to go through, but we have this governance structure. We have a wide number and diversity of stakeholders, because the trouble is we're in occupied ground here. You've got national authorities for medicines, national authorities for disease control. You've got all of the international organizations, all of whom are associated with various aspects of quality control. So how do you then put in place this system that adds that information, which I think most people who are involved in the area will agree there is a gap. Next slide, please. So the answer is really we're going to adopt a phased approach. At the moment, we're putting in place stage 1, which is basically a proof of concept. This has never been done for veterinary vaccines before. So we want to prove that it is possible and to generate useful information on the basis of a voluntary scheme with manufacturers supplying information on the quality of their vaccines, which is then examined and the information made public. Clearly, then, the next step is to link that pre-qualification into procurement decisions. And again, this has to be a voluntary procedure. But I think if we can show through stage 1 that this is a valuable exercise to people wanting to seek purchase vaccines, then there will be a willingness to link it into procurement decisions. And we don't want this to be a project which has a life and dies. We want pre-qualification to be a sustainable system. It should be possible to do that through cost recovery from those who use the system and from manufacturers that benefit from the system. And that's really the final stage, to turn this from a project into a sustainable system. I see I've run out of time. I'm just going to say a couple of quick words. Next slide, please. Just to say, because we are not duplicating what it is that is done by a licensing authority, we can't. You know, we talked, it takes 2 years to approve some of these vaccines. Many months of man-months of work. We're going to adopt a risk-based approach. We've identified what is the key characteristics and properties of a vaccine which are needed to be known by those wishing to procurement, and then we will look at compliance of those particular aspects. So, and rely for the overall quality that this is a product which has been approved by a national regulatory authority. Next slide, please. So, you know, you could say, what are those characteristics? Well, they'll be very familiar here. Inactivation, characterization of the virus seed, 146S content, batch potency, purity, and consistency. They're listed there. Next slide, please. Obviously, we will need to be sure that the product is safe and that it's efficacious and that the claims made for the product in the product literature are substantiated by studies that have been conducted in line with minimum international standards. Over and above what can be done at national authorizations, because these are products that have been out there, we'd be looking at information on the amount of product that had gone out, had there been product recalls, had there been regulatory actions that have been taken. And that all helps give assurance about the quality of the product which has already been on the market. Next slide, please. So where are we at the moment? So we have developed this approach which I'm presenting to you today. Martin and the team have done a lot of work on developing the technical and administrative guidelines which have been out for public consultation and we hope will shortly be adopted by the Standing Committee and published on the EuFMD website, because the whole thing relies on transparency. These are transparent standards and requirements that will be out there. It's all been done with a lot of consultation with stakeholders. We have letters of agreement with organizations that can provide suitable expertise, and we're putting in place all of the procedures and IT systems. So all with the objective that we will open the scheme up for expressions of interest to submit applications as from January next year. And just to say, we've already had companies saying we would be very interested in submitting our product, so that's a really good start. Next slide, please. In fact, I think it's just one more. So just to say, you know, the system is to assure the quality, this principle of qualification assurance, and that we're adopting a phased approach. Sorry, I ran over, James. Thank you.
David, that's fine. It's a really important, interesting talk. Thank you. The fifth of 5 of our speakers is not with us in the room, but is here virtually with us. Jeff Hammond from the Global Alliance for Livestock Veterinary Medicines, or GalVmed, is hopefully going to join us on the screen in a minute. And then hopefully the camera will move away from me.
Well,
I'm sure I don't normally have that much.
I can't see any slides.
We can't see you. We can't see any of your slides at the moment, Jeff, but we could at least hear you. Oh, there you are.
So sadly, you can see me, but no slides.
I'm sure the team are there. Right, we can now see at least one slide and your opening slide. Jeff, over to you. Thank you.
Thanks, James. Hello, everybody. I'm really sorry I'm not there, especially as I missed one of Pascal's parties last night by the sound of it. So wish I was there. I'd also take this opportunity to thank EuFMD for arranging this virtual link to enable me to give a talk at almost 3 o'clock in the morning here in Australia. So thanks very much for that. I will attempt to present the ag results foot and mouth disease vaccine challenge project using it as an example of an innovative vaccination strategy. Now I've got to see if this works. It worked before. Yeah, bit of a delay on the slides. Ag results is a $152 million multi-donor initiative that uses a pay-for-results prize competition rather than an upfront grant to incentivize the private sector to invest in high-impact agricultural innovations that help achieve reducing food insecurity, improving household nutrition and health, and increasing livestock productivity. There are a number of sponsors which are listed below on the slide, which include the Australian Government, the Bill and Melinda Gates Foundation, the Canadian and UK governments, USAID, and the World Bank. There are, of course, considerable challenges to controlling FMD in Africa. Firstly, no suitable vaccine addresses all Eastern African regional risks. FMD is a complex disease with high within serotype variation, which increases the difficulty of developing an appropriate vaccine for the region. Vaccines currently available are not effective against all local strains. Secondly, even if a suitable vaccine is developed, the private sector is unlikely to distribute in the region due to current market conditions. In most countries in the region, governments consider vaccination to be a public good and control FMD vaccine purchases. Also, no multivalent FMD vaccine has been formally registered in the region, and vaccines are mostly procured through specialized processes or tenders, since government purchases are often reactive to outbreaks, leading to an unreliable demand and an unattractive market for vaccine suppliers. The Ag Results Foot and Mouth Disease Vaccine Challenge project was officially launched on the 22nd of January 2020 in Rome in Italy. And that's probably the last time I saw most of you. It is an 8-year, almost $18 million prize competition that supports the development and uptake of high-quality FMD vaccines tailored to meet the needs of Eastern Africa in 6 target countries. The project has 3 objectives. Firstly, development and registration of high-quality FMD vaccines tailored for the needs of Eastern Africa. Secondly, increased vaccine production and regional purchases to create greater market stability and to reduce the price eventually. And thirdly, development of a private sector model for buying and distributing FMD vaccines to complement the public sector efforts. To achieve this, a cost share mechanism has been developed to stimulate both public and private sector buyers. The project has an 8-year timeline over 2 phases. Phase 1 is the development phase, which is expected to take a minimum of 3 years. Sorry, the slide has gone crazy, so I will move on to this slide. To be eligible for the cost share phase, the vaccine has to meet several conditions. Firstly, to meet the target product profile standards, the vaccine must be quadrivalent, at least 6 PD50, and contain serotypes AO, SAT1, and SAT2. It must have a 12-month shelf life, 6 months duration of immunity, be DIVA compatible, and have a vial size appropriate for use by smallholder farmers. Secondly, vaccine registration must be achieved in at least one of the target countries, and these target countries are Burundi, Ethiopia, Kenya, Rwanda, Tanzania, and Uganda. The good news is that the first vaccine has already been submitted for registration this very month, so that's a good step forward. The vaccine must be approved by the AgResults judging panel, which is composed of FMD industry and regulatory experts. And the judging panel will review FMD vaccine applications and grant approvals to those that meet these eligibility criteria. Next slide, please. This slide just highlights the location of the regional pools that Don mentioned yesterday, containing FMDV serotypes with specific strains located within each pool. And just to remind you that the pool for Eastern Africa is pool 4. Next slide, please. Foot and mouth disease virus status within Eastern Africa is complex, with most countries in the region being endemic for serotypes AO, SAT1 and SAT2. And you can see from the green highlighting that each of our target countries potentially contains strains of all of these serotypes. In addition, some of the countries neighbouring the project target countries will also contain numerous serotypes and strains. Next slide, please. To further highlight the complexities, this map shows that there are at least 5 serotypes, OA, SATs 1 and 2, and 3 in buffalo, with at least 15 different circulating strains or topotypes. There have been at least 10 reported from Ethiopia, 5 from Kenya, 3 from Tanzania, and 7 from Uganda. And of course, all the surrounding countries have their own strains circulating, which adds to the complexity and highlights the difficulties of controlling FMD in the region. Next slide, please. To help provide a solution to this complex issue, and in order to demonstrate the regional relevance of vaccines entered into the AgResults competition, the World Reference Laboratory for FMD at Pirbright has assembled a panel of regional circulating East African FMDV strains, which can be viewed at the website address shown on the screen, as we don't have time to go through them now. This Eastern African reference antigen panel comprises 16 FMDV strains clustered into 4 serotype sets that are representative of the viruses currently known to be circulating in Eastern Africa. For each vaccine submitted to the competition, cattle sera generated by vaccination will be tested for relevance to the region by virus neutralization test against this representative panel. Each vaccine must achieve a predefined titer for a pass, and the details of which are also available in a paper on the WRL website. Sera from cattle given 1 or 2 doses of vaccine can be submitted for testing, and there is a minimum of a 70% pass rate required for each serotype set. So importantly, this is the first time such a novel approach has been used to assess vaccines before use, and I'd like to congratulate the pioneering work done by the WRL and the RefLab network to progress this approach. Next slide, please. Here are some of the project achievements so far. We've added more than 400 contacts to our database who receive regular project updates, including 31 vaccine manufacturers, NGOs, reference labs, research organisations, regulators and governments. There's been the development and adoption of the TPP as a standard for the FMD vaccine quality and efficacy, engagement with global FMD vaccine manufacturers to ensure that they are aware of the competition rules and are able to participate, engagement with national FMD vaccine regulatory authorities and assessors, collaboration with the UK VMD for harmonising the evaluation of FMD vaccine dossiers with the regulatory authorities in target countries, collaboration with WRL and adoption of the East African FMDV reference antigen panel as a standard to test vaccines for relevance to the regions, development of a public-private partnerships framework to address the challenges in the vaccine value chain, including delivery, cold chain, etc. Introduction of a cost-share mechanism which provides financial incentive for public and private sector buyers to make proactive vaccine purchases. And we've also published a project review article and several GalVmed blog articles explaining the project. Next slide, please. Finally, as a summary to demonstrate that this is a workable and successful example of an innovative vaccine development program, we've shown that manufacturers are willing to accept and adopt the predetermined specific requirements for a vaccine to be accepted into the competition. This is the first time vaccines have been tested for expected efficacy before use in the region, or I think anywhere else for that matter. It's the first time multivalent FMD vaccines have been submitted for full registration in the region, rather than just being used ad lib for emergency use. This now gives the purchaser more confidence in the vaccine, and both government and private sector buyers will be reassured when purchasing a vaccine to aid their progressive control programs. This hopefully will lead to an increased uptake and hopefully improved FMD control in the region. And through this innovative process, we expect FMD vaccine dossiers to be submitted for registration in 3 target countries by the end of this year, and hopefully more in 2023. For further updates, you can please look at the GalvMed or the AgResults homepages, and I would like to thank you very much for your attention, and sorry about the slides being a bit confused halfway through. Thank you very much.
Jeff, that was great. Thank you very much. And perhaps you'd join me in thanking all of the speakers very much for a great session this afternoon. And if you'd like to come forward and join the panel now, please.
I'll stay where I am.
Yeah, please do.
Okay, we're going to start another panel session with our speakers from the second session. So yeah, once again, I'd like to thank all the speakers, and I think it's a sort of a range of topics that we've covered. I was trying to think how we sort of filed, but effectively we've had two presentations on sort of innovative tests. One of them to develop an alternative potency test for FMD vaccines, which is really important, and another interesting sort of development to better predict efficacy and hopefully develop new adjuvants and new vaccines for FMD by looking at the glycosylation profiles on antibodies. We then had 2 sort of interesting presentations from David and Jeff. And Jeff, I sympathise with your pain at 3 a.m. in the morning. Having worked a lot for Australia, I know how that feels. But I think those 2 projects, different ways, are basically reaching the same objective, the objective of authorising or evaluating vaccines to ensure that they meet international standards and provide high quality vaccines for the region, and also improve the predictability for demand to enable manufacturers to invest in their manufacturers. So there's two very different projects, but hopefully achieving the same objective. And then we had a sort of a very nice presentation from Claire. I'm sorry, Claire, I didn't think we met before the actual panel session, but Claire was now looking at better ways of managing animal movements and disease in those animals, and she gave a good example of Marseille, actually. And I was reflecting, Pascal, of your interesting history of the Black Death entering Marseille, and I just wondering whether we should relabel it Marseille flu when we get going forward. But I think that the first question I'd like to pose is to Jeff, actually, and say that given the investment from of $18 million, I think you said, and over an 8-year project, I think the hope, I assume, is to expand the success of this project to other regions. I just want to know if you could comment on the sort of take-home messages for other groups who may be contemplating this in other parts of the world. Thank you.
Thanks, Martin. I think for me, the key to this project is having defined criteria for a vaccine, and then having the vaccine manufacturers agree to meet those criteria so that they're presenting a vaccine that would be useful in the region. I think the cost that you mentioned, the $18 million, is partly running the project through this period of time, but also, it's a word we don't use, but it's to subsidize the vaccines that are produced later down the track, so that the vaccines can be purchased by the farmers or by whoever's going to purchase those vaccines at a cheaper price than would be normally available in the market. So this process will reduce over time so that the amount of money paid for those vaccines will increase gradually as the money from the project diminishes. But I think that's a key issue here, is that we're providing vaccines that have been tested and shown to be hopefully effective in the region by laboratory testing in reference laboratories, so trusted laboratories, and then also reducing the price to the end user by making them available through a cost share process. So I think that's the 2 key things that have appealed to me about this project, and that I think other people could take on board for the next round. But certainly it doesn't have to be that amount of money. You can have a shorter term process.
Yeah, Yeah, thank you, Jeff. And I mean, David, do you want to make any comments on the ag results project?
No, I think that's right. And I think the other thing, Jeff, is the use of that, the WRL panel and the work that Don and the colleagues have done. Because I think you know, if you're going to move from reactive national vaccination, control by vaccination, which is often then just linked to what is the strain causing the outbreak at the time, therefore what is the vaccination that we're going to use. You know, very reactive. Whereas with the panel, it's saying, you know, what are the likely strains circulating in that region, make a bigger market immediately, but also have a vaccine which is relevant to all of those. And other than an incursion of a new strain, it should then cover all of the circulating strains. And I think that's a really fundamental change of approach and increases the predictability of demand.
No, thank you, David.
Could I make one other comment on that, Martin, before we move on? Because it's very relevant that when we first started this project in the region, each country wanted to see a vaccine made with their own local strains. And I think the provision of the panel and the testing process we've abated that problem, and we've convinced most of those countries that it's not necessary to have your local strain in the vaccine, but a strain that has been shown to match those strains and provide protection in laboratory testing. So that's been a very important step forward.
Thank you, Jeff, for that clarification. And perhaps open to any questions from the floor on these projects, perhaps the project surrounding the East Ag Results project. Are there any questions? Yes, Brian.
Thank you. I suppose it's trying to link three talks. So David and Jeff and Aldo's. The question is the current vaccine potency test still appropriate? Because we've got the East African Initiative largely looking at serological, which you were making a strong case for, Aldo, and and we're looking at prime boost as a way of measuring post boost as well, what the serology is. So I could see why a post single vaccination protection from virus infection was appropriate for an emergency vaccine, but should we move on from that?
No.
Ah, well, that
I say yes.
Well,
ask the right question then.
I mean,
yeah. David, you take it.
Yeah, I mean, I think what's difficult with moving away from it is that in all of the in vitro correlates, you have to relate it back to something. And I think what we have to think of the potency test, because it's nearly always done with the homologous virus, certainly for quality control purposes, is it isn't really a test of efficacy, it's a test of quality. And the problem with moving away from it and saying use multiple vaccinations or something is you need something that is uniquely sensitive to changes, as Aldo said in his presentation, in the formulation of the vaccine. So I've yet to be convinced that there is anything that is as good a predictor of the quality of the vaccine in terms of immunogenicity as the potency test. But as soon as you've got enough data, then I think you can bring in vitro alternatives. So that's my view on that.
Yes, thank you, David. I've asked now Aldo to address that, but to add to Brian's question, what I would say is, can we envisage a time where we don't need any animal tests, certainly for the manufacture of the vaccine, where we see good predictors, we have GMP, we don't routinely, as you said, Aldo, use the immunogenicity test now. We rely on a serological batch potency often. So it's a sort of second part of the question. So how important is the immunogenicity test? Because for other vaccines which aren't in the pharmacopoeia, you don't have to have an immunogenicity test. It's up to the manufacturer to decide on what tests they are going to do to sort of validate the efficacy of the vaccine. So it's only those vaccines which are listed in the pharmacopoeia which require that sort of gold standard immunogenicity test. So it is an interesting one, Brian, but I hand over to Aldo, who knows the subject much better.
No, I personally, I think that EuFMD was started because countries in Europe didn't believe each other. So standardizing methods for instance, serology, for how well the vaccines work, that has been the standard for control of FMD in Europe, and that's the reason that we controlled FMD in Europe, and not because we all set different standards. So the standards are very essential, I think, and it's not that when you vaccinate animals a year later, you still should have 75% protection against a needle challenge. But the moment you have 75% or 3PD50 against needle challenge, you know that you have a good vaccine and that it will protect for 1 year if you have a good quality vaccine. And vaccination boost is often offered by vaccine producers, and there are reasons for it, especially if you look at maternally derived antibodies. But it is extremely expensive. A lot of people do not realize that the vaccination campaign is more expensive per animal than the vaccine dose. So if you make a good vaccine and that follows the OIE or the WOA standards and the European Pharmacopoeia, and you have a good quality vaccine, then you don't need a booster. You need a booster. You need protection after 1 shot, not after a boost. But what we see if we have vaccines that do not work that well and we boost, we see often that also the antibody titers drop down much more quickly than if we just only have a good vaccine at the start. So that personally, I think. For economic point of view, a good quality vaccine costs a little bit more, but then you save a lot on vaccination costs, and there have been economic studies for that, so that's a good reason to do it. We have good standards, and leaving those standards for others, then I think that's not the amount of time and animals that have gone into these setting up these standards is enormous, and it's difficult to duplicate that, setting up new standards. So we should not leave them. So I think it's something that we have these standards, and we should just have to do a potency test when we register for registration. Especially if you look at the graphs I showed, for one serotype we clearly needed more antigen than for the other, and it was a huge difference to get the same level of protection. So it's every strain is different, so you have to have set somewhere follow the standard and set that standard, and then you can use surrogates of protection. But I think we can't just only look at surrogates.
Thank you, Coralie, say comment.
I just wanted. Is it working? Yeah. I just wanted to mention for rabies, for example, there is now an in vitro potency assay, but then all the health authorities, the industry work together to really replace the NIH potency test that was really a mess for ethical reasons, of course. And for FMDV, it's much more complicated because, as you said, we've got several strains. It doesn't mean that we can't achieve, but we need a good quality that is really linked to protection. And for now, it's not that easy, but doesn't mean that we shouldn't try.
The difference is that you have one rabies virus, and you have not only 7 serotypes, but so many variants within a serotype.
But we were speaking about sustainability. Is it model sustainable? We have to think about maybe different.
So that I think then leads very neatly, Karel, onto some of the measures that you didn't discuss, particularly the measure of avidity. Which is so different, I think. I mean, not just for FMD, but for other virus families as well. And it's often, because it's rather poorly characterized, I think, often not discussed because we don't understand it, so we don't like talking about it. But actually, I think it's really interesting in terms of discussing exactly this point and the utility of different strains.
Yeah, that was really our goal. It was really to see if we could see some avidity reactions between the different strains. We are struggling developing the tool. So for now, we are working with an ELISA avidity test. We were trying with first for SPR with technology, but and I'd like to go there deeper later on. But we are just set up this tool, and the idea is indeed to see if we can have avidity, good avidity with cross-neutralizing effects of strains. That could go with different parameters, as we said, everything. Immunogenicity in animals, I'm not sure that we could avoid it, but at least potency.
So something for the future then, maybe?
Yes.
Thank you.
Do we have any further questions from the floor at all on any of the topics? Again, it's a quite great range of topics from the project in East Africa, which is then moved towards the alternatives to the immunogenicity test and correlates the protection of alternative methods, in vitro methods. Any other questions from anyone? Yes, we have one there at Simon. Simon.
I mean, this one's for Claire. It's kind of you're kind of raising a kind of an interesting idea of kind of using identification of animals and kind of their sort of monitoring of animals for sort of temperature, movement, sort of and sort of using that as diagnostics for diagnostics. But how likely do you think it is that farmers would be willing to share that data with governments? And kind of would there be enough trust to actually kind of collect centrally all that data and actually kind of analyse it and kind of for the farmers to actually kind of want to participate in their schemes? And if kind of, so how could you actually make that kind of sort of useful and kind of actually incentivise farmers to actually participate?
Yeah, thank you very much. The data ownership is indeed at the very core of what we do. The data always belong to the farmer, which is a very interesting question mark for us is indeed what you are just raising. Because we just started working a bit with the EMA on this question. Farmers are quite collaborative on what they do on the farm. I just think it's really important for them to have clarity on what the governments want to do with their data if they allow access to them. But that's really just like access to their data needs to be consensual, and at the moment there are some farmers who would be interested in doing that. We see that, for instance, for like breeding perspectives, for instance, or genetics. But I'm raising another question mark to your question mark, which is are governments ready at the moment as well to harvest this kind of data?
I think there's a big international variation in the answer to that question, which of course you know very well in raising it. And I think that the situation in Europe is probably very different in relation to the farmer acceptability, the farmer acceptance of doing this kind of work in North America, or some parts of North America. But I think it's a really interesting and important question, which is certainly for the Northern Hemisphere. We're perhaps a little bit away from that in other parts of the Southern Hemisphere, excluding Australia, dare I say.
Yeah, also we do have very good examples as well of this kind of technologies being used in South America, because the more, like the bigger, the more the width of the territory is also a very big adoption factor. When you cannot control, when you cannot check visually your cattle every day, it's very useful to have this kind of monitor systems, because, well, it really improves your quality of life as a farmer, your productivity, your efficiency. But indeed, having a penetration on other markets of such technologies from the Southern Hemisphere is something we are really, really interested in. We always say starting with electronic identification is always a good start. We do have some examples in North Africa, for instance, of countries interested in adopting such technologies. But I think it's, well, once again, I'm really not working in the same field than you, so I'm sure it's being debated a lot. But for me, it's also a question of economical development in general as well, like equality of chances, access to technologies. So it's a question I cannot really answer myself, but yeah, they are very good questions.
Yeah, thank you. We have another question at the back, Don. Thank you.
Yeah, thank you very much. I guess the roles are reversed from this morning, so maybe I can ask Aldo a question. Or perhaps for some comment. I mean, you're aware, of course, of the work that we've been doing together to try to bring together the heterologous results, where we're trying to define serological cutoffs for protection. And I'm just wondering, maybe you could share a little bit of the challenges there in the idea of bringing together the data, and perhaps that there isn't very much data in this space, that's worth saying, but there's also the challenges of coming up with a single unified model that explains all of the data to actually generate that correlation between antibody titer and protection. And then the final point, of course, is, you know, remembering again, you know, the project that we did that was funded by EuFMD that brought together a number of European laboratories. There's also this additional challenge that when VNTs are measured in one laboratory, they don't always even approximate to the same value that comes in another laboratory. So just interested in your thoughts, Aldo, since you're sitting up there, regarding this concept of moving beyond just homologous responses, but into the space of heterologous responses.
The analysis was an interesting part because we tried to set up some standards for the ECHO results, and in the end we saw that it was very difficult to have a standardized cutoff, and we ended up with different groups, and that clearly shows that there is differences. But the biggest, if you look at the differences between strains, that's one thing, but the differences between laboratories, it's even bigger. And then one of the things that we're trying to do at the moment, it's also one of the questions that Kees van Manen asked, is set up control sera that are related to that 50% protection. But that's something that we still have to validate. We have made some for some strains, but we haven't looked at it in different laboratories that well. We did something already more than 10 years ago together with Purbright and Belgium for Omenissa, and then it was also probably also the differences in the potency tests. So these things are difficult, and then one of the things in the analysis was what we did was just make sure that we just selected one cutoff, and for a group of viruses and a different for another group of viruses, and that there was always differences between viruses. That was the most frustrating part of it, and that's. But then still, if you have some cutoffs, that will help. But then that was only tested in the World Reference Laboratory. So the differences between laboratories was also huge, because some of the sera came from our laboratory, and when we compared the titers found in Purbright and our laboratory, there were also huge differences. So that's it remains challenging, also looking at titers.
Can I put the question back to Aldo and Don? Because I think that this area of between laboratory comparison, between virus comparison, is not unique to FMD, and actually it's most characterized in human influenza, human seasonal influenza, where it's a huge industry of serology and antigenic characterization and sequencing on a scale that is almost unimaginable, I think, to this room, and this has been contested by some, but one way of that this has been brought together is by the use of antigenic cartography, taking a multi-dimensional analytical approach to this. And Don, I mean, you were involved in Al-Ludi's work looking at this for at least one serotype, but I mean, is there not more scope for doing collaborative between laboratory analyses on a more routine basis if you really want to use this? If you really want to use serology for both potency testing and for antigenic characterization of variation?
Perhaps I can start. We every year we have a proficiency test, and we in fact, David Mackay once started with it, making standard sera, but it was a little bit lost after he left. But standard sera is something, rabies is an excellent example, where you have just a standard serum and you have to have so many neutralizing antibody units, and then you're protected. So that's the reason that we're also looking at that for foot-and-mouth disease. Yeah, and then the
cartography.
It's very difficult. We did it once. We did an immunization study with 10 type A strains, and we immunized 5 cattle with each of them, and then we looked at all the results in ELISA and in in the neutralization test, and we used then principal component analysis. But it was very difficult to get non-overlapping results, and completely different from what they find with influenza. And but yeah, influenza.
But it's a question of scaling it up, I think, and you gain confidence, but the more analyses that you do like this, I mean, it's thinking that something. This didn't work the first time for flu either.
No.
Right, so, and I think it's important to remember that. No, thank you.
Thank you. And I think we have 2 questions, virtual questions. Buda, do you want to?
Yeah, thanks, Martin. Yes, first of all, I would like to acknowledge our online audience. We have had like 840 people connected to the session today, so that's really great number. So we really appreciate their participation. And here are 2 questions. The first one is to Aldo. Why FMD vaccine quality is dependent on 146S virus particles rather than 12S particles?
Yeah, that's a good question. Why the body responds better to 146S, and I believe that it's the repetitive presentation of epitopes, but Brian is probably better in that. But that we clearly see huge differences, and it's clearly if you vaccinate with 12S, there has been potency tests done in guinea pigs, and you needed 70 times more 12S to get the same protection as with 146S. So,
I think it's related to the GH loop that is really linked to the particle size and the structure. So, and which is full of neutralizing. We've got a well-known neutralizing epitope there. So, I don't know if.
Oh, but the GH loop is also present on the
12S. Yeah, but the 3D structure, I think, is more.
So we've done a huge amount of work, which unfortunately we can't publish, on mapping the antigenic surface of FMDV through structure analysis. So I'm not contradicting what you're saying, Aldo, I'm confirming really that it's that repeating structures that engage with B cells. There's also some epitopes that fit at the 12S interface, so they do not exist in 12S. But there's also epitopes, and we've got, not the one you're thinking of, but we've got a llama antibody that binds specifically to 146S. That binds solely in 12S. It only binds when it's in the capsid. As soon as you disrupt the particles into 12S, that epitope changes and the antibody doesn't bind. So it's not simple, and there's quite a complex change in the surface structure beyond the repetition of epitopes when the particles come together.
No, that's a good point. We've done now in collaboration with China cryo-EM structures of particles with complexed with llama single domain antibodies, and we see antibodies that are very close to the twofold axis, and we thought, oh, the reason that the 146S specific is because probably they react over the twofold axis, but we see them just above the twofold axis. But the moment the virus particle falls apart, that structure changes and it doesn't work. But also some antibodies that are 146S specific that exactly react with epitopes around the 5-fold axis, which is still available in the 12S particle, but still the conformation of the protein changes and they do not react anymore. So that's exactly what you're saying. Yeah.
Excellent. Okay, well, we have one, I think we've run out of time, but we have one final question I think we've got probably time for, and we sort of end the session on a sort of regulatory issue. David, it's directed to you, and we started on regulation. Perhaps it's fitting that we end on this subject again about modern vaccine platforms. And it is my impression that the modern vaccine platforms are inherently safe. Is there a lack of confidence in the safety of these platforms, and is the fear justified from the regulators?
No, I mean, I don't really kind of agree with the premise that there's a lack of confidence in the platforms. Of course, there's a massive range of different platform technologies. You know, it ranges from vector vaccines through to a standard way of constructing mRNA vaccines. So it depends on which platform you're talking about. But I think that certainly both within Europe and within US, it's moving very much from that case-by-case evaluation of whatever was put together to looking at the inherent safety of the platform. Then once you've established that, it becomes much easier. So I think that's the right approach, and I don't think there is a kind of inherent difficulty. I think, if you look at, compare examples that have come where, say, the platform is a well-established vaccine itself, like the HVT for turkeys for Marek's disease, then it becomes very easy to use that as your platform because everyone's very familiar with it and it's been administered to billions of animals before you even start.
Thank you. I think James may want to make a comment, but I will make just a comment on my personal observation of having the COVID vaccines. When they got back to the UK and had the AstraZeneca vaccine and then started seeing the news of people dying of blood clotting issues and strokes and things like that, and you're going to select the vaccine platform that you think is safest. So I think that I'm fearful of having any vaccine. If you're healthy, you don't really want it. And I would say is look to Brian perhaps. Is the adeno vector model sort of proven that it's not sort of fit for a sort of routine vaccination programs and could only be used in emergencies? I don't think it's not administered anymore, is it?
In England.
Yeah. So just someone perhaps any comment or perhaps.
But that's a strain issue, not a safety issue. That we've moved on in terms of the strains being used in the UK. And I think that what you say you don't want to take a vaccine. A point is that a very clear old vaccine paradox is the safest way to be protected is for everyone else to be vaccinated and you not to have one. But that can't work collectively, so we all have to be vaccinated for highly transmissible diseases. That's how they work. And so I was very pleased to get my boost a week ago, in contrast to you.
Yes, well, that's good. Probably the time to end the discussions, and I'll hand over to Boo, who's got some housekeeping issues to mention.
Yes, thank you. We are running out of time. Just one housekeeping message. Tomorrow's last session, session number 6, starts at 2 p.m. So there is a change directly in your apps if you're using EuFMD apps. So you will notice that there is a change. So please be here at 2 p.m. For the last session tomorrow. So with that, I get the floor back to James.
Yeah, so can I thank all of the speakers? Can I thank you for making the session as good as it was? But please, in particular, let's thank the speakers for really great talks this afternoon. Thank you.
Yeah.
And thank you to the online audience who we can't see.
We're going to have a beer now.
Okay, that's it.
Thank you.